Research Center started off as an infectious disease center, but along the line, it's added non-communicable diseases to its focused areas.
WACCBIP is providing something to local and regional students that we probably don't have in many places within the country or within the region. Facilities, financial resources, as well as an environment that encourages student learning and research. And that has significantly increased students' output in the Department of Biochemistry, Cell and Molecular Biology.
WACCBIP has a whole range of collaborators at the institutional, national, regional, and international levels. These collaborations include the Noguchi Memorial Institute for Medical Research Ghana, the Department of Biomedical Sciences at the University of Ghana, among other departments, the Ghana Health Service, and the Ministry of Health. The center also has collaborations with institutions within and outside Africa.
Currently, there are about 6 labs in the center, and these labs do research based on the faculty who is in charge of these labs. We have a malaria group that has 3 different units. There's an immunology unit, there's a vaccine development unit, and also a transmission unit in there. We also have a molecular biology lab that has 2 faculty in charge there. One does work with African animal trypanosomes, and the other one, who is myself, works in Purulent ulcer and antimicrobial resistance. We have a huge virology group. They do research, anything that has to do with viruses. So they do HIV research. They even look at some bat viruses and gastroenteric viruses. More recently too, we've added a COVID lab, and this lab does routine testing for COVID positivity and negativity. And we also look for seroprevalence across the population. So people who have been prior exposed to COVID, but maybe have been asymptomatic, so they don't know.
Hello, how are you doing today? Oh no, I need a little sample of your blood for the lab. It's not painful, just a little sample of your blood. It's not painful, it's not painful. Blood. Okay.
Check. Check.
Is everything okay?
No, please, there's a problem.
What is it?
I don't know how to communicate to the patients that we need a sample of your blood for the lab.
No. You see, that is why we need sign language interpreters in all health facilities. Don't worry, I'll handle it.
Okay.
Bring her to the lab.
Okay, please, I'll do that. Look, please, we are here.
Oh, good. I hope you are well. Don't worry. We are here to help you. All of us. To do that, we need a sample of your blood. A sample, Don't be afraid. Your blood, small sample, okay? For our diagnosis. You see, please, it's okay. Take it easy. Take it easy. Don't be afraid. Oh my God. How am I going to explain this to her? It's okay. It's okay. You take her back to the ward. You'll be fine, okay? You need a sign language interpreter.
Hey, stop looking at them.
Who told you I'm interested?
I can see it in your eyes. See?
Doris, they are deaf and dumb people.
It's true. Ah,
I hear these people are friends though.
how do you even introduce them as your friends?
You know, every physical condition has its medical cause. It has nothing to do with curses.
Oh, hello, sir. Your money?
Oh, I see. You are not deaf?
No, I'm not deaf.
That is interesting.
Yes.
Aha.
Boss.
Oh, okay. I will need your assistance to communicate with a deaf patient.
Oh, okay. Which hospital?
Legon Hospital.
Wow. Actually, I've just been transferred from the Alpha Clinic to the X-ray department.
Really?
Yes.
Wonderful. That makes my work easier.
Yes. I think I can help.
Yes.
Okay.
Please.
Okay.
This boy can be cases for this.
Let me see how far.
Show me.
You!
Hey! What is this?
Masha!
Ah, what is it?
Warn your son! Warn your son! Or else you will see what you don't want to see and hear what you don't want to hear. How many times have I not warned him not to play with my son? Or come to my house?
Oh, Ejapia! Ejapia! Ah, what's wrong? If Kofi plays with your son, are we not from the same clan?
Hey! Don't you know there is a case on you and your husband?
Me and my husband?
Yes!
Me and my husband?
Yes!
Very good. My husband is in.
Did you say curse? All right. We'll see who that curse is on.
Deal with him! Curse him! Deal with him! I'm chipping. Come on. Wow.
Doctor, we can't find Amma.
Where is she?
I don't know. We've looked everywhere.
What?
Yes.
In that critical condition? Oh God. This is unbelievable. We have to find her. We have to find her at all costs. I'll handle it.
Thank you.
My friend, thank you very much for coming. I'll call you when she turns up.
I'll shout. What more tests do you need? We know the people who did all the antenatal tests. How can you tell us our child is deaf? We will solve our own problem. Let's go.
Excuse me, sir. Is there any problem?
What I say is final. We are not going to any pastor. We are going straight to the village.
I am not ready to take my daughter to the shrine.
No one in my family is deaf. In your family too, there is none. So how can you tell me my daughter is deaf? We must look into it. This is purely spiritual.
What about going to do the test? So the doctor can determine how to help her.
It is my daughter who is deaf. What do they need my blood for?
Our blood. I think that is what will help her.
Blood is sacred. I am not giving my blood to any doctor. I will call Papa at the village to arrange a meeting with the diviner.
Thank you.
Chikere. Sumanga. Baba, Jamato, Baba, Sumaga, Jamato. Oh, great diviner, eye of the gods, the great owl whose gaze pierces through the thickest shadow and the darkest night. There is a deity that must be appeased in your family. She has been abandoned and has come with a vengeance of deafness. You must pay the due and purge your child. 3 rams and 3,000 Ghana cedis.
Your Highness, can we pay in cash?
Oh, the gods can take cash, even mobile money. Chikere, pala wezu.
Hey! Something is happening to her.
Don't worry, my daughter. The spirit is leaving her. Mali.
He said she was going to get better when we get home. But now, her condition is worsening. Please help us, please.
This is highly demonic. She needs deliverance. I can feel it in me that there's a bad spirit inside this child.
Prophetess, can you deliver her?
That is exactly what I am going to do. Let us bow before the Lord. Jesus, we trample on every serpent and scorpion. According to the word, we rebuke every demonic power of high places, every altar that stands against the authorities of Jehovah. Be brought low. Jesus' mighty name. Jesus' name. Jesus' name. Oh, Father. Jesus. Jesus' name.
Kojo, she's not getting better.
What should we do now?
Let's go back and see the doctor.
Okay.
Let's go.
Doctor, thank you very much for the explanation. We got it all wrong. We now have a better understanding of what genetics is and and its implications on deafness.
How can we thank you, doctor? Many people out there are not privy to this information.
Especially the people of our village. Doctor, there is a deaf community in our village. Is it possible for you and your team to come and explain this to the people?
Oh, that's a very good idea. It's an opportunity to reach out to the community and educate them. We will. Why not?
Thank you very much, doctor. Then I'll quickly arrange for the meeting.
Thank you. Thank you. Thank you. She's as normal as any other child. Just take good care of her. Okay? It's okay. Don't need to cry.
Ago! Nana and the council request tomorrow morning. Nana wants to meet everybody at the village square. The great Asparrow has spoken. Ago! People of Asempa, Nana and the council request your presence. Nana wants to meet everybody at the village square. The great Asparrow has spoken.
Nana and Council welcome you all to this urgent but very important meeting. Nana want to cut straight to the point. Nana, speak.
People of Asempa, the issue confronting us is a very serious one which needs to be addressed. I'm not an expert myself, but our forebears have told us over the decades the mysteries surrounding deafness in our community. Much as we have tried, we've not been able to find answers to this problem. But today, our son Kojo and daughter Agnes are here with some medical officers from Legon to educate us on the subject of deafness and its possible causes. Dr. Wo and Dr. James, you are most welcome to this community.
Thank you very much, Nana.
Please, you have the floor now.
Thank you very much, Nana.
As part of our work as clinicians and researchers, we investigate what makes each person unique and different. We do this by examining your blood sample. How many of you here have ever given their blood for testing?
Blood. I can't give my family.
Blood.
My family.
No, blood is sacred. We don't give blood. Yes.
It will never happen like that. I don't give my blood.
It is very important to give blood to be tested because every information that is used in building our body is packaged as genes, and this can be isolated from our cells in our blood sample. This we inherit from our parents. Please, can you bring the chart? You have to look on this chart. It will help you understand it better. Please. Our bodies are made up of billions of cells. Inside each cell is our genetic material. Chromosomes are the packaging of our genetic material. We usually have 23 pairs of chromosomes, and we inherit one copy from a mother and one from a father. If we unwind the chromosomes, we can identify the genes. Genes are the basic building blocks that makes proteins. Genes are like the blueprints for making a machine, and proteins are the individual components of the machine. Proteins makes us who we are and help us grow and function properly. Our genetic materials is made up of our DNA. Deoxyribonucleic acid is the very long chain of 4 chemical letters: adenine, guanine, thymine, and cytosine that combine to make our genetic code. We all have some variations in our genetic code.
Genes variations are like spelling errors, where for example there's a T instead of a C. A variation in the gene that makes it faulty or not working properly is called mutation. Faulty genes are caused commonly simply through chance. They have to occur in one individual for the first time at some stage. They are not related to anything the parents could have caused or prevented in any way. They occur because the process that replicates and divides their genes so we can pass it on to the next generation is complex, and mistakes occur. For each gene, we have two copies. One copy comes from the mother and the... Hello? Oh, okay. Good morning. Welcome to day 2 of WACCBIP Research Conference 2022. I hope you all rested well after a very marathon session yesterday. Like I promised yesterday, today is a bit lighter, so and you'd have more chances to interact with the invited guests and amongst yourself to get to see how you can foster more and more collaborations. So a few housekeeping announcements before I invite the director to introduce the keynote speaker for today. Please remember that once you are in this room, you always have to wear your nose mask. Please keep it on. We are still not out of the woods yet with COVID-19. Also, I wish we could project this, but please remember to engage on social media. Any of the things that stand out to you, that resonate with the theme for the conference that you want to tweet or put on Facebook, please remember to always tag WACCBIP along with the tweet. And the hashtag for the conference is hashtag WRC22. Today, this morning, not long after the keynote lecture, we have a very interesting panel discussion, which I think you'd all be interested to partake of. It's on biotech innovations and startups, and we are really excited about this year's edition. So do stay tuned and engage. So without much ado, I want to welcome the director, Professor Gordon Awandari, to introduce the keynote.
Good morning. So we are getting our keynote this morning from within. So one of the things we do with the keynote speakers is we try to have a balance between internal, regional, and international. This morning we are going to listen to one of our own, Dr. Peter Kwashi. Dr. Peter Kwashi is a senior research fellow at WACCBIP and a deputy director in charge of research here. He did his bachelor's in biology at McGill and did his MPhil and PhD also in McGill in experimental medicine. After that, he did a postdoc in Toronto for a year or two. How many? 3.5 years. Okay. 3.5 years. After which he moved back home to join WACCBIP, first as a Creek Africa Network Career Accelerator Fellow, which is a very competitive fellowship scheme, which he won, and that's what brought him back here. Since he's been back, he has gone through the levers and established himself here as one of our major research leaders at the center. Since COVID struck, he used his expertise in HIV research and repositioned the center to conduct research into SARS-CoV-2 virus, which we have significantly made an impact on in the Ghanaian response to the the COVID pandemic, and also provided very relevant data to the general global knowledge on this disease. Ladies and gentlemen, it's my pleasure to welcome Dr. Peter Kojokwashi.
Thank you, Prof, for that kind introduction. I actually put a few slides of introduction in here because even though WACCBIP knows me, some of the people listening might not know me. So I'll just start with that. And what I'll be presenting on is insights and opportunities from SARS-CoV-2 research. So this is some of what I'll present here today. Will be presented in more detail by Keseho, and if you look at the posters, you might see some snippets of similar information. And what I really wanted to get across with this talk is how we took advantage of this pandemic and really tried to move fast. And I was sort of positioned to do that because when you work in HIV, you have to move fast. So basically, that was the mentality that we brought to the game. And when the pandemic hit, myself and Dr. Bediako really tried to get everything together and make sure that as a center, of course, supported by Prof, because we didn't have any money for some of this research, you know, we just cobbled together what was available, and we started. So let me start. Okay, so like I said, a brief background. Okay, do you want to advise? Okay, brief background about me. Next slide, please. It's not working. Okay, so I did my master's degree at Concordia in Montreal, which I focused on mechanistic enzymology, and that was in biology. Then I moved on to McGill. Next slide, please. Did my PhD in experimental medicine at McGill, and that was in the lab of Mark Weinberg, who is since passed on. And that's Francois Barry Sanusi, who got the Nobel Prize for HIV research in 2008. And that's me over there, in case you didn't see me. So what I focused in there was mostly HIV work, HIV integrase, reverse transcriptase, looking at drug mechanisms and resistance. And it was a departure from my master's, but next slide, please. It was successful enough that I tried to do something which was a bit more adventurous, which was go and crystallize the protein that I'd been working on for a while. And I spent 3.5 years doing that. Actually, my timeline originally, I said if it doesn't work in a 1.5 years, I'm going to quit and do something else. And it was always close enough. Anyone who knows HIV field knows that integrase was the last structure, one of the last protein structures to be solved. And just when we got the crystals, and then we were basically, I spent almost a year trying to solve the structure. It was a low resolution structure. And then Peter Cherepanov at the CRIC got a bunch of structures which really told the story that we were trying to sell. And then I cut my losses. Well, I'd been talking with Prof. Arundari for a few years about coming back. So came over to WACCBIP, started working on, next slide, please. Next slide, please. started working on looking at HIV genotypes in West Africa because I realized that not much work on HIV had been done in West Africa. So that was the work we were doing, and that was funded by the Crick African Network. And that's the work we were doing when COVID struck. So we had the opportunity, all the fellows had the opportunity to transition, and I decided not to, but Crick African Network was kind enough to let us keep our fellowships even as we did COVID work. So now I'll go into the COVID part of it. Next slide. Let me see. Okay. Yeah. So just a brief chronology. In March 11th, the first positive test for COVID-19 was confirmed in Ghana, and March 12th it was made public. And initially, like I said, we mobilized to form a WACCBIP COVID-19 response team. We initially helped with testing at Noguchi, and then we decided to develop a comprehensive research program. And of course, it's not as if some of it was thought of in the beginning, some of it came as we evolved. And the first thing we did was sequence the circulating viruses. I believe the first sequencing result we got was in May, and second one in June, and we submitted that manuscript in July 2020. We also wanted to do diagnostic ELISAs. Remember that in 2020, there weren't many diagnostics available. So that was one of the things that we did, and looked at evaluation of RDT, rapid diagnostic tests. A bunch of tests were coming into the country with suspect, all of them were claiming 99% sensitivity and specificity. But because there was a misconception that you could use those for diagnosis, it was important to evaluate it and show that you can't use them for diagnosis. And this was the team. This was during the short lockdown we had in Ghana. We were, as samples were going to Noguchi, we were getting blood samples, and we were banking blood samples for future work. And so this is basically the WACCBIP COVID-19 research program. We have serological studies. Mostly we started with seroprevalence because that's what eventually funding was there for, as well as sequencing. And then, so this is just basically looking at prevalence, transmissions, infections, and then we moved on to trying to do more immunological studies as well as virological studies. And of course, in the span of this time, I can't really talk about everything. Okay, so like I said, as samples were going to be tested at Noguchi, we were getting blood samples and banking them. So this is just saying if a sample is taken for qPCR and it was positive, we would bank it as positive. If it was a follow-up sample, we would bank it as a follow-up sample. And that's how we got some of our early samples. And at the same time, we started, once we had those samples and we had evaluated some kits, we started doing community seroprevalence. That is, to try to estimate the levels of the virus in the community or the levels of exposure. And this is just a picture of us in the market. You can recognize this gentleman seated right there, Dr. Bediako, and that's myself and the entire team. Next. So this was, normally when people do seroprevalence studies, they do household studies, but an idea that our director suggested is that if you go in public spaces, you go to the markets, Laurie, stations, and then you go to the malls, you're going to get the entire cross-section of the population, right? And that was the strategy that we used. And so in, I believe in August 2020, we had the first evidence that actually in Ghana, we were seeing high-level spread of SARS-CoV-2. At that time, when we started this work, we expected to see maybe 5%, 2%. And what we ended up seeing, I'm showing the numbers here, but if you look at the percentages, it was actually on average 19% across board. And then because we had that data, we had the preliminary sequences, we had the preliminary seroprevalence studies, we managed to get some funding to do some more work. And the first was Rockefeller and followed up by FCDO. And of course, TBEL funded a lot of the early work. And so I'm not going to present the sequencing in depth because Collins will talk a lot about it. But as of June, we had sequenced and deposited about almost 2,000 samples, 2,000 whole genome sequences in GISAID. And that had allowed us to track the progress of the outbreak in Ghana right from 2020 all the way to June. And he will talk a lot more about that and how the different waves were led by different variants, and sometimes not the same variants that were leading waves around the world. And these samples were collected from all over the country. So the data that we garnered was quite representative of the Ghanaian outbreak. And that culminated in, this was our second sequencing manuscript. I think when this was, I had one slide that I skipped because this was not responding. But basically, go back, please. Forward. Okay, yeah, so this was a summary of a lot of the sequencing we did from 2020 all the way to, I believe, September 2021. And then we also collaborated on an Africa-wide sequencing, Africa-wide genomic manuscript led out of South Africa. And also, due to some of the sequences we had deposited, we were contacted by Public Health New York about potential intra-host recombination. And that has also been published in Nature Comms. And then, like I said, we had extra funding to do the seroprevalence. And the Rockefeller funding was to do seroprevalence across 3 countries, Ghana, Nigeria, and Burkina. I'm only presenting the Ghana data here, but you can see as of June to December 2021, we had high-level seropositivity across the country. And you remember that the first work we did in 2020 was around 20%. And now we're at 54, 56. Some places are quite high, but in those places we didn't have the sample size to really say anything. But you can see that in Greater Accra region, which is the epicenter, it was quite high. And we were able to show that these peaks were really linked to high level to the waves that we were experiencing in the country. You could see that basically, you could see that basically this was the transition between the D614G variant to the second wave, which was Eta, Alpha, and 827. So in the south of the country, there was Alpha, which was mostly brought in through air travel. And in the north, you have Eta and 827, which is which was very similar to what was circulating in northern Nigeria and some parts of Burkina Faso at the time. So we think that those were probably brought in through the land borders. And then you see that when Delta came, seroprevalence went up to 40%, and Omicron, it went all the way up to 80%. And this is very telling because right after Omicron came, January, February, cases dried up. Testing, there were almost no positive cases in the country for about 2-3 months. And when you you break this down by weeks, if this thing would work, you see that seropositivity peaked in January and then actually started to decline. So in the field, even when you were getting positive tests, it was very faint. And we think this, coupled with what Collins will tell you, is the variant that actually restarted the 5th peak that we're in, which restarted the 5th wave that we're in right now. Because when antibody levels decline, and this is on a population level, right? As the antibody levels are declining in the population, that means that their susceptibility to new infection is coming up. At the same time, we know from our sequencing that the 4th wave was driven by Omicron BA1.1, Omicron and then BA1.1. And data out of South Africa has told us that BA1.1 does not protect against BA4, BA5. And actually, in May, when we had our 5th peak, it's predominantly BA4. So it makes sense that after we had about 2-3 months of almost no cases in the country, at one point we had only 4 cases in the entire country. Then it started picking up in May. So that made sense. And this is just summarizing what we saw during the Delta wave across Burkina, Ghana, Nigeria. And this is during Omicron wave. So what that told us is that, 1, the virus is circulating in the country and it's circulating freely. 2, people are not dying. Contrary to what modeling papers are saying, that thousands and thousands of deaths, you go to the hospitals and they're not overflowing. Even in the peak of Omicron, where we had the highest peak of recorded cases in the country, treatment centers were empty. And we know this because we get our samples from treatment centers, and it was very difficult to get samples during that time. So, going back. So, there had been published reports that IL-6, IL-10 were all important measures of disease severity. And so this is something that we wanted to do. And next slide, please. And along the way, we got some funding to do some immunology work. The first one was to do some serological assay development. And that was also a bit early in the pandemic. That was through Cambridge Alberata. Next slide, please. And right after we got that, there was a group out of Southampton that had, again, this is very early. There were no commercial antibodies that had managed to raise some antibodies against nucleocapsid in rabbits. And we worked with them to evaluate nucleocapsid ELISA. And we used that to show, and that was published in Journal of Infection. Next slide. So basically, this is Western blot showing that the nucleocapsid ELISA, they used a bunch of other human coronaviruses to test for specificity. And you could really detect show specificity with the sera from the rabbits against SARS-CoV-2, and that was used to develop the antigen ELISA. Next slide. Yeah, and basically we use that to screen patients here and in Southampton, and we could show very good correlation between the samples done in Ghana and in Southampton. Basically, to summarize, the assay was most useful at CT values 30 and below, as can be expected for most antigen ELISAs and antigen kits. And this is something that we were looking to roll out on a larger scale for mass screening, but it does take a long time to do the ELISA, and at that time the cost of antigen RDTs had come down and they were quite good. So, so basically, why am I presenting this part? Because at the same time we got the plasmids to produce this antigen, and we had the plasmids to cleave it and do everything. So I decided to use that to do our antibody ELISA, because ultimately we will have to be measuring antibodies. And so we produced the protein in-house and confirmed by Western blot. Next slide. And then we optimized it using negative samples and SARS-CoV-2 positives across the 3 waves. And next slide. And we're able to show that there's a clear difference between positives and negatives. Of course, there is some cutoffs. And then we're able to look at different waves of the pandemic and show that we have increasing levels of antibodies in samples collected from each wave corresponding to the last wave. And this is something that agrees with the assays that we did in Luminex. We did Luminex using antibodies, using baits which were immobilized with spike and nucleocapsid, and we saw that in each wave there were increasing levels of antibodies. So the assay that we developed was quite good correlation between that and Luminex, of course, at a much lower cost. Next slide, please. But back to that question, why is the death rate so low in Ghana? We wanted to look at, next slide, we looked at the immune system from some of our patient samples, and we basically looked at cytokines that were expressed in those plasma. And the takeaway is that it appears that we might be seeing an attenuated cytokine response system. And this is just showing symptomatic, asymptomatic, pre-COVID negatives, COVID-19 negatives during the pandemic, and non-survivors. And you can see that in the asymptomatics, for the cytokines which are supposed to be really involved in severity, the levels are quite low. Even when you compare it to pre-COVID samples, which are much older and which would be more likely to be degraded. And then if you look at those non-survivors that we had, they are much higher. So we think this may be indicative that the larger population of people in Ghana don't tend to get the cytokine storm that is reported. And those that die, at least early on, do seem to show evidence of cytokine storm. Of course, for these patients, we didn't have multiple samples, but for some of them we did. Next slide, please. And we're able to show that unlike the cytokine storm trends that have been reported out of Japan and out of Europe, we actually see a decline in these cytokine levels. And this is something that we submitted the first results along with other work. And we are still working on this. We're now stimulating the cells to see whether it's true. But it also matches up with the cellular populations, because it does look like maybe we do have an attenuated T cell response. I'm not really going to talk about it because Keseho will talk about it. I just want to give a snapshot. And then along the way, we got a Gates grant, Global Immunology and Immune Sequencing for Epidemic Response. And that allowed us to do even more work. And I'm just going to present a little bit. Next slide. So there was a slide, I think because of this, it skipped. But when we first started evaluating RDTs in Ghana, what we saw is that people who had asymptomatic infection, about 40% of them never showed any signs of antibodies. And this is just a viral infectivity assay in using patient plasma to inhibit viral infection in Vero E6 cells. And for this particular one here, we're using the original virus, not the original virus, but the D614G virus. And what we see is that in wave 1, the plasma from wave 1 is not able to inhibit that virus. And wave 2 had a bit of inhibitory activity against that virus. But that also brings me to say that maybe that's why when we did those kit validations, we weren't seeing the high-level positivity or high-level sensitivity that we were expecting, because there were no antibodies that were in the samples. And then also we saw that vaccination status actually increases. Being vaccinated actually increases the chance that you have antibodies, which is has already been established. And this is ongoing work that we hope to complete in the next few months. But it's quite interesting to see how the variations occur across the waves, and we're going to be adding in the different viruses for the different waves, Eta, Delta, and Omicron, of course. But cell culture does take quite a bit more time. So in summary, we built a rapidly expanding research pipeline. We showed that the SARS-CoV-2 situation in Ghana evolved rapidly, and likely over 80% of us have been exposed to SARS-CoV-2 since 2020. We've developed some homemade assays, showed potentially attenuated T-cell responses to SARS-CoV-2 infection, but then the question is, like, why would T-cell responses be attenuated? And go back, forward. And I'm just showing this to say that there's quite a body of literature showing that malaria infection can modify your immune responses. And we are in a malaria endemic region, as is much of West Africa. So next slide, please. So we just wanted to see, there was a hypothesis that maybe malaria is protecting. And so here you have symptomatic plasma from symptomatic individuals, and this plasma from asymptomatic individuals. And what we see for 4 different blood stage malaria antigens, that we have higher levels of antibodies in the asymptomatics versus symptomatics. Next slide, please. This is CSP again. Next slide. Next slide. And this is RH5. I believe that's what's in the vaccine. So this is, again, work that we're doing. We're now stimulating the cells to see if it's, we'll see some stimulation, some confirmation of that. And then we've also submitted, next slide. Next slide. Yes, we've also submitted some grants to try to extend this work, do single cell sequencing, and really try to understand that on a more mechanistic level. So this is one of the key findings that we have. It's something that we had thought about, but now we actually have, I still say it's still preliminary evidence that malaria is really indicative. I wasn't expecting it to be quite as consistent as that, but we'll keep working at it. But along the way, we also had opportunities to create research partnerships that we can continue to grow for different work. Next slide, please. We applied for a grant, ARUA, which is, we became the West African hub for vaccine, the West Africa Vaccine Hub for ARUA, that's Association of Research University, African Research Universities Alliance. And next slide. And then we applied for a networking grant, a network grant where we are hosting WANIDA, which is the West African Network of Infectious Diseases, ACEs. And it's through that network that we got some of the partners for the Rockefeller project, and we leverage that partnership for projects as they evolve. Next slide, please. And of course, we have in-country networks across the country, Ghana Health Service, Christian Health Association of Ghana, and the different universities who have supported our work. Next slide. And we have to thank the participants and, of course, our funders. And thank you for your attention.
Okay. We have about 2 minutes.
Yeah.
Where's the microphone? Microphone? Any microphone?
So it's really interesting. So my question would be, perhaps malaria infection could be protecting COVID. So I'm not sure whether any African countries are malaria-free, but if you gather the data from malaria-prone area, right, and then compare with the Western countries, it does not. Do you see, better correlation? I guess you need a lot more work to do. I'm just curious.
Yes, that's a good question. So actually, if you look at the mortality data in Africa, we tend to think of Africa as one lump thing. But if you look at the mortality data, it's not uniform. And it's mostly in West Africa where you see that the severity is quite low. And yes, ideally, if we can compare Ghana and, say, UK, which is one of a grant that we submitted along with Let Out of the Creek to compare people in the UK, Ghana, and West Indies for what is it that is protecting. We strongly think it's malaria, but it could be something else, or it could be a combination of factors.
Right, so the genetic component is going to be different between the Caucasians and African, right? So
yes, so we've done some work there. I didn't present that. That's why I think it's a combination of factors. That's still quite preliminary, but it does look like there is at least some genetic component. It's not as, I don't think it's as strong as it could be, but yeah, because I mean, they're Ghanaians, different. Well, Africa is very diverse, so okay, I'll leave it there.
Yeah, so I think just to add that basically when you superimpose the mortality data on malaria, you see quite a good correlation between mortality from COVID-19 and malaria endemicity, an inverse relationship. And so, like you were saying, in Africa, if you look at the sub-Saharan African belt, you see that the the impact has been much less there in terms of severe disease and mortality. Whereas if you look at the northern part of Africa, in southern, where there's almost no malaria, you see that the impact was higher. You disagree?
No, if you don't mind, I think I'm very quiet when I'm just listening because I want to really just have what we are saying supported by a real well-designed study. And I should say that I have been fortunate to be just undertaking such study funded by Gate Foundation in collaboration with Liverpool School and London School. This is a large study where we are studying the correlation between malaria and COVID. So the design is really such that, and it's involved two African countries, Burkina Faso, where I'm working, and Kenya. And also are trying to combine, to niche a kind of clinical trial on that, where malaria cases with COVID are treated with some malaria treatment, just to see if there are some interaction. Data are under analysis, that one quite, but it looks like there is some interaction, but not yet really well described. But at least I think by the next month we should be able to provide more data supporting that findings.
Yeah, and there's some data out of Senegal too, which seems to be confirming the same thing.
It's interesting, very interesting thoughts you've got there. It seems to me that possibly you should be looking at regulatory T cells that might be malaria activated and then going on to suppress these COVID-specific responses to dampen down these pro-inflammatory cytokines, but T-reg cells might be the answer.
I think what I personally think is about, I think the mechanism is rooted in, what you call
attenuation,
learned innate immunity. So, there's, when you look at the work we've done on malaria, you see that in areas where there's high exposure to malaria, subsequent malaria infections, there's a much muted cytokine response, which suggests that they've been tolerized to the inflammatory antigens. And we think that there's a similar mechanism going on.
Okay, thank you. Nice talk. So, at the beginning of your talk, you mentioned somewhere that there is some in vitro work done using blood sample. So I want to hear a little bit more how successful was your SARS-CoV culture using the blood samples. This is my first question.
Okay.
The second one is for the seroprevalence. Did you investigate just the IgG or IgG and IgM? So if you investigate also the IgM, I just want to know what was also your attitude when you have somebody who is IgM positive. The last one is regarding the higher seroprevalence. So did you try to, let's say, look at the presence of antibody in old sample, let's say before COVID-19? Because I'm wondering if there is little bit cross-reactivity with other kind of, let's say, disease, which can explain maybe the high level we are getting in Africa. Over.
Okay. Your first question, I didn't quite understand it, but I'll answer. I'll start from the last question. The answer is, before we do anything, we validated these kits using negative samples, and we don't think what we are measuring is cross-reactivity at all. Actually, IgM was very rare, usually less than 3% or so. It's mostly IgG that we're measuring. Of course, when you get high levels of IgM, you can't really trust it as much as you can trust the IgG. And like I said, we validated it, and you can see that there was a progressive increase as the pandemic went, and it matched. You can I think I had done one presentation where you can actually align it with the waves. So we don't believe, when you do, because there were some early papers which said it's all because of cross-reactivity. But if you do an assay where you're measuring antibodies and you don't control it, you don't properly control it, or you're just measuring positivity, you're going to get positivity. But the question is, have you controlled it? So a lot of the early papers, which had about maybe 100 samples in one hospital, or, it was, I believe if you have a hypothesis and you want to prove your hypothesis by designing your experiment in a certain way, you'll get what you want. And so, yeah, the evidence doesn't support that.
Okay, thanks. Thanks for the interesting talk. Very cool work you're doing there. Now, you mentioned the models predicting this high mortality, which of course is like the simplest model that don't account for demography. So what you really see is the risk of severe infections really goes like exponentially after 65 plus. Now, if I'm correct, like the population 65 plus in Ghana is about 3%. Compare it to Germany, where it's like 22%. Yeah. Now you cannot even one-to-one compare it, because like they are way older. If you go to compare the percentiles, then it looks way older in Germany. Now, if I just take the case fatality, essentially it should be it's about 6 times higher in Germany, yeah? But that estimate is kind of rather conservative, because it doesn't take into account the full demographic, but just taking 65 plus as one bunch, which it definitely is not. So you wouldn't expect too many deaths anyway. Yeah. So I'm just wondering, if you account really for demography, yeah, how strong is your effect actually? And adjusted for demography, would you see would the inflammatory response really correlate with a kind of malaria incidence? And particularly if you look at other countries, like say at Kenya, yeah, where in Kenya usually you don't have a lot of malaria, it's just across Victoria Lake where you have a bunch. Yeah. Now, there they actually saw a lot of severe cases. Now, what's the difference? Or what is would this mechanism has any genetic component, particularly to West Africans? And how can you actually adjust for demography? And maybe permit a second question. Like I don't believe it that there were, between the waves, there were no cases at all. Yeah,
oh, yeah.
It is just they were not detected.
Yeah, there were cases.
But they should have been there. Yeah. Okay.
Okay. Sorry, maybe I missed. Not that between the waves there weren't any cases. It's the one that I was referring to was recently, between the 4th and the 5th wave, because testing facilities dried up, and positivity went almost to 0. As in the tests that were actually being conducted, there were no positive cases for a period of time. Now, as answering the demographic question, what I haven't shown is that actually most of our positives were in 40 and above. Most of our positive people. So in terms of the risk, it still follows the worldwide trend. But in Ghana, if you talk to clinicians, they'll tell you that most of, right now, the only thing that they look for is not necessarily age, it's comorbidity. Because they didn't find, you have people who are really old, of course, they will die at a higher clip than the younger, but it's not an increased risk over having the comorbidity. So comorbidity is the only thing that they really track. I don't know if you had a lot of questions in there.
Yeah.
Yeah.
Yeah. But I think it's a very relevant question.
Yeah.
About what's the impact of the age. But I guess if you look at within Africa, that's why it's always good to compare within Africa. You look at countries in the north and countries in the south, some of which have similar age demographics as those in the sub-Saharan malaria belt. You would see that there's still a difference. And so I think it's beyond just the age factor. Anyway, Daniel, you want to clarify something? Okay. Daniel is a member of our team, so maybe he's going to provide some additional perspective.
So actually, when you adjust for age, 60 plus actually have much more infections than any other age group. So it's not different from elsewhere.
Mm-hmm.
The older population actually get more infections. They show more seropositivity. Younger people show less seropositivity. And then about the malaria relationship profile that I referred to. So there's a paper from WACCBIP which shows, so you have COVID mortality, not infection rates, because mortalities are more likely to be reported than infection. So COVID mortality across Africa, and then malaria incidence across Africa, you will see a very direct inverse relationship. So where COVID mortality is high, malaria incidence is very low. There's a paper we can look up later on. Yeah.
Thank you very much for the clarification. So let's appreciate So we will quickly move to the next section, which I believe will be chaired by Prof. Leon Stark. You protest? It's Nancy. Oh, Nancy, sorry. Is she online? And Reverend Dr. Goyon. Is Nancy online? Is she online? No, I think. Because she's not in the country, I know that, right? Or you're here to represent her. The husband is here, but she herself is not here. She's online? Okay. So she's online. So she's going to join online. But Reverend, you can take the chair and moderate things from the room. Thank you very much.
Right, we have Collins Maranga, who is going to give a presentation. And this presentation will be for 15 minutes, and then we'll have some time for interaction.
Good morning, everyone. Yeah, so today I'm going to talk to you about the spatiotemporal dynamics and genetic diversity of SARS-CoV-2 across the COVID-19 waves in Ghana. So I think since the pandemic started, we've seen a huge number of deaths just within a span of 2 years. About 6 million people dying across the world, and almost 600 million confirmed cases. And most of the cases were in Europe, Americas, and Africa, I think, saw the least confirmed cases. And most of these cases can be attributed to how fast the virus has been evolving. And you can see that in the initial early virus, which was like Alpha, you can see the number of mutations are a bit fewer. And this kept increasing on Delta variant. And you can see that Omicron, if you just look at the spike protein alone, you are seeing a lot more mutations compared to the initial variants. And across this time, I think we've seen huge spikes in number of cases. The cases always go down, and then all of a sudden you see them increasing. And that's where we talk about the different waves. And in Ghana, I think we've experienced about 5 waves, as Dr. Kwashi talked about. And all these different waves, you can see Ghana has always lagged behind during the different waves. And now we are having the 5th wave, which I think started around March, April in other countries. But in Ghana, we started seeing it towards the end of April, May, and June. And in total, I think in Ghana, there has been almost 1,400 deaths of people carrying, and this can be attributed to these different waves that have occurred. The main question that we set out to do was to always wonder, is it the same variant that is circulating across the world that is causing the different waves in Ghana, or we are actually having our own different variants in circulation? And to do this, we decided to collect from way earlier on in the pandemic. We collected data, and the most important thing I think was for us was the time of collection, the age of the participant, the travel history, and the location. And the travel history was important because we wanted to know whether the people arriving at the airport or crossing through the borders are the ones responsible for the different variants that we'll be seeing in our sequencing. And we did sample processing and sequencing by mean ion sequencing, and we generated the genomes. And we've always deposited most of our data sets in GSAID for the rest of the world. And apart from just asking the question about which variants are circulating, we also decided to look at what is the distribution of the different variants across the country, and this necessitated the collecting of samples from most parts of the country. And as I'll show later on, we also studied the trends, what are the variant trends in different parts of the country and across time, as well as look at the genetic diversity of these variants, and also look at the phylogenetic analysis. And we do this using already established next-strain pipelines. So far today, I think as an update of the data that Dr. Kwashi presented, we've sequenced about 2,300 genomes, and in Ghana, we've done about 2,274 genomes. And we've also sequenced some samples from our neighboring countries, that is Nigeria and Burkina Faso. And I think we are continuing these efforts to sequence samples from these countries. And you can see that in Ghana, I think we've had samples from almost every region across the country, but most of the samples have basically come from Greater Accra, I think because most of the cases, or Greater Accra has a higher population, as well as most of the confirmed cases actually came from Greater Accra, as well as the Ashanti region and Western region. And we also looked at samples that were arriving at the airport. I think this one we did about 110 samples, and these were done in January 2021 and around March and June 2021. And if just to start off in terms of the variants that were circulating, I think out of the 15 samples that we helped sequence for Burkina Faso in collaboration with Burkina Faso partners, you can see that most of these samples were collected at around around September, October 2021, and the Delta variant was more circulating, including the subvariants of the Delta virus. And for the Nigerian samples, which were collected way back in 2021, I think the highest variant that was circulating was the Eta variant, as well as the L3 variant, and some samples that we collected in January 2022 had the Omicron variant. So coming back to Ghana, in the Ghanaian samples, I think most of the variant that was circulating was Omicron. So just to say Omicron as well as its subvariants, we've labeled it as BA star, and because there's a lot of subvariants of Omicron currently in circulation. And the second highest overall in Ghana was the Delta variant, followed by the Alpha variant and the 318 variant. And from what you can see is that basically almost 70% of all the variants that we've seen in the country are basically coming from variants of concern. And these are variants of concern that have been seen across the world as being responsible for most of the cases in the countries. And you can see that most of the travelers that came to Ghana came from all different countries, from the United States, from Europe, as well as our neighboring African countries. And just to mention that some of the samples that we screened and detected variants from travelers actually hadn't been detected in some of those countries as of the time that we detected through our sequencing. Now, to just look at the distribution of the SARS-CoV-2 variants across the country, you can see that we have some variants that were concentrated in different regions, and some of these variants include the A27 as well as the Eta variants, which were mostly in the northern regions. These are colored by orange. You can see that northern east, northern region, upper east, upper west, they had concentration of 827 variant. And then regions such as Ashanti, Savannah, Volta region, Greater Accra, you have a mix of a lot of Omicron, Delta variant having the highest proportion in terms of number of cases. And what we actually see is that through time, most of these variants of concern that have caused highest number of cases is that what I'm showing here is a plot that is showing the density of mutations across different variants. And all these are variants that we've seen in our samples to date, all the way from the A1 variant that was initially, as well as from the B variant, and all the subvariants of the B lineage. And this, I think the B variant was defined by the D614G mutation. And you can see that this is basically showing the number of mutations across all the different variants. And you can see that all the variants that have a high density of mutations were the variants of concern, or at least the variants that caused a huge number of cases. And this corresponds to the number of samples that we sequence from those lineages, as well as showing that it's not that all the samples come and they have the same number of mutations. So you see that some samples have very fewer mutations, and probably across time they end up having a high number of mutations. And this could be because probably there's a high rate of evolution of this variant. And where I'm highlighting here is the Omicron variant. You can see that we have the BA variant, which is basically the highest in terms of number of mutations, almost over 60 different mutations in some samples, and the high density is around 50 mutations across most of the samples. And And what we see is that if you put these samples on a phylogenetic tree, is that you realize that the Omicron variants, because of the high number of mutations, they are the most diverged. And you can see that we have Delta variants, the Delta variants also highly diverged. But you can see that all these different lineages of viruses, they're actually a little bit completely different, but there's a bit of sharing of some mutations. And I think there's a tend to believe that Omicron has a higher mutational fitness. So this plot is just showing the number of mutations on the y-axis over time, and we are seeing that as COVID is getting a high number of mutation, if you calculate the mutational fitness score, you get that Omicron is having a very high mutational rate compared to all the other variants that have come before it. And if you look at the mutations that they have, you tend to think that there's a bit of recombination that is going on to generation of some of these variants of concern. Because if you check on Omicron, there's a lot of sharing, almost 10 different mutations between Omicron and Delta variant, about 8 mutations with Alpha variant, and some sharing with Beta variant and all the other subvariants. This is just the spike protein, and you can actually see a lot of sharing of mutations between the Omicron variants compared to the other variants. And it's interesting to see that most of our samples, they have the spike D614G mutation, I think, which was one of the defining mutations for most of the variants of concern from early during COVID-19. And there's like a cluster of mutations here, I think, around the RBD binding domain, whereby you have a high frequency of mutations, I think, including the N501Y, E484, which has been changing from A to K to Q, and it's been responsible for most of the different variants of concern that we've seen across time. And if you put those variants across time, you get the sense that they are changing from, because during earlier on, we had all these kinds of different minor variants that were circulating. And one of the major variants that was circulating at the time was the B1 lineage. And over time, you see that we had introduction of the B.11 lineage during around May, June 2020, August 2020, September 2020. There was an interplay between the B11 and the B1 lineage, and as well as some lineage like the B1.485, but you can see that it went out and we didn't see it after around November 2020. There was expansion of the B11 lineage during that time, and after some time we started seeing the introduction of, we actually detected alpha variants in travelers way early in time, and after that it dominated most of the circulation around January, February, and March in 2021. And after that, we started seeing B1318, which I think for us in Ghana, we thought maybe this was a variant that we should monitor, because it dominated transmission from around March all the way to July or August, thereabout, when we had the introduction of Delta variant. And you can actually see how, because of the mutation sharing between all these different variants, you just realize that, okay, this new variant has just acquired maybe an extra 2 or 3 new mutations, and now is dominating circulation. And Delta variant, I think, was one of the most devastating variants across, especially in Asia. And then later in November 2021, we detected Omicron in circulation, and it was majorly dominated by the BA1 lineage around January 2022. But since then, you can see that Omicron has just taken over. There's no other variant that is circulating. It's just the Omicron subvariant or variant as well as its subvariants. So, and if I put this in a spectrum, you can actually see that the interplay between the different variants. So there's a point where you see that there was sharing between the Eta variant, which as I said earlier, was circulating in the northern region, and the Alpha variant, which was circulating mostly in the metropolitan areas like Greater Accra and Ashanti region. And you can see that there was a lot of interplay between them. And then we had the 318 variant, which came and took over transmission across the country. And then later on, the Delta variant, and now the Omicron is just going. And just to break this down a little bit, you can see these are the Omicron subvariants that we are seeing just in May and July. Between May and today, you can see that we have a little bit of BA2, which I think was responsible for most of the cases in other parts of the country, in other parts of the world. But in Ghana, I think initially we had the BA4, which we detected way back around May in May samples. I think we had a BA2 case in April, but you can see that it hasn't taken root in Ghana. But then the BA4 variant, which took root, and then later we had the BA.41, which has actually taken over the circulation out of all these 256 samples that we've done between May and today. You can see that the BA.41 is actually the highest circulating variant. But just to point out, I think there are other subvariants such as the BA.5, which we detected also in May, and there's its subvariant, which is the BA.52, which is also in circulation, and BA.521. And these are variants that we should keep an eye on because they've been responsible for, or they are said to be highly transmissible or have some level of immune evasion. And then we have recently we've detected the BE1 variant, which is also said to have some mutations that are responsible for some level of immune evasion, and its subvariant, the BE.1.1. And now we've also detected the BF5 variant, which is also a new variant that we should keep an eye on. So it will be interesting to see in the coming months which variants are actually going to stand the test of time, because I think in Africa we've seen, because most of these BA2 variants, which were responsible for most of the cases around the world, I think they didn't take hold in Ghana. And just the same as we had seen with some variants such as the 318, which was high cases in Ghana, but not most of the other parts of the world. And 827, I think we have to keep an eye on some of these subvariants so that in the coming weeks we see which one are responsible for the waves. And I think data from laboratory experiments and epidemiology studies, they suggest that there's a greater role of immune evasion than increased transmission of Omicron subvariant. I think this is something to think about. So just to summarize, I think we've had about 50 unique SARS-CoV-2 lineages detected since more than 50, since March 2020. And 7 of these, that is the Alpha, Delta, Beta variant, Iota, Kappa, and Omicron, are responsible for most of the cases or most of the sequences that we've sequenced. And they've been taking turns to dominate local transmission in Ghana. And there's a significant diversity of all these variants of concern, with some of them having more than 20 to 65 mutations. And we have some mutations that are unique to Ghana, and this indicates some level of ongoing transmission or local evolution and suggesting significant transmission. Now, it's basically inevitable that a new variant will emerge, because seeing how they keep kind of recombining and coming up with a new variant, and with different degrees of transmissibility and immune evasion, the question is then, when are we getting new variants? Or will the new variant have more severe disease in Ghana, or should we be worried about it? And will the existing vaccines protect us? And I think we should do a little bit more, and I think most of the studies that we are doing here would assist in those efforts. And we have to continue doing sequencing so that we can monitor the spread of these Omicron subvariants, and this will basically inform disease control measures as well as development of new therapeutics. I think we have some research outputs. I think Dr. Kwashi has talked about it, and I would like to acknowledge some of our funding sources, all the way from Rockefeller, Ghana Health Service, the University of Ghana for providing us with the environment to do the research, all the COVID-19 teams across the country who have contributed, especially the testing laboratories where we get our samples from, all the way from Noguchi to Yemachi, as well as all the Ghana Health Service facilities that are dedicated to contribute samples for sequencing. Thank you very much.
Thank you, Colin. So we want to have some time of interaction if there are any questions. And I'm wondering if we have any online. Yes, ma'am.
So I'm just curious whether the incidence of the Omicron spread is something to do with the vaccine, because all the vaccines made until that time is against the earlier version, earlier variants, right? So although we think that maybe the current vaccine can protect from the Omicron, I think that the efficacy is much lower. So maybe that's why the Omicron took over, because other variants could be at least handled by those vaccinated individuals. Again, I do not know the vaccination status in Ghana, but any correlation with that?
Yeah, I think there's about 19 million vaccine doses that have been given in Ghana to date. And I think studies from South Africa suggesting that, some of these Omicron variants have some level of immune evasion, or they kind of, the neutralizing antibodies do not actually work very well on this Omicron variant. So yeah, it's true. Yeah.
Hi, my name is Jerry Joe Harrison, and so when we talk about the viral population, we always refer to them as a quasi-species. And so because we are not doing single virus sequencing, we are doing an average of the nucleotide pool that is always there. And so following all the analysis, the question I want to ask is, do you see co-evolution of the various variants that emerged? And could they, if they are independent, could they have been present from the beginning, but because the nucleotide pool was dominated by others, we did miss these things, and then over time, as they begin to build up, we begin to have them take control. So the simple question is, do you see co-evolution of the various variants over the course of the years that the pandemic has been with us? Thank you.
Yeah, I think a quick answer to that one would be, if you look at the mutation, the number of mutation that is shared between the different variants. So you realize that, let's say for B1, then we had the D614G mutation, and then we have the N501Y mutation that is present in almost every of the new variant of concern that we are seeing. So we would not assume that they were there, they just didn't pop up early enough. But I think it's just an evolution and some level of recombination between some of these variants of concern, which tend to have, For example, Omicron shares about 11 mutations with Alpha variant. It shares almost 12 mutations with Delta variant, and shares even mutation with Beta variant. You see, so I think there is just like a continuous evolution or kind of introduction of new variants through different mechanisms that I think has.
But what if these are mutational hotspots within the genome that are actually emerging over time, so that we've seen the earlier ones were the ones that were very easy, the hottest, and then over time we've seen these the hotspots increase, and so therefore it may not necessarily be dependent on the previous variant, and that we should probably look more into go to the single virus level and see what are the various species that are there, and whether this can inform how our tracking happens in the future. Because for all you know, we are seeing mutational hotspots spring up as the virus acquires various mutations. And so therefore, by going one step further, we get ahead of the virus and probably even redesign our vaccines. What do you think?
Yeah, so I agree. I think there's a level of intra-host diversity whereby you tend to have, I think, some level of even more than one virus infecting, and this can contribute to what you are talking about. So I think, yeah, I agree. It's good to look at single virus every infection level and see the level of intra-host diversity, and that's, I think, one of the things that we've been trying to do with our data, is to actually look at each infection or each sequencing of every sample. Is there a level of intra-host diversity and a chance of having multi-clonal infections or different variants within the same sample, so that which could span the rise of new variants. Yeah, so I agree. Yes, sir.
Yes, there is one.
Hello, Collins. Very insightful. I wanted to ask if you could comment on how these variants have played a role from infectivity to mortality, because I realized you gave a kind of timeline and trend line from when each variant arose. So can you comment on that, on how that played a role in infectivity and mortality?
Yes, so I think the most infectious variant, or the one that caused a lot of mortality, was the Alpha and the Delta variant. And this was compared to the initial variants, the B1, the B11, I think there was a little bit low mortality in Ghana. But when we started having these variants of concern, that is the Delta, I think some variants of concern didn't even take hold in Ghana. For example, the Beta variant, which was responsible for mortality in South Africa and other parts of the world, did not take hold in Ghana. But the introduction of, let's say, Alpha variant, the Delta variant, I think those two variants were responsible for most of the mortality, or most of those 1,400 cases of people who died in Ghana. Yeah.
Thanks so much for the talk. Regarding this immune escape hypothesis and so on, maybe one should really take a close look at the clinical studies also of the vaccines. Yeah? If I take the study from Biotech, that study showed that a full vaccination cycle protects from infection, actually. Yeah? That's what the study showed. Not a flaw with that study was, that was under rather low incidence conditions, because they did that study like in the first half year. Now, assuming that actually you don't get sterile immunity, but you just reduce the risk by, let's say, 70%, yeah, of getting infected. Then if incidence is high and you have a lot of infectious exposures, you're still pretty sure to get infected. Yeah? And that's what we see with all these variants that are being more and more infective. Now, definitely what we should see is a mutant can only invade or take over if it either is more infective than the others, or if if it's net more infective, given the immune status of a population. And now with the BA.4/5 variant, we see them pretty much spreading everywhere. We don't see evidence that the BA.4 or 5 is any kind of more contagious than the other. Yeah, so it seems to be kind of random where, you know, BA.4, BA.5 started first, and in those countries they're spreading more. But maybe can you comment a bit on that? Because like if actually it has to do with the vaccine, we should see very different patterns across the globe, but we don't really see that, right?
Yeah, I think we will
okay,
bring this to an end. And thank you, Collins, for
all
the
right.
presentation.
We'll discuss during tea break.
I think for the next session, we'll have our director moderating.
Great. So we thank Collins for a very elegant presentation. I think we can agree that Collins is now joining the superstars at WACCBIP. So. So well done, Collins. Now we're going to one of the highlights of our conference, which is the panel discussion. We at WACCBIP, we have had an interest in scientific enterprise over the last few years. And that's because we recognize that the work that we do here, both in training and capacity building and in research, we realize that there's the need for, you know, off-takers, you know, who can take on our products in terms of students we've trained and also in terms of the knowledge that we've generated to implement that if it's policy direction or develop into a product if it is research innovation. So we have taken a keen interest in the scientific enterprise landscape in Ghana and Africa as a whole. Part of it is also driven by the World Bank. I see Sylvia and Adeline here. The World Bank have always insisted that they want us to send our students to industry for internships. And we have always insisted that there's no industry. As far as we are concerned, the industry that we want to send our students is non-existent in Ghana. So we've made it a point to try to encourage our trainees to create or help create this industry that we think is necessary for our sustainability and for advancement of science and innovation in Africa. So some of our own have started to take bold steps towards this direction. In addition, others have preceded them, and others are also in the pipeline to take these steps. So today we've, you know, we've been lucky to have about, you know, 4 of some of the major players in the scientific enterprise ecosystem in Ghana. And we would like to have a panel discussion about some of the driving factors that led them in that direction, and also what those who are still planning to go there, including myself, what we can learn from them as the leaders in this field. So I think one is joining online, right? Roberta? Yes. So is she online? She's online. So we'll start with those
Yes,
here.
I am.
Okay, good. Great. So we would, I'll call on those who are here to join the panel. So first is Mrs. Margaret Williams. Margaret Williams is Senior Clinical Research Associate and CEO of Magan Clinical Research Organization. And she'll sit next to me. I'll take, if there's one lady, obviously, if there's one lady, she has to sit next to me. The other is one of our friends, Mr. Lord, or Dr. Lord Anthony Bassing, who is he's a scientist, an engineer, innovator, and entrepreneur. I think a lot of us have heard about his exploits in the Ghanaian innovation space. He's a product of KNUST, but he recognizes that there's only one university. So he has sought validation of his degrees here. So he has attended a workshop in WACCBIP before, where we started to cleanse his degree. Subsequently, he has been at Noguchi to further validate his degrees. So Lord, welcome, sir. And then our own Yabi. So you all know Yabi. Yabi is a research fellow. Dr. Yabediak is a research fellow here, and he's now a CEO of a new, growing, and globally popular biotech called Yamachi. And I will take credit for part of the name because I think he got part of that from my local language. So I take credit for part of the name. But Yao is one of the people that WACCBIP are very proud of because he's part of the cadre of scientists that we take credit for having brought back onto the continent. Peter is another, Lily is here. These are people who we think that WACCBIP played an important role in giving them the comfort to come back to the continent. So we want to take credit for this brain repatriation. Yawaniwa is also at the back there. He was in Singapore minding his business, and then he saw that WACCBIP was doing something exciting, and he also packed and came back. And I think none of them have told me that they've regretted. So if they have regretted, they haven't told me. So so as far as I know, they are all happy that they came back. So we are proud that we have helped repatriate some of this talent. And then our fourth panelist is joining us virtually, and she's Ms. Roberta Annan, the global impact investor, a renowned one of course, and a founder of Impact Fund for African Creations, or Creatives. And she is coming from the angle of how do we raise funds to do this type of thing. So it's very, very important aspect of this discussion. So ladies and gentlemen, you're welcome. So because because we want this to be as informal and interactive as possible, I'm going to start by going around the table. I would like each of you to basically take 5 minutes and tell us how you got into this scientific enterprise space. And we mean, why did you decide to go and make money instead of being in the lab and working for free? So, so we want you to be really clear with us about what drove you to take these steps, and basically what should people who are thinking of doing the same, what are the boxes they need to check to be sure that they are ready for this challenge. So I'll start with you, Margaret.
Thank you so much. It's such an honor to be here. I'm actually a bit hesitant about starting by saying that I also got my first degree from Tech. After that,
thank you.
Yeah.
It's okay. It's okay. We'll sort you out.
Okay, now I'm even more afraid to say that I got my MPH from Legon. So, okay, so I had my first degree in biological science from the University of Science and Technology in Kumasi. And from there, I did my MPH here at School of Public Health. And I guess during my interview for the MPH, one of the questions that I was asked was, why didn't I want to go into bench science? And my answer was very simple. I'm very squeamish. I knew that I wanted to contribute to health, or the improvement of health in the African region, or across the world, really. But I knew I didn't want to deal with, and these were my exact words, the icky stuff. So I wanted to find a way to contribute. And through my MPH, I was able to venture into the arena of clinical trial management. So when we talk about clinical trial management, it's basically about how new investigational products, including devices, are developed and go through testing to ensure that they are safe and they are effective when they are used by human beings. And I know most of the scientists here know more about this than me. Before we get to the human phase, we do preclinical testing, where you do the pharmacodynamics and pharmacokinetics to ensure that toxicity levels are assessed and rendered safe before you do the first testing in humans. So before I get into the science bit of it, with discussions with people that I had worked with, I realized that there's no reason why, as an African public health person, we cannot have our own organization to manage clinical trials in our own country, in our own setting. Because nobody can understand the various norms and cultures that we have, the things that you have to circumvent to be able to enter a household or a community, apart from the knowledge that you already have from your MPH background or clinical trials background. So from there, I developed this company, Macro, which provides clinical research monitoring services, mainly across Africa, but extending to wherever else we get projects from. And in addition to that, we didn't just focus on hiring people locally in Ghana. We have local CRAs based in all the countries we work in. Burkina Faso, Nigeria, Liberia, Sierra Leone, you name it. We've got CRA presence there. Because all these trials come into our region. Because let's face it, when it comes to infectious diseases, you're not going to have a lot of infectious diseases to check in other parts of the world as much as you have in Africa. We don't like to boast about that, but that's the reality. So if we are able to, and at the risk of saying what a lot of politicians say, capacity building. We're building our own capacity because who can do it better than us? And that was the reason why we decided to set up this company. And the additional benefit is we actually have partners in Francophone Africa, so that irrespective of where the projects come in, whether it's Anglophone or Francophone Africa, we can provide clinical trial monitoring services.
Great. Thank you very much. So how many employees do you have, if I may ask?
So currently we have 20 CRAs across Africa, in some of the countries I mentioned, and all of these are consultant CRAs. So based on the projects that come in and the location of the clinical trial, we're able to determine which CRA will best fit or is closest to the area where the project is being held.
Thank you very much. Okay, So I'll go to Lord.
Okay. All right. Thank you very much for the opportunity. So for me, my journey started somewhere in 2012. I had decided that I was working at Kofanochi Teaching Hospital at the time. And one of the things I had realized was that when you ask somebody to go and do a lab, the person would go and would not come back. And then after a long time, the person comes back and you're like, why didn't you go to do the test? They'll tell you we didn't have money. And when I did a survey of all the labs in the area, I realized that the fees were quite exorbitant. And then, naive as I was, when you check on the internet and you look at the reagents, so I didn't understand why they were charging that high when the reagents were not that expensive. So I decided that I wanted to set up a lab and reduce the cost so that everybody can be able to do a lab. And then I started it. But within 2 weeks, I realized that I had made a very terrible mistake. Because one, even importing the reagent alone, and then the number of middlemen you have to go through to be able to get it. I realized that those who were even charging, say, LFT, people were charging at the time about 20 cedis. They were even doing very well. Because I realized that if I charge 20 cedis, I would not even be making profit. So I put my lab on hold. I started within a month, I had put it on hold. And then I was fortunate to come here for the workshop, the cell biology workshop. And during the workshop, I had the opportunity to talk to some of the invited guests who had come to talk to us about cell biology and the rest. And then one of them actually asked me whether I'd heard about lateral flow and lateral flow training and all of that. I hadn't. So I started to Google search all of these things. And I became fascinated with point of care tests, being able to develop some of these point of care tests myself. I didn't have the knowledge. I didn't know where I was going to do that, but I was very interested. In 2014, I had the opportunity to, following the workshop that happened in Oguchi, that happened here at WACCBIP, I went to Mali, where they did the same workshop. This time was malaria. And then over there, we were giving mentors. And one of my mentor was working with the Army Research Institute. So again, I expressed my interest in developing point of care tests. So she got me a few contacts and then a lab in San Diego that was into teaching people how to develop point of care tests. So between 2014 and 2016, I had the opportunity to go to this lab and then learn how to develop point of care tests. So when I came back in 2016, I started to develop these tests. So I did a 3-in-1 test for vaginal infections. I did a malaria test kit. I did a pregnancy test kit, hepatitis B. All of these are approved by the FDA and are on the market currently. But then that wasn't enough. There were new diseases that were coming through, and these you could not use the normal lateral flow test to diagnose them because they needed molecular tests. And we all know that before you can set up a molecular lab, you needed equipment, you needed the personnel and all of that. And around that time, I started to hear about molecular points of care tests, LAMP and other ones. So I was fortunate to get a scholarship to go and do a PhD in biomedical engineering, focused on developing molecular points of care tests. So again, but at that time, I had set up a system where I had enough staff to take over the company and manage it while I went back to the US. In 2019, I was able to complete. I had finished developing a molecular point of care test for the disease yours, Treponema pallidum, subspecies petinui, which is currently undergoing clinical trials. So I came back. In fact, when I finished in January 2019, I had a job with Abbott. And it was good. The money was good. And for a few minutes, I considered forgetting about Incas Diagnostics and focusing on Abbott because there I had almost everything I wanted. But then after a while, I decided that no, the whole purpose in building my capacity and doing all of this was to come back and set up this company and be able to produce not only the point of care test, but also molecular point of care and then subsequently other tests that will come out. So I came back in May 2019, and we've been at it since. We are moving right now. We do both the test. We also do the normal lateral flow test, the antibody antigen test, and then we are right now working on a test to use CRISPR, molecular point of care test. We are also moving into antibody production ourselves. We procure all of these, and the exchange rate and all of those things, it makes it quite difficult. So we've been working on being able to do all of this ourselves. And currently we have about 37 full-time workers in the company. This year we are hoping to employ 5 more people, especially to help us with the antibody production part. So that's how the journey has been.
Great. Thank you very much. So you are doing product development, but are you also offering the testing services?
No, currently we do not do the testing services. We just do the product. So our main our main services is training. We train people on how to develop points of care tests and other things. But our main area is the product development.
Good. All right. I'm going to go to Roberta next, so that she's not she doesn't turn off the microphone and go to sleep. So, Roberta, since you are not in the room, we have to make sure you are engaged. Otherwise, you may switch off and go and take a nap. So, please let's hear your story.
Thank you so much. In fact, I'm really honored and privileged to have been invited to be part of this panel discussion. So just a bit about my background. So I'm actually a scientist as well by education. So I have a BSc in biochemistry and biotechnology from Carleton University in Canada, and a master's degree in biotech from Georgetown University in Washington, D.C. And during my master's education in the States, I was exposed to 3 facets of biotech. The first being the science, which is obviously obvious research-based. The second being entrepreneurship, and the third track being IP, so intellectual property, and how to kind of look at the legal aspects of the biotech. And while in that program, I identified that my passion, although I really loved the science and wanted to be in the lab, I was more of a people's person, and I was more of a risk taker in the sense that I wanted to start a business, raise capital, just come up with different interesting ideas in the biotech space. So I was really, really interested in that facet of the program. So shortly after graduating from that program, I got the opportunity to work in fundraising. So everything kind of adds up to where I am now. I got a job. In fact, I applied for United Nations Biotech, and it was very difficult to get into it. But I started as a consultant first as an intern, and then later as a consultant. And I started working on building fundraising strategies for programs within the United Nations system that obviously would have maybe a bit of donor money, but then needed more money to help sustain the project. So I'm sure many of you are familiar with how development funding works. So normally when there's a project, there's a bit of funding that is dedicated to that, but the funding usually lasts for a few years, and then sometimes the project actually needs to run a bit longer. But of course, the UN or like the donors are not able to put more resources into it. So my role as a resource mobilization officer was to go around and talk to the wealthy people of the world, like the Rockefeller Foundation, the Ford Foundation, the Bill and Melinda Gates Foundation, Carlos Slim Foundation, just to mobilize and bring more funding to support. And my remit was really on climate change, environment, and energy groups. So I was working mainly on climate change adaptation projects around the world. And after a few, because I always had the interest in coming back to the continent, because during the role that I was playing as raising money from very wealthy people. I know that many of you are aware of the Sustainable Development Goals and that there's a 1.6 trillion funding gap, right, to meet the SDGs in developing countries. I know the importance that private capital has to play in addressing socioeconomic challenges, especially in developing countries. And you know that a lot of the wealth is concentrated I mean, if you look at the combined net worth of the world's richest people alone, right, it is multiple countries' GDPs combined. So there's a lot of importance in that kind of relationship. And I wanted to be able to draw some resources from wealthy individuals, wealthy foundations, wealthy corporations, into bringing it back to the continent. So I decided to also leave my very comfortable position, salary and benefits, and living in New York City to come back to the continent. And I set up my company. Initially was Roberta Annan Consulting. There was a consultancy for a few years, and then later on I got other partners to come on board. So now we employ 30 people on our payroll, 10 extra consultants in multiple countries. We have a presence in Geneva, we have a presence in New York, we have a presence in terms of consultants in London, in Dubai, in Mauritius. And our aim is to basically be the conduit to allow funding to flow from, I would say where funding is concentrated into where funding is really needed. And in terms of the asset classes that we are interested in, we're interested mainly in early stage ventures. So we consider ourselves an advisory and a venture building firm. So we support in family offices and wealthy individuals allocate funding to opportunities like what we've discussed here today in biotech, in creatives, in tech, in innovation, and as advisors. But we also work in venture building. So what I mean by venture building is when there's an idea that we think is viable but needs infrastructure support, we come in as a firm, we provide initial funding, so we inject funding, and then we also build the infrastructure that is needed for that venture to survive. And then we help to put a team, because in businesses you need a team, a management team, and you need financing. And so we ensure that the venture has that at the very early stage. And then as soon as they get to point of valuation, then we start looking at external resources to help them to help them grow, whether it's to get more external funding. So biotech, being that I have a master's in biotech, it's obviously my natural affiliation is to venture into that sector. In terms of the landscape of funding, I see that African lacks in funding altogether. Depending on, it doesn't really matter which sector, but especially in the research and development, especially in biotech, we are really lacking behind, and there's so much talent, and we all know that a lot of our talent is taken from here and kind of groomed overseas to work with the Pfizers of this world. So I'm really excited to see that people are coming back and starting their own ventures, and our role will be to support these ventures, right, to help build their capacity, help them raise the funding that they need to be able to survive and to be competitive, on the global scale. So we've actually also raised the fund. So one thing that I like to mention is I like to look at asset classes that are not so attractive to investors and make them very attractive. So some of them being like creatives and tech and innovation is really to ensure that you package it properly so that it's more palatable for investors, or find a very innovative financing approach or model, so that it becomes a model in which does not only target private funds but also public funds, and looking at blended approach. So we do a lot of blended finance strategies so that we can receive both from the public and the private, and to see how some of the public funds can also offset or de-risk the private investments that can come into play. So I'm really looking forward to hearing the rest of the panelists, and to also see how we can, work together to really build this industry and to support our really intelligent scientists, not just to be in the lab, but actually to go out there and create and get the funding they need for it. Thank you.
Thank you very much. So, who do you get your money from? The funder or the fundee, or both?
So in our venture building platform, we don't charge the venture because most ventures don't have the funding. That's why they need us. So we charge the investors. They pay us to allocate their resources. In fact, we're coming up with a fintech solution in the next month, which is called Ago. And that will also allow a lot of the ideas you have to kind of come onto our platform to raise capital. So it's going to be geared towards very early stage ventures. And biotech, of course, is an asset class that we're looking at.
Okay, great. I think you'll be having many clients from this room after this.
Really looking forward to that.
Okay, so finally, our own Yaobi. Yaobi,
yes.
What got you here?
Well, I think I'm the opposite of Lord. So, in the sense that I never thought I would be an entrepreneur. My vision for myself was to be more like Prof. Awandari. I mean, in fact,
you mean poor and
not poor. I wanted to be a pro vice chancellor. No, I wanted to be a director of an academic institute. So that's actually what drew me to Prof in the first place. I mean, it's true. I credit WACCBIP for repatriating me to Ghana. I always knew I wanted to come back. I didn't quite know exactly how. I didn't want to just land here and then struggle. So I was looking for the right opportunity. And when I returned to Ghana and took up a faculty position at WACCBIP, I thought, I'd finished the journey. I thought I'd done 2 postdocs after my PhD, and I'd finally landed back home in Ghana at an institution where the goal was to do world-class science and train the next generation of leaders. So at the time, I thought, this was where I was going to build my career. Eventually, Prof will leave, and I'll fight Peter for the directorship. And yeah, we would build WACCBIP into bigger than Noguchi, and we still would do that. But my goal my goal was really to I saw myself as an academic. But somewhere in and I say the time because it's not been that long. Somewhere probably in 2019, I began to explore biotech. And this was because I was frustrated with how hard African scientists had to work for relatively little job security. We don't have the promise of regular funding. WACCBIP looks great, but it has probably put all the gray hairs, the few hairs left on Prof's head. And if you look at Dr. Kwashi and I, we've aged considerably since we came back. It's extremely stressful as an African scientist because you're always looking for resources and your government isn't interested. I'm very happy that Dr. Ban was so honest yesterday. African politicians are not really interested in funding research, even post-COVID. And it's because they think in 4-year cycles. They'd rather build a school or a hospital that people can point to and vote for them than invest millions of dollars in research that may yield something in 10 years. So it doesn't really fit. It's not just African politicians. It's politicians anyway. It doesn't fit with the way they think. And so as an academic, I began to think I'm going to spend my life going from grant to grant to grant, always worried about getting the next grant. Now, academics everywhere worry about that. The difference as an African academic is the pool of resources we can apply to is relatively small. We don't have an NIH to apply to. We can apply to NIH, but obviously there are very few grants that would focus on Africa relative to an American scientist who's based in the US. We don't have a UKRI or a MRC the way the UK does. We don't have the German equivalent. We don't have a Japanese equivalent. We are looking for other people's governments to fund our research. So I began to ask myself, whenever I ask these questions, even when I would ask at Wellcome Trust events, Wellcome Trust is a British funder similar to Gates, they would say, your government needs to give more. Because the funders are looking at our government saying, we can't keep spoon feeding you. I know the lady from the World Bank is here, probably thinking the same thing. We can't, at what point do African governments step up and pay for the research that is for their own people? But if the answer is, well, we'll wait for our government, all of us sitting here know how long any government anywhere in the world takes to do anything. And so I began to feel that I would be old and jaded if I just waited for the government. And so that began to shift my thinking away from, well, away from purely academic research to, okay, are there other ways to fund research? In the US, biotech is a $700 billion industry. This is commercial research done at a very high level, but it is done commercially. So it builds from academia. And as I began to think about it, I realized that the gap in Africa was we had no biotech. The last 15, 20 years have seen incredible investments by the World Bank, by the Gates Foundation, by a lot of overseas funders in building training capacity, places like WACCBIP, where we are trying to build the next generation of scientists. Where should those scientists go? WACCBIP, even if we blow it up to as big as we have dreams for, cannot absorb everybody that we train, and we shouldn't. The universities in the US pump out PhDs at an astonishing clip. Most of those people are no longer in academia. So you need an outlet for the talent. And the outlet is typically not academia. The outlet is either public institutions that drive research, but increasingly in a more, in the way the world is becoming more and more capitalist, the avenues for where people like us work is in industry. So I got to the point where I said, am I going to wait for someone like Lord or someone else to create an industry and then I'll go and hand them my CV and ask for a job? And in fact, it would be more painful if it's one of my own students. Or am I going to try and do it myself? So even though I haven't had the benefit of a business education, I'm a complete bench scientist. I have a PhD in immunology. I've done years of research in immunology, but I had no experience in business. I decided to ask around, and I talked to people, including Roberta, actually. That's how I met Roberta, about how to start a business. Because I decided that I didn't want to constantly be dependent on overseas grants. I wanted to be part of creating an ecosystem where the students that we train at WACCBIP can see a future for themselves on the continent outside of academia. So talking to, and I got advice from a lot of people, and that's one thing I would say. Don't assume you know it all, because you definitely don't. You probably know less than you think you know. And so talk to people. So I spoke to my academic mentors like Prof. Awandare, Prof. Jean Langhorne, who was my most immediate postdoc supervisor. I spoke to a number of people. I spoke to industry executives. I managed to talk to people who started biotech companies in the US. But then I also spoke to Africans who were in the business world, not necessarily in science, but knew how to build a business in Africa, and got advice from them about what to do. So very quickly, that path led me to found Yamachi. Prof is right. The yam in Yamachi is knowledge, and I know it's from his area. Amachi is Ewe for health, for medicine, and then Machi is Tree for good morning. Put it together, new dawn for health in Africa. But it's also nice that it sounds like Yamachi, we are marching, or Yamawachi. So any of those work. But Yamachi is a biotech company focused on oncology. So we are a cancer research startup based in Accra, but with a Pan-African focus and really trying to build a global brand. We focus on cancer because majority of the research in Africa focuses on infectious disease, despite the fact that we have a growing burden of non-communicable diseases. Cancer already kills more people in Africa than malaria does, and yet there's hardly any cancer research being done on the continent. So we felt that was an area to go into. It's also an area that is relatively open. No one is trying to do cancer research in Africa. So as a young company, you don't have that much competition. So that's what we're aiming to do. We're trying to understand cancer among African people. But the goal is to leverage what I believe is Africa's probably biggest untapped resource, which is our genetic diversity. Africa is the most genetically diverse continent. Our human population is the most genetically diverse of anywhere in the world. Evolutionarily, everybody came from Africa, right? Homo sapiens emerged in East Africa and then migrated out. So really, despite how different we might look. Everybody in this room has some descent from Africa, right? Even my mother, who is considerably lighter than even me. So the idea is, Yamachi is founded on the premise that Africa has the world's most genetically diverse gene pool. Africa has a growing burden of cancer. Cancer is a genetic disease. Put those three things together, Africa is probably one of the best places to study cancer anywhere in the world, and yet, unfortunately, no one is studying cancer in Africa. So what we are aiming to do is build the world's most diverse knowledge base on cancer. The data we generate, the knowledge we generate, identifying new targets for how to treat cancer. My goal is that in probably we would potentially, have the next, who knows, the next BRCA gene is waiting to be discovered among African people. The next, the next biomarker that will redefine how cancer treatment is done is waiting to be discovered. But beyond the specific science that we want to do, the vision of Yamachi is to try to redefine Africa in the eyes of the world. Africa is constantly viewed as impoverished and poor and needy. I don't believe that is true. I don't think anybody in this room believes that is true. Yes, we have our problems, but we have great potential, we have great talent. The idea is to try to be part of a new ecosystem of science that takes the great work that is done at institutions like WACCBIP, provides people with opportunities to innovate and excel, similar to what Lord is doing with his company, and really create a brand that the world will recognize as excellent. So excellent first and African second, right? So we're not just pigeonholed as African, we're globally relevant. So in the last 12 months of operation, Yamachi has been operational for just over 12 months. We were operational from April 2021. We've built a lab. We now have whole genome sequencing capacity in our own lab. So we've been working with WACCBIP to do sequencing. In fact, together we did the first breast cancer exomes ever sequenced in Ghana. This is within 12 months of being operational and through a close partnership with WACCBIP. We now have our own lab as well. So we are building capacity beyond what WACCBIP has, and that is also obviously also available to WACCBIP as and when needed. We've employed 32 people. So in 12 months, we've provided jobs now for 32 people across a range of disciplines. So beyond the science, we also have software engineers because we build our own software for clinical data capture. We have obviously finance and admin teams. And I'm very proud to say that our workforce is about 50% female. Our senior leadership is closer to 60% female. And our lab is 80% female, right? So that is not what people think when they think of Africa. They don't think of a room full of female scientists working in, doing cutting edge research. So that's part of how we're trying to redefine. Finally, milestones we're proud of as a company. We're the only the only Ghanaian company in our batch of Y Combinator. So this relates to trying to find money. We're looking for investment. Like Roberta, I've talked to nearly every high net worth person in Accra trying to get them to fund what we're doing. In the end, we found money in the diaspora. Ghanaians and Nigerians and Africans living abroad who thought they wanted to contribute to what we were doing. And then we began to expand and we began to raise money from venture capital. So we have investments from US venture capital firms. We closed our seed round, which is the first round of funding to fund what we're doing because we're trying to do it big. But 3 million is a drop in the ocean compared to what we need, but it's a start. We were the only company in Y Combinator from Ghana out of only 15 companies from Africa, out of 400. So Y Combinator gets about 15,000 applications for every batch. They pick about 400. 15 of those were African, and Yamachi was the one Ghanaian out of those in our batch. But beyond that, the money is nice, but just raising money doesn't mean anything if you don't do anything with it. So what we've managed to do in 12 months, which I think speaks to the power and the agility that you can do if you're a private company, is we now have relationships in 6 African countries. We've built a network with access to samples, cancer patients in Ghana, obviously, Nigeria, Kenya, Cote d'Ivoire, Senegal, and also we have partners in South Africa. So this is all done within the last 12 months. We are now beginning to get our projects going, but it's exciting to see the traction we are beginning to build. But additionally, we also have partners in the US and in Europe. So we have a long way to go, and I'm very happy to be part of this discussion. My process may not be the same as everybody's, but I think it speaks to what you can do if you have a dream and you're willing to work hard And I'm very happy to be here. Thank you.
Great. Thank you. Thank you very much. So I think that your presentations have covered a lot of ground, so that's good. We've saved time on some of the questions. But there are two issues that I want us to talk about. One is the key enablers. So as you go through this journey, what do you wish you could be supported with? Not in terms, apart from raising money, of course. What government policies, what institutional regulations, and so on and so forth. What are the bottlenecks that you think, if they are removed, there will be a boom in private enterprise, in the scientific private enterprise? So I'll start with Margaret.
Thank you, Prof. I need to say, this is, I was just nodding along as you were speaking. You were basically speaking to me, because from the very beginning, when I thought of macro, I'm like, why do we have to sit down and wait for other people to come and tell us how to do things in our own setting? And we're not going to venture into the other bit of it where we don't have support locally. But it's about being able to get your own funding, being able to make your presence felt, for people to understand that, yes, we are capable. Yes, we can do it. I always said in previous meetings that I'm in that in this country alone, I cannot count the number of professionals and scientists that we have in all aspects of science, whether it's epidemiology, immunology, you name it, all the logies that you can think of. We've got the best amongst the best. So why do we need to sit back and wait for others to come and tell us how to treat our own diseases, how to develop our own vaccines, how to develop our own treatments? So, you were just speaking to me, and I've just been nodding along with everything that you said. So now I'll come back to Prof's question. So the bottleneck, it still comes back to being able to understand that when we are in our setting, and we see the diseases that we're dealing with, and yes, I also agree that it's not just the infectious. You've got diabetes on the rise. You've got all these cancers on the rise. Obesity. We don't usually see it as a disease in our setting, but it is, and it's rising. Even amongst the youth, you go and you do a diabetes testing, and then, sugar level testing, and you realize it's through the roof. And people don't realize that it's because of what we are putting in our bodies. But I digress. We need to be able to understand that amongst ourselves, how can we continue to build our own youth as they're coming out of school? When I finished my MPH, I did not even know about clinical trials. It was one of my professors who mentioned that there was a clinical study in Navrongo about rotavirus. And that's when I started working in clinical trials. And I lived in Navrongo for 2.5 years to be able to do the clinical trial management for that study. And that's when I developed the interest. And what most of the students coming up don't realize is, it's not just about being a medical doctor or being a pilot or all these high-end professions that are out there. As a CRA, I can tell you that somebody coming from Europe can earn as much as $800 a day just by monitoring. So they come into our country to monitor studies that we already know about, to monitor diseases that we already know about. And then they want to pay a local CRA less than a quarter of that. Why should that be the case? So these are the things that I would have wanted some kind of policy or some kind of support, so that when we are marketing ourselves, people understand that we not only bring the same amount of knowledge, but a lot more knowledge, because we understand the context in which we are living, the context in which we are dealing with these diseases. So that's what I would actually say, that this is the bottleneck for us. Yeah.
Great. Thank you. Let me go to our virtual. Yeah, good. So
hello.
Yeah. I just wanted to make sure you're there. So you are the fundraising person. what do you think government can do to boost investment in biotech, for example? What are the impediments to people putting their money in investment? You know, you mentioned the keyword risk. You know, all the panelists sitting here had to take a risk to do what they are doing now. You need funders to take a risk to put their money on your idea. What are some of the things that government can do to encourage people to invest in biotech? Because that's what we need now.
Yeah, I think it's really interesting. And also just to dovetail a bit from what the previous panelists just talked about. The major issue with driving foreign direct investment into Africa, it's not even just from a biotech perspective, but I'll obviously come to biotech. I want to talk about the general issue. There's a perceived risk about Africa, right? That doesn't actually exist. So if you look at it, most investors have this perceived conception about Africa, that Africa is corrupt, there's corruption, there's, lack of financial infrastructure, even trying to repatriate your funds back is a challenge. So many things that deter investors from coming into the continent. And then there's the actual risk, which is what, when you're on the ground and you make an investment, you actually experience, when you're on the ground. So I think when you're sitting from the outside, it's difficult to understand what the lay of the land is or what the risks are on the ground. So you cannot make a judgment based on that. So I think what government can do is to start demystifying some of these perceived risks that exist in the outside foreign space. And I think we need to start promoting our own success story, because I think by directing the narrative and pointing out some of the success stories like Yamachi and here on the panel, if we're able to get the government to start highlighting these, it's going to attract more funding to come into the space, right? Because within a matter of 12 months, they've been able to have significant milestones. So if you look at the trend in which and the direction in which they're going, it's quite typical of a biotech company or a biotech startup in the US. So they aren't doing anything different. So that means that in Africa, although there are challenges here, we need to start letting people understand. I think we also need to use maybe public relations or PR tools to really position and tell our stories to the outside world, so that they see that the talent exists here, the opportunity exists here as well. And we just need to find a way to highlight and to support each other and to build our capacity to be able to accept more funding. So that's number one. Number two, I don't always rely on government, but of course we do need to set policies to ensure that if you're operating here, for instance, I think somebody mentioned that they have a Pan-African venture. So they work with researchers in different countries in Africa, right? So if you're talking about, talent, that's one thing. But if you have a business and you're talking about trade, that's a different matter altogether. We have businesses that we are grooming that are that exist in different African countries. So Nigeria and Ghana alone, you have two countries between that, and there's a language barrier, there's a policy barrier. So I think even on the regional level, if government start putting things in place to ensure that the policies are not just policies that are concentrated in country, but then policies that can also be accepted, in regionally or even, through the African continent, throughout the African continent. Then that is also important for investors to see. That's why I like policies. I like the what the African Continental Free Trade Agreement has set up, in a way in which we can not just look at things from a country perspective, but from a Pan-African perspective, and looking at ways, systems in which, if you're talking about Pan-African trade, how do we look at currency, And I know that there are, banks like the Afrexim banks that are working on like a Pan-African payment system, and there are solutions that people are creating to be able to solve some of the challenges that create, because you need to create the economies of scale. Investors want to see that, okay, I'm coming to Yamachi, but is it possible for a Yamachi setup to be in South Africa, in Nigeria, and in Kenya, so that it's not just a Ghanaian thing. Can we have the economies of scale? Because most of the funding that is coming into the continent at this point is very large ticket size funding, right? So it's not, that's why it's more difficult for early stage ventures to raise money, because funding comes into private equity, it comes into infrastructure, it comes into larger like 25 million to 50 million to 100 million, billions of transactions. That's what a lot of investors are looking at. When it comes to very early stage investments, it's really, really, really difficult, and that is why it's important to start thinking about economies of scale, to start thinking about the replicability of the business that you're creating, right? In a way to be able to attract future funding. It's really important for you to think about the initial infrastructure of the business, making sure that everything is perfect. And I like what Yael said earlier about excellence. This is not an African story. This is a business story, first of all, right? Let's focus on getting the business right. And then, yes, we are located in Africa. So we need to, kind of start thinking a bit, about how to position ourselves strategically to offer excellence. We should be able to offer the same. If you say to a client that I'm taking samples and I'm going to basically sequence this genome, I want to be able to deliver it within a month, deliver it within a month. All of these things are important to have a competitive play to raise more funding. But I think in terms of government, I know that Ghana has just recently launched a development bank. These are the type of ventures. And, of course, on the policy side as well, government should also support to ensure that the policies are set in place so that when investors put money into ventures, they're able to pull back their money, on time, and it's more attractive and palatable for them as investors. But I am I'm a strong advocate for private sector development, because I feel like government and private sector, there should be a partnership, between government and private sector. But the idea or the main impetus for government is to really empower the private sector. Because when you empower private sector, you address a lot more socioeconomic challenges than you think. You start creating jobs for women. You start creating jobs for young people. Africa has a very, very large young population, 65% under the age of 25. And how are we going to capture this huge demographic that's going to need jobs and opportunities? Is through entrepreneurship development. And this is where private sector can come in and be empowered to empower and to even address some of the big challenges that government will face in the next few years if this is not addressed. And if you look at an example of, let's look at the US, for instance, where private sector is so empowered. If there are policies that are being set, they have a way to lobby to ensure it's policies being set that are not working for them. They have a way to lobby to ensure that that policy doesn't get established by government. So that is the power of private sector, and we need to encourage that in Africa. And it's the only way that we can drive our prosperity. And I feel like everybody here has, been exposed to, on this panel, has had the opportunity to be educated or exposed to other markets, and they know what's happening. And just bringing that knowledge back to the continent to drive its prosperity using, startups and biotech and in other sectors, I think it's the way in which we can start empowering private sector. So the private sector can play its partnership role with the public sector. But what is happening now is that public sector is actually driving private sector in Africa because of politics, right? But we need to think outside the box and to start thinking about, how we can separate the two, but also work together to promote the development of our country.
All right. Thank you very much. So I'll come to Lord. You are, walking the walk now.
Yes.
I want to, take it from where Roberta ended. So I want specific government policies. So yes, we agree that private sector should drive, but I often hear about people hesitating because of certain policies. For example, tax. Are there certain tax incentives that can be put in place that will allow people to invest in biotech? Or are there certain taxes that you think are a nuisance? What about regulatory? Are there some regulatory processes that you think are too difficult and that can be simplified, or removed altogether? I want some specifics in that direction.
All right. Thank you very much. I think that generally the environment for young businesses, not just biotech, makes it very difficult for businesses to thrive in Ghana. When you are an African business, the burden of you are expected to have a higher test for quality. You are supposed to have a better price and everything more than a company that is coming in. When I make pregnancy tests, okay, and usually if somebody wants to buy a pregnancy test, or somebody wants to suspect that she's pregnant, she'll go to a pharmacy and then buy a pregnancy test, and she'll keep testing until she either has her menses or she probably tests positive. When it comes to our local production of pregnancy tests, the first thing you will always ask for is the quality. Nobody will ask any of the pharmacy whether the quality of the foreign product. And I think that it's because we tend to project our inadequacies in others. So I know that if I were the one producing the pregnancy test, I might probably cut corners. And so this other person who is producing the pregnancy test is definitely cutting corners. And I think that has actually is one of the things that is a major problem for us. And I would give you specific examples when it comes to talking about tax incentives and all of that. Now, you are expected to have a cheaper product than things that are coming in from China, for instance, products that are coming in from China. And it's because they assume that you are producing locally. However, when a product is coming already made and it's coming from China, it is classified as a medical product. So there are a lot of tax incentives that are given to that product that is coming in. You are buying raw materials. The raw materials we buy are paper, and then we process the paper into diagnostics, and then of course plastics. These also attract very high import duties. So the raw materials cost more than the finished product. And yet you are expected to make the product and make it as cheap as possible. And yes, we do that. Our pregnancy costs about 50 pesos. Okay, which is cheaper than all the pregnancy tests that come in. And it's because there are a lot of middlemen in between, so they all put their cap. But when you look at the cost, the cost of raw materials alone, if the cost of raw materials are reduced, the cost of pregnancy tests will come to 10%. So that is one of the major problems. Major other problems with we buy all our raw materials because in the whole of Africa, we have very, very, very few companies that are manufacturing diagnostics. In Ghana, we are the only one that is actually doing the development and manufacturing of diagnostics. And because of that, most of the raw materials produced are produced in Africa. So you have to import every single one of them. And you would budget at the beginning of the year, you budget with $6 to $1. And then within 2 months, it's moved to 8 cedis. So that means all the budget you did is basically useless. So almost every 2 weeks, you have to draw a new budget and all of that. And you cannot keep changing your prices as these come along. So it becomes very, very, very difficult to actually plan and to raise money. For us as a company, money is not, raising capital is not, because we are already on the market, we are selling, we have a lot of customers in Africa and outside Africa. So we are actually generating revenue. But all our revenue goes into these import duties, taxes, and all of that. When it comes to tax, one of our major problems is the lack of understanding of what a biotech company generally is by the stakeholders. They give you arbitrary charges. So this team will come to the office, look at what you do, you explain what you're doing. They're like, oh, okay, so you are not a lab. No, you are not a lab. Okay, then you're a pharmaceutical company. then you are like, no, you are not a pharmaceutical company. You are still a startup between this and this. Then you go in and by the time they bring you the charges you have to pay, you realize that they are taking almost every money that you've made. So we've tried. We started by making brochures explaining exactly what we do. I remember we are located in Kumasi, so I remember very well that 2 or 3 years ago, we did a brochure and we sent to all the tax people, GRA. We shared about almost 30 of them, just trying to explain this is a company, but it's not a lab. This is what we do. This is what we do. This is what we do. These are our products and all of that. When it was time for us to get our tax clearance certificate, we went there. They looked at our financial statements and all of that. And then the first thing they looked at was the revenue we had generated for the year. And the first thing, I remember that it was a deputy somebody. The first thing was like, hey, Munyes Kapao. To it, like, you make a lot of money. But I was trying to explain to him that when you look at the revenue, yes, I can make 50,000 cedis a day. But that same day, I can make expenses around 80,000 cedis. So if you're just looking at the money you are making and not looking at what goes into it and the expenses you're making, and then the infrastructure you have to put in place and all of that, it becomes very, very difficult. So for us, for instance, all our funding, external funding, has come from external places, just like Yamachi. In 2020, the Bill and Melinda Gates Foundation gave us equipment worth $2.8 million, just equipment. And even that, when you got to the port, we had to pay almost 1 million Ghana cedis to clear the things from the port. So there are so many barriers that make it very difficult. Ghana has a talent for losing talent. Okay, yes, because they will frustrate you to the point that I have a lot of people. I think both of us we are in the tech ecosystem, and so we have a lot of other people that are not necessarily in biotech, but in other tech spaces. And I see most of them leaving Ghana almost every single day. And it's because it's not money. We don't necessarily need government to give us money, but it should make certain things quite simple. When it comes to regulatory, I think that's the last point. As a consumer, I am always very happy with the rigorousness that FDA goes through when they have to check our product. As a consumer, it makes me happy that at least the welfare of citizens is taken into consideration. And I don't have a problem with going through that rigorous thing. I mean, it's a very good thing because it helps us build our capacity. Most of our products, we have about 8 products. About 7 of them had gone through the regulatory process successfully. So I do not have a problem with it. I just have 2 challenges. The first is the length of time it takes. When you send a product to FDA, they are trying right now, but it can take between 4 to 6 months for your product to come in, okay, or for the results of your product to come in. And these are products that you've put money in and you've made. So it becomes very difficult. You have to put this thing on hold. You cannot sell them or anything. And most of our products have an expiry date of about 2 years, okay. And you cannot just they take about 300 samples of our 300 products. So you cannot just make 300 products. You run at a loss. So you are producing about 2,000 test kits, for instance. And then you take 300 to the FDA, and they take half a year to come out with sometimes half a year, they'll come out, and then they'll ask you to maybe do something on the packaging or anything. So if you are not very careful, it can take up to a year. What that means is that the product only has half a year or a 1 year to be on the shelves. That is problematic because if they do not buy all of it, you are going to have a lot of expired product that you cannot do anything with. The last one, which for me has been the most critical, is that our system or this market, the biotech market and the diagnosis market, is based on speculation. Maybe speculation is not the right word, but for instance, when you sit on radio and then say that a product doesn't work, okay, what you have done, probably you're talking about a particular product, but what you've basically done for a young company is to say that the company doesn't produce quality product. There are two different, there's a way to say it that will not mean that a company doesn't produce a very good product. I'll give you an example. In 2020, we developed an antibody test for COVID. Okay. It was basically because at that time COVID was new and we felt that, oh, we could do something about it. And so we took a long process with our own funding. We didn't get any funding to do that. With our own funding and sleepless nights, and we developed it. It was tested with 34 other products from different countries. And because it was an antibody test, none of them went through. However, there was a lot of interest in our product. So I remember there was a time on radio, the FDA boss, I'm not trying to pin it, but the FDA boss was asked about our product. And the comment was that it doesn't work, which is completely different from it didn't meet the sensitivity that or the threshold that you want. Okay, just by saying that it doesn't work, what that means is that you didn't create a good product. And if you didn't create a good product, that means all your other products are not good. And that actually affected us a lot. Some of the people that were already buying our other products that had been approved started to ask us, oh, if this doesn't work, then how sure are we that this works? And then you have to spend time explaining and explaining and explaining. So I think that generally, a lack of understanding of what the market and the space is one of the major issues or challenges that we face as a company. Okay, we are in Kumasi. We do our manufacturing in Kumasi. We have branches here. We also have, we are opening a branch in Nigeria, in Kenya, where we are going to do manufacturing. And one of the things, and the reason is that when you take a product from Ghana and you are shipping it to Nigeria for it to be sold there, it is more expensive than if somebody is in Nigeria is ordering a product from China. And we are neighbors. Okay, so it prevents expansion, especially in the African continent. Our COVID tests were shipped, it was approved by the FDA in the US. So we were shipping it there. We found it easier to ship our product and sell it in the US than in even Liberia. Okay, we had an order from Liberia, and shipping it to Liberia was so much of a problem. So I think after the African Continental Free Trade Agreement, it's in the right step. But I think that policies around getting seamless trade between us will be very good. It costs us more to produce the product in Ghana and ship it to Nigeria than to actually set up a manufacturing plant in Nigeria to produce the test kits. And that is something that I cannot, but there are a few of the major challenges that we've been having so far. Thank you.
Great. I knew there were a lot of issues under the bubble, so it's good that you've put them on the table. I'll go to Yao with similar ones. I know that from the academic institution point of view, you are aware of all the issues with procurement and Lord mentioned the clearing of equipment from the airport, which is totally ridiculous. I mean, the taxes, they say that they waived the duty, but there are so many other taxes that are even more than the duty. So the duty is probably, 10% or 15%. But then you have National Health Insurance Levy, you have ECOWAS levy, you have that. When you add it up, it's like 60% of the value of the equipment. So what they have waived is less than what they are still taking. So I think these are some of the critical issues that we need to get across to government, because if we really want to promote, science and enterprise, some of these barriers have to be knocked down. So maybe 5 minutes to top up what Lord has said. I'm sure you have a few more examples of frustrations in the system.
I think Lord has said a lot of them. I think, Prof, you've continued. The biggest thing for us trying to set up a cutting edge lab has been import duties. I think we have, I think we shared this one. When you buy a $40,000 machine and then you pay duty of $10,000, it's now made the machine $50,000. And when, the problem is not just it makes us non-competitive locally, it makes us non-competitive globally. So as a company like us we're not competing, Yamachi is not competing with Ghanaian companies because there's no other company doing what we do in Ghana. There are very few even in Africa. Eventually, we are going to have to compete with biotechs in the US who can pick up a phone and order an antibody or order a primer and get it that same day. You order it here and it will come in a month or two. In fact, it may be cheaper to buy a plane ticket and go and just pick it up and bring it back, right? I mean, we joke about it, but every scientist here has done that. Prof travels, he brings 5 extra pieces of luggage full of, Peter has done it, I've done it, Lily just, this projector, Lily brought yesterday or 2 days ago in her suitcase, right? I mean, so this is what our governments need to understand.
Don't report us to the...
Yeah, I don't report us. But we all, I've done it and they've opened my bag at customs and I'll say, I'm at the University of Ghana and I'm bringing reagents. And, sometimes they'll charge you, other times they won't. But we cannot be competitive, right? How are we supposed to compete and do internationally competitive science, whether as academics or as private people, if we have to go through these problems? So when, these days I get approached by GIPC to go on their missions because they've heard a bit about Yamachi and they want to brag about Yamachi to the rest of the world. Problem is they want me to pay my own tickets, right? So I'll get invited with really nice email, but then they'll say I have to pay my way. So I can be part of the mission if I pay. Now, I think that's how they operate. It's fine. But it would be nice if, in addition to trying to take credit for what people like us are doing, they would actually call a meeting and say, how can we as a government make things better? Because obviously, if companies like Incas, Yamachi, Macro do well, you can take credit as a government. And I have no problem with that. You can say, look, every country does it. Rwanda advertises on Arsenal's shirt, right? You want to encourage your investors to come to your country, so you talk about the good things happening in your country. There's a lot more that could happen if the government would take an interest in trying to remove the barriers that drive people out. So yes, import duties are a big thing. I think you made a really good point. They don't understand what we do. So maybe we need to find ways to engage. Because you're right, you make diagnostic tests, but you're not a pharmaceutical company. You're not a diagnostic lab. You're not Lancet, right? But they will try to group us in a category because they don't have categories to account for who we are. Now that is a problem with their coding system, not with us. So we need to evolve our systems, our tax systems, to understand. If you're a company like us, there are certain capital requirements you have to maintain, right? So you have to have enough, I think it's $500,000. You have to have $500,000 in your bank account to operate here if you are, if you have a foreign entity. But if the shareholders of that foreign entity are all Ghanaian, why is it that you are treated like you are completely foreign, right? So these are all things that I think they come from a system that has not evolved. So we're using archaic models to try in a 21st century world. And I think that's what holds us back. It's probably not just in Ghana, many African countries. So I really liked, I'll end with what Roberta said about directing the narrative. The biggest challenge companies like mine face is convincing international investors to invest. Because they hear all the horror stories. They know how expensive it is to import things. They know that our currency fluctuates. And so if you're providing services and you're charging cedis and your inputs are in dollars and then the cedi drops, you can't just hike your price. And so they know that adds risk. So there's huge perception of risk. Recently, we're trying to raise money. An investor described what we do as we are working in the third world. Who uses third world these days anymore? At least they say LIMIC or emerging markets. But that spoke, I stopped talking to him because I realized it was a waste of my time. But for those perceptions exist, and unfortunately the capital is in people like that hand. They have a very warped and uninformed view of Africa. So we have huge barriers to overcome on that side. The least we can expect from our government is to try to minimize the burdens for us doing the work, but then also helping us to reframe and reshape that narrative so that when we approach investors, they actually have a slightly different, more positive view of Africa. And that, I think the governments can do a lot to begin to reshape the image of our countries, which will then make it easier for entrepreneurs to approach investors for funding. So yeah, that's
great. Thank you very much. All right. So before we wrap up, I would like to take just maybe 2 questions, comments from the audience. I said 2, and there are 5 hands up. Maybe I'll ask the 3 of you to meet and come together with 1 question. Okay. Yes, let's take a few comments. Let's make it short, 30 seconds each. 30 seconds.
Hello. Yeah. Big thanks to all the panelists for the very informative and expressive insights on our issues. I believe I'm just trying to add on a new perspective, though I'm no expert here. I think from what a few of the panelists have pointed out, definitely there's an issue of understanding. And one big institution I think we might be leaving out here is the media. Because in this whole eco space, we are all trying to be interdependent. Okay, what one aspect, what one institution or one aspect of the system does directly affects the other. Like what Mr. Lord talked about, citing that example. I could cite my own personal example when I went to do internship at a hospital. And they asked me, "You are a biomedical engineer, what do you do? Who are you? Where do we place you?" And they told me, the comment they passed was like, "Our hospital fitani." That's from the local parlance, like you are a mechanic who works in the hospital, that kind of thing. And it was, it blew my mind. So I think having experts collaborate with those in the media space, especially on how scientific work can be translated into enterprises and how this whole thing is linked in a dependent ecosystem will help a lot in averting some of these issues. So I think that's also one aspect you need to look at. Thank you very much.
Thank you. Thank you.
Okay, thank you very much. I'll just go straight to the point. My question is to any of you can respond to it. The first one is, so the different companies that you run, is there a provision that you do idea development? Say a student has an idea and you help the student develop the idea down to like a translational thing down to the finished product. Do you do that? And then the second part of my question is, since we are in an academic environment, all of you have gone overseas for your studies and you're back, which is a good thing. But what do you have to say to students who currently are doing academic projects but have interest in biotech? For Dr. Yao Bediako, you had your early career in academic, and then you had this switch to biotech. What was the switch like? I don't know if you get my question.
I can try. I think we are. Prof, should I?
Yes, please
okay.
Go ahead. Yeah.
I think everybody's journey is going to be a little bit different. In terms of whether we help students, I mean, we have a couple. In fact, we have an internship program where we have WACCBIP students who now work a bit at Yamachi. Obviously, our projects are beginning to grow, but the idea is we'd like, industry can be just as quote-unquote basic science as academia, right? It's just what the direction of your research is usually dictated by what the company is interested in following. So we do hope that over time we can have more and more students flowing through and help people to begin to think innovatively and translationally about the science that we all do. In terms of my own experience, I mean, the toughest part for me has not been the tech, it has been the business, right? As the CEO of a company, I'm now a businessman. And so sometimes, I mean, I have long conversations. In fact, today I have one I have to have after this, talking about business models. How are you going to, a lot of talking about now I know what EBITDA is, right? I'm sure some people here don't know. I've never heard of EBITDA, EBITDA. Go and look it up, right? The idea of revenue versus, your actual net profit or loss, right? These are things that as a scientist, I'm not prepared. Nobody in immunology, I was learning CD4 and CD8 T cells, right? So as a scientist, if we're very technical, the thing you have to recognize is you're going to run a business. No matter how good your science is, your business will fail if you don't have strong principles behind your business. So what I managed to do was surround myself with a team of people who had some of that experience. So I think that's why you need to, everybody will have different challenges, but the hardest thing about going from academia to biotech is not really the science. It is going from working in an institution where you write a grant and you do work and you publish papers, and that's what you've been trained to do, to doing something that you have no training in, which is understanding how a business runs, and then trying to marry the two. Because my business people don't understand the science. So I have to stand in the middle and try to explain why the scientists want to spend $50,000 on one machine, right, when we don't have any revenue. Oh, because it will really, So that has been my personal, and I'm still learning a lot. So I hope that helps a little bit. I'm happy to talk afterwards as well.
All right.
And can I?
Yes, and
can I make a comment?
There's a question online about how do you do your clinical trials? I think.
Okay. All right. So just to add to what he just said, sometimes when you are going into business, I think one of the things you need to do is look at your strength. Okay. I am still in academia. I lecture at Kenya State, Department of Medical Diagnostics. And one of the things I have found is that when you're a CEO, you do little science. You do more fundraising than science. And as somebody who is a lab person, I like to sit in the lab and develop things. It can be depressing. Seriously, because you realize that you've gone the whole year, you have not sat in the lab. I don't know whether I'm the only one that feels that way. It feels like you are not doing anything in life, that kind of feeling. Okay, so sometimes you have to decide whether you cede that CEO position to somebody else so that you do like what you love doing best. So, and then when it comes to students, yes, ideas and all of that, I think the best thing is to approach somebody who is into the space, discuss with the person, I have this idea and all of that. But make sure that you sign an NDA, because in this world, you go and sell your idea, and by the time you realize somebody's in it. So create the habit of having an NDA. It shows that you're a very serious person. And let the person sign, and then you explain your idea. If they can take it up, it will be very helpful. How we do our clinical trials. So that's one of the things that has actually helped us with academia. So when we develop a particular test, we write for grants for it, and then we usually have somebody, independent person, validate our product for us. So for instance, the Yours Kit, we have a Grand Challenges Africa and Grand Challenges Canada is sponsoring a clinical trial on our Yours Kit. So when the results come out and they are good, then we proceed. So that's usually how we do our clinical trials.
So you mean you do it yourselves, you contract a company like Margaret to do it?
So when it comes to we don't do the clinical trials ourselves, but usually you have people who are probably in the department or anything. You just tell them, oh, I have this product. Is it possible to do a clinical trial? They write a grant, and if they get it, then they take care of you. You just provide the number of kits they use. So, for instance, KCCR did some work for us on some of our products. We sent the number of kits they wanted, and then they did, and then they shared the results with us. So, and then after that, they publish, and then you have your
and then you can use that through the
to the FDA, and FDA usually accepts it if it's coming from Noguchi, KCCR, or any of these established organizations. Yeah.
Great, thank you. So we have to wrap up. So I'm going to let the ladies send us away.
Can I jump in? I think there was a question about startups, and sorry, like if you have an idea, if the support. So one thing that we're doing is we're establishing an incubator. There's a physical incubator space. So if you have, it's opening in September. I can share more details about it. If you have an idea that you want to develop, we'll have a launch to invite people to apply. So you can come there, and for 9 to 12 months, we have a rigorous program to help you go from ideation to prototype. And then if the prototype is one that we want to kind of help you develop further, then we can put you on our venture building platform to raise capital for that. So these are some of the things that we're doing as a firm to support, people that have ideas. And of course, a lot of these ideas will come from the universities. So we are looking at ways in which we can partner with the universities to get more, opportunities to back. Thank you.
Thank you. So, Margaret, you give us the final word, and then we.
Okay.
We can
Well,
close
thank you, Prof, and thank you, fellow panelists. I think this has been very insightful for me because it's always good to see that one is not alone. I hear the comments coming from my fellow CEOs, and I'm like, these are the problems I'm dealing with. After the first year, then I'm told that my international consultants who are working on projects outside Ghana have to pay 20% tax to Ghana government when the project is not here and they are also not here. Meanwhile, I have a contract with them where that was not stipulated, so then the company loses. So these are some of the challenges. I will say, for my point of view, the Ghana FDA has been very supportive with how our business is done, in the sense that for projects, clinical trials that are done in Ghana, they require a local monitor as a requirement for your clinical trial to run in this country. So they have been very supportive. Of course, we also have challenges from our projects where they say the FDA takes too long, but for me, they are doing what they can to protect our citizens if the project is being done here. So for me, I will just say, for all the students who are interested in venturing into the clinical trial arena, you can reach out to us on www.macro.org. We're actually starting an annual clinical trial readiness training starting in October, where we give practical know-how on how to conduct and manage clinical trials. Because, as we've all said, we have so many diseases, and we're always sitting down and waiting for people to develop the diagnostics, develop the treatments, develop the medications. Why? When we have WACCBIP here, we have Noguchi, we have Dr. Vidya Eko's Yamachi. Yes, we have Lord's Diagnostic Company. Why should we wait for somebody out there? We have Roberta's company to help us get funding. Why can we not come together and put our heads together and do this ourselves? It's called, I think it's from the South African or East African, the Ubuntu spirit. Let's come together as people of one continent and know that together we can actually make a change ourselves. Okay? Yes, we do need the international donors and the international support, but basically most of the activities that we can conduct ourselves. Thank you.
Thank you very much. So we've had quite an engagement online as well. Folks are mostly supportive and agree with the problems you've articulated. And I want to acknowledge, significant contributions from Dr. Elizabeth Diallo, who has put in a lot of text here, as well as Edem Apewukin, who has also put some suggestions. And they are both calling for a movement that you should all get together and push for some of these changes that need to be made in order to facilitate the industry. So anyway, ladies and gentlemen.
Something quickly before you wrap up. So Dr. Elizabeth Diallo is actually my Francophone partner.
Okay, I can see that.
She just sent a message to say thank you very much for acknowledging her comments.
Okay, thank you. All right. So it's been more than we expected, actually. So that's great. Let's acknowledge the panelists. So I want to thank them very much for taking time off their busy schedules to come here. I think by doing this, they've donated several thousands of dollars because these guys, every minute costs some money. So I think that by spending 1.5 hours with us, they've essentially donated a lot of money to us. So we are very grateful to Margaret, to Roberta, to Lord, and Yao for joining us. And thank you very much to all of you and those online for engaging and contributing to the discussion. So thank you.
Thank you. Thank you.
We are on WhatsApp all the time.
Yes, let's give a round of applause once again for that stimulating talk. So just before lunch, we're going to have a series of very quick turbo talks. Most of the poster presenters have uploaded videos which will be played, but before then, we'll call 4 of them to give a quick synopsis, 2 minutes, of what they will be presenting in their posters. So I'll call in order Dr. James Abugri, Kobe Reinhard, I can't read the other word, Paul Akinduti, and Eric Gaba. So in the order I called, so James Abugri.
Okay, so I'm James Abugri from the C.K. Tedam University of Technology and Applied Sciences, School of Chemical and Biochemical Sciences. So we partnered with Dr. Teresa to do tissue tropism of trypanosomes. So what we have done in my poster is to take samples from cattle in Navrongo to look at how the trypanosomes, which tissues they prefer or which tissues we can find them in. So we took about 56 samples from 28 cattle, that is for adipose tissue, and from the blood. And we realized that we could find the trypanosome parasites in both the blood and the adipose tissue. So this is what my poster is about. So poster number 77.
Thank you. So I'll call Kobe Reinhard. I apologize, I can't read the last.
Yeah, Kobe Reinhardt, and my poster is about simian malaria in human. So let's all, a quick shot of imagination. Imagine a world where malaria is actually free, but then comes a time where there is now cross-species infection from the monkeys to humans, and this will pose another threat and challenge. So my undergraduate project, we sought to start screening plasmodium infections that have already been identified in Southeast Asia. Most popularly known is the Plasmodium knowlesi, which is now becoming an emerging public health concern in Southeast Asia. So we sought out to screen individuals living in communities inside the Mole National Park who have had interactions with monkeys to see if there will be any possibility that there's already been some of these Plasmodium infections circulating in that community. And we went on ahead to also characterize other 4 screen other 4 Plasmodium infections known to be specific to monkeys. Yeah, from the results, we didn't get any hits, but then an interesting thing was that these were asymptomatic patients, and at the end, we realized that there were more ovally infections compared to malaria infections, which has not really been reported in some studies. So please come check out my slide 48 in front of the malaria lab. Thanks.
Okay, and Paul Akinduti.
Okay, thank you very much. My name is Paul Akinduti from Covenant University, Nigeria. Actually, the work is on cluster analysis of Escherichia coli 0157 from human source, water, and animal products. What we did was to check for the relatedness of E. coli 0157 from human, animal, and from the water source. From our analysis, we realized that there is a relatedness in antibiotic resistance pattern from the animal product and the human strain. And likewise, from that same study, we realized that there's a geospatial distribution of some of these E. coli in urban and peri-urban area. That's to tell us that there is a high rate of spread of this E. coli 157, causing a lot of hemolytic colitis among human subjects, particularly in urban area. We had many people consume some of these contaminated food products, I mean the animal food products, and we are able to find out that it is not common in rural, but it's very more common in peri-urban and urban area. Actually, the work we still working on it. We're still working on the samples where we want to do the genetic diversity and likewise phylogenetic of some of these isolates. My poster is on is number 73 up there, I think in the third floor over there. You can have a time to check it. Thank you very much.
Thank you. And then Eric Gaba, and then after that, we have some uploaded videos that will run before we go for lunch. Eric Gaba. Eric Gaba here. Okay, so Theo, start the videos then.
Evaluate the effects of breeder disease of Pilatus amarus extract on the kidney of adolescent-induced diabetic albino rats. The plant was obtained in Lagos, Nigeria. It was identified and authenticated at Oden Department, University of Lagos. 25 Bristol albino rats were used for the experiment, which took place for 21 days, and they were grouped into 5, where we have the control group, the group 1, 2, 3, and 4. The control group received no treatment with Pilatus amarus, and also there was no induction. Group 1 received only treatment with Pilatus amarus, and there was no induction. Group 2 received treatment and was induced with diabetes on the 8th day after administration. Group 3 and 4 received the treatment and also was induced with diabetics with different doses, which are 300 milligram per body weight and 500 milligram per body weight respectively. So observed that there was no significant difference in the level of albumin, protein, and creatinine for all groups. However, there was significant increase in the level of urea concentration in group 3, which was administered 300 milligram per body weight of Pilatus amarus extract. This, in conclusion, it was actually concluded that Pilatus amarus extract is not dose dependent. However, further studies should be carried out in order to determine the reason behind this increase in the level of urea concentration. Thank you.
Natural compounds that can inhibit 5-alpha reductase 2 was the main aim of this work.
Chemical immunosensor is a device that gives real-time disease information about the composition of a system by connecting chemically selective layer, usually called biorecognition unit, to a transducer. And this is good for point of care detection of pathogens. Because of it, rapid response, high sensitivity, selectivity, and ease of miniaturization. So the biorecognition unit normally uses a PDOS, which is a conductant polymer, by recognizing an amino complex. But because of sensitivity in terms of electrical current conduction, we added hydroxyapatite to the PDOT to form a highly sensitive biorecognition unit. 10% of the hydroxyapatite and 20% were mixed with the PDOT, and after conducting cyclic voltammetry tests and impedometry tests, it was found out that the composite that we produce, that is a 20% hydroxyapatite and 80% PDOT, give a very high conductivity of current, and which is good for cancer detection. Thank you.
My name is Stephen Okukwe, and I'm presenting on missed clinical malaria infections, and these are results from a diagnostic accuracy study in Ashanti region of Ghana. So currently, the WHO recommends all suspected malaria cases to be confirmed by either microscopy or RDTs. However, recent studies have found the accuracy of these particular tests to be quite limited at low parasite density. So we conducted a diagnostic accuracy study in order to assess the extent to which infections are missed by these particular tools, using a highly sensitive quantitative PCR as a reference. So we recruited a total of 1,040 suspected malaria patients, that is individuals who were referred to the lab to go and do a malaria test. And briefly, we had more females than males, and as more than half our said population did not sleep under mosquito nets, and also more than half presented with extreme fever. So unsurprisingly, the PCR had the highest parasite prevalence of 42.1%, followed by RDT with 24.5%, with expert microscopy having the least parasite prevalence of 17.5%. So when we compared these particular results to quantitative PCR, we found RDT to be more sensitive than microscopy. However, both tests had comparable specificity of about 98%. So interestingly, out of the 138 positives which were detected by quantitative PCR, we realized that both microscopy and RDT missed about 186 samples, and this corresponds to about 40% of the clinical infections which were missed by both microscopy and RDT. And this particular result is quite significant because it means that these particular individuals were not treated for malaria. So these people may serve as reservoirs for continual transmission. So we conclude by saying RDT was more sensitive and that these tests missed infections. So we recommend that infection training should be given to improve microscopy, and that there's also the need to improve existing diagnostics. Thank you.
Dear member of jury, good morning. The topic we are going to present to you today is titled Endophytic fungi from Kanango dorata are promising candidates against Entamoeba histolytica. As an introduction, we can say that amoebiasis caused by Entamoeba histolytica is a major public health problem. Moreover, the emergence of resistant strains to available drugs highlights the need to identify new compounds. In this regard, exploring endophytes from plants traditionally used could be a promising approach. Therefore, this work describes the anti-amoebic activity of extracts of endophytic fungi isolated from a Cameroonian medicinal plant, namely Kanango dorata. 10 endophytic fungi grown on potato dextrose broth were subjected to heterocyclic extraction. Afforded extract were tested for amoebicidal activity, and the cytotoxicity activity was evaluated on viral cell line. Extraction yield range from 12 to 200 milligram. Globally, all these extracts show strong amobistal activity, with IC50 varying from 0.39 to 3.57 microgram per milliliter. 8 of the tested extracts show significant selectivity with index greater than 10. This result obtained suggests that endophytic fungi from Canango dorata could serve as a source for the identification of potent amobistal compound. Further bioregarded fractionation is currently ongoing to purify and identify active ingredients. Thank you.
Thank you so much for running those table talks by us. This is just a teaser of what you'd be viewing after lunch. We have a good 2 hours between now and when we have to return back to this room. Lunch is served on the same 2 floors as yesterday. The instructions still remain the same. Please, when you go up, get seated and your table will be called for you to go and pick your choice. Yesterday, the second floor was quite unpopulated, so if I were you, I would head there first today. And right after lunch, please spend the next hour interacting with the guests who are here. Share your research ideas, mold them if you have to, make contacts. That's why one of the very good benefits of the WACCBIP Research Conference. People make connections. You just heard from Lord Bassin how he made a connection that has changed his life. Don't miss this opportunity. You have an hour to view the exhibition booths. You have an hour to also look at the posters. Remember, the posters are being judged and a winner will be picked. So please, if you can contribute to picking a good winner, please, by all means, let us know. Enjoy lunch. We'll be back at 3 o'clock on the dot. Thank you.
There is no promise we cannot keep. There is no commitment we cannot honor. There is no delivery we cannot guarantee. There is only one Bethel Logistics Company Limited, a licensed international freight forwarding and clearing agent, a trusted global brand with an extensive network for the movement of air freight and ocean freight worldwide. We are the trusted name for road freight into Western and Northern Africa. We are fully accredited by Ghana Customs and Excise. We cater for all your freight forwarding, customs clearing and forwarding, cross trades, inland transportation, internal cargo tracking systems, customer freight costing and consultancy, cold chain logistics management, logistics and warehousing, quality inspection, and much more. In Accra, Tema, and Takrady, we are always available to ensure easy movement of cargo countrywide. Call us on 0204 528 900 or 0244 707 859. Email info@bethellogistics.com. Locate us in Accra at North Ridge, adjacent Accra Girls High School, or in Tema at Community 4, or in Takrady on the 2nd floor of the GCB building. Website bethellogistics.com. Bethlog, experts in shipping logistics.
Biotech, Africa's genomics company. Established in 2002 in Pretoria, South Africa, Biotech is a rare and unique company, as suggested by the name, and we thrive to give reliable and efficient services to all our customers. How it all began. With the concept developed in the year 2000 to speed up the arrival of primers in molecular labs in the country, Dr. Oliver Pricer, co-founder and executive director, together with a venture team of 5, secured a loan from Swiss government and got rights to be the very first company to synthesize primers locally, and thus it all began. With capacity to synthesize over 1,000 primers a day, Inqaba remains the only company that does this in Africa. This is made possible by the Mermaid 192E. Using an ABI 3500 exome, we offer DNA sequencing from various templates, and we offer primer design, PCR amplification, cloning of PCR products, fungal and bacterial identification, and much more. In 2007, we launched Next Generation Sequencing Services, which provides NGS projects design inputs, quality control library preparation, and sequencing workflow using either the MySeq, NextSeq, or the PacBio systems. We have more services that include
the Cell Biology Lab, the Medicinal and WACCBIP, the
to share their talent. He had always looked for years. Second postdoc. And so hearing about WACCBIP, hearing what someone like me potentially could thrive, return to Ghana.
The Center of Excellence here. And part of that funding gave us a huge chunk of money to buy core equipment that could be used and shared by all faculty in the department and opened up to faculty within the university and within the sub-region. We have a core facility for flow cytometry. We also have a core facility for sequencing of DNA. Just recently, we've even upgraded it to include a NextSeq Illumina sequencer, which is probably one of 2 in the entire Africa. And that has the capacity to sequence both DNA and RNA, which is world-class. We have a confocal microscope core facility, which can be used to take images of cells and for us to understand actual cell biology as it happens in real time. We also have the protein expression lab that I talked about, and we have state-of-the-art equipment in these laboratories that help us, again, make proteins, express proteins, and take it to the next level of trying to identify what these proteins actually do within cells.
Since I've been in the virology department, I've been doing mostly HIV research, that's my specialty, looking at how the different genotypes we have in Ghana affect the ability of treatment and the progression of the disease. And of course, since the pandemic, we have been right at the forefront of doing research of SARS-CoV-2, that's the virus that causes COVID-19 in Ghana. The HIV research that we've done here at Virology, prior to my coming, there's been some findings that in the Ghanaian population, some of the genes that may be involved in progression are deleted in some of the population. Actually, a few of my colleagues did that work. And since I've been here, we've been looking at the new drug which has been introduced in Ghana, dolutegravir, and our data, which is yet to be published, is showing that it is one of the best drugs to be used, and we are also selecting some mutations for this drug.
My experience in WACCBIP here has been very great. I came here in 2014. So after my undergraduate, I joined the lab in 2015 and I served as a research assistant. When I entered, I didn't know anything about research, but I've got opportunity through mentorship and training. Yeah, sure. I think there's fairness in the enrollment process. For what I know, I was fortunate after my master's, first year master's, I was enrolled into the PhD program. Then what I have seen so far, I realized before you get admission to the program, you write in exams and they try to balance, look at gender balance and also the international and the local students. So there's fairness in the selection process. So my PhD thesis or project seeks to develop novel diagnosis for detecting Plasmodium species. So Plasmodium species are parasite that causes malaria, and currently we have RDTs and microscopy at the point of care for detecting the species. But unfortunately, these tools are not sensitive or specific enough for detecting the species. So there are more reliable tools, which are the PCR or molecular-based diagnostic tools. And if I can give you an example, that's what we are using to detect COVID now. But the unfortunate thing is these tools are not readily available in terms of the reagent. So my project seeks to develop these reagents here in Ghana so that we can make them readily available and also cost-effective for detecting infectious diseases.
The center runs 3 programs at the master's level, which includes Master of Philosophy in Molecular Cell Biology of Cell and Infectious Diseases, Master of Philosophy in Biochemistry, and Master of Philosophy in Molecular Biology. The PhD level includes PhD in Biochemistry and PhD in Molecular and Cell Biology of Infectious Diseases. The center also engages the community in its activities from time to time.
What was of common interest to all of us was that we wanted to create a world-class research and training environment to do basic science. Now, this was led by me, but with support from, several colleagues here at the Department of Biochemistry and also at the Noguchi Memorial Institute for Medical Research. In terms of the resources we need sustainable funding, predictable funding. If you look at all the countries that have developed, they have predictable funding for research and for science. If you look at the USA, they have something called the NIH, which they pump billions and billions of dollars in every year as part of their national budget. It's locked in. Ghana, we still don't have that. We've been talking about a research and innovation bill for a long time. Still not been enacted. We need to have such things so that we as scientists, there's a fund that we can apply to access funds to do research and to innovate on a sustainable basis. As we say it, we compete for funds from overseas, and that is not predictable nor sustainable because a lot of the funders, they keep asking us, how much is your government contributing? Okay, we've given you 8 million, we've given you 6 million, we've given you 5 million. How much is your government contributing?
Everybody knows that Africa has problems. Our problems are all over the news. We get offended when people point out only the problem. But it's also very clear that the only people who will solve Africa's problems are Africans themselves. We are waiting for malaria vaccines to be developed in Europe and North America for us to use. We are waiting for people, for President Jimmy Carter to come and eradicate guinea worm in Ghana. If Africa and in Ghana is going to achieve what I believe and we all believe we can achieve, we have to take responsibility for our problems and come up with locally relevant solutions to solve those problems. For me, WACCBIP represents a place that is trying to do its bit to contribute towards this. We are trying to train the next generation of scientists, not to come and work at WACCBIP. We are training the next generation of scientists to think of ways to solve problems they see in their own community.
International students are helped through WACCBIP to obtain accommodation, and basically what we do is that we liaise with the hostels and the halls on campus and arrange some of the rooms for them before they actually come in. At WACCBIP, we provide at least 2 forms of scholarship. One from the ACE World Bank grant, and the other from the Delta's program.
Because we are in the tropics, a lot of patients report to the hospitals with high temperature. This is usually attributed to malaria, but we found out that malaria answers, let's say, 40% of those cases. For the other 60%, they are presumptively treated as malaria. And in most cases, the patients keep going back because it's not malaria. So what else is causing it? It's not clearly known. And the diagnostic systems in the hospitals are not advanced enough to take care of a variety of disease conditions. So this project was undertaken by one Nicholas, who looked at a whole panel of pathogens. So this was a discovery project. Okay, so once a child reports with fever and it's not attributed to anything else but the infection, and they check for malaria and malaria is not there, they then take those samples and look for all manner of pathogens. Okay, and they were able to discover a whole variety of viruses and bacterial pathogens that explain those other febrile illness. So going forward, that would instruct a new way of attending to patients in that category, so that we can. What we haven't done is to develop diagnostics that can be used in the hospitals, but that is the next phase that we can look at. The second is that
In the lab, you face consistent pressure to do more with less while trying to keep up with a growing workload. Success means having instruments that you can rely on to overcome diverse challenges. The Agilent 6470B Triple Quadrupole LCMS, the latest version of our trusted workhorse, gives you the robustness and versatility you need to overcome these challenges. The 6470B has a proven reputation for robustness with non-stop operation, backed up by thousands of users, with numerous application notes and peer-reviewed articles to reduce downtime and increase efficiency. Enhance versatility with a wide array of ion sources to analyze any application. Ion source options include ESI, the standard choice to broadly ionize analytes of interest, AJS, which enhances the desolvation power of standard ESI for ultra-high sensitivity, APCI, for increased signal-to-noise for very small analytes, and MMI, to carry out ESI and APCI simultaneously for a complementary analysis. With the new WACCBIP, you can reduce maintenance time by 92%. WACCBIP lets you remove the capillary and run the cleaning cycle in approximately 30 minutes, compared to about 6 hours for maintenance by venting and disassembly. So you can gain back valuable lab time and keep up with high demands. From analyzing environmental contaminants and ensuring food safety to complex life science research, the 6470B meets your need for reliable, accurate results. These accurate results are provided by our Mass Hunter software, including Quant My Way, which enables you to personalize the software with tools that highlight only the capabilities of interest. With comprehensive end-to-end workflow solutions and application services, you can ensure proper sample preparation, expertly developed acquisition and data analysis protocols, and fully customizable reports to get the answers you need as soon as possible. Our solutions are designed to work together to provide efficiency gains that help you get more done and reduce operation costs. The Agilent 6470B Triple Quadrupole LCMS is unbelievably robust to increase your lab efficiency and remarkably versatile to keep up with your evolving demands. Visit agilent.com/chem/6470B for more information. Decades of innovation have brought us into a new era of mass spectrometry with the design of Altivo, Agilent's revolutionary triple quadrupole LC-MS. At just 1/3 the size of our previous triple quad LC-MS instruments, Altivo integrates seamlessly into the stack of the Agilent Infinity Lab LC System Series, saving bench space while offering the same power and accuracy of our larger systems. The footprint of Altivo is 70% smaller than that of similar LC triple quadrupole mass spectrometers, so you can triple your lab's capacity in the same space. The efficiency-optimized Agilent Infinity Lab LC Series system can sit directly on top of Altivo, making it the perfect partner for high-throughput mass spectrometry. The innovations that enabled Altivo were specifically designed to maximize quantitative performance, enhance instrument reliability and robustness, and promote greater uptime. The more ions you get into the instrument, the better your MS results. With Agilent's Jetstream ion source, a superheated nitrogen sheath gas surrounds the electrospray to quickly and efficiently desolvate the spray plume and tightly cluster the resulting ion cloud, maximizing the number of ions that are created and transported into the LC-MS. WACCBIP features fast ion injector exchange without the need for system venting and improved diagnostics that guide the operator to appropriate corrective action, leading to less downtime for maintenance and higher throughput for your laboratory. Once the ions enter Altivo, they pass through our innovative precision-manufactured Cyclone Ion Guide. With its twisted and tapered dual hexapole configuration, the Cyclone Ion Guide provides maximum ion capture, compression, and transmission across a large pressure drop region, giving you better, more reproducible results. From the Cyclone Ion Guide, the ions continue to the first quadrupole. Better mass transmission improves analytical results. Each quadrupole assembly includes DC-only, wafer-thin pre- and post-filters, which cancel fringe field regions at the quadrupole entrance and exit to improve ion transmission. This allows for faster switching, enabling more MRMs to be performed. With the analyte selected in Q1, the ions pass through the vortex collision cell. Its innovative twisted and tapered dodecapole hexapole geometry maximizes ion collection, fragmentation, and transmission. The high-efficiency detector offers a wide dynamic range to ensure linear responses for your most challenging analyses. The detector was designed to be easily replaced by anyone in the lab. Working together, the compression and focusing of the Cyclone Ion Guide, the improved ion transmission through Q1 with its pre- and post-filters, the optimized fragmentation in the vortex collision cell, the improved transmission through Q2, and precise data capture of our innovative detector technology give you better MS results. Altivo uses an entirely redesigned electronics architecture, making it the smartest LC-MS on the market. New intelligent diagnostics tools enable Altivo to monitor several parameters in real time to ensure that it's operating at peak performance. If it isn't, it lets you know so that you can take appropriate action. For food safety labs, Altivo offers the selectivity and sensitivity needed to confidently monitor for and identify contaminants, even in difficult matrices, helping keep the food supply safe. For environmental analysis, Altivo provides robust and accurate measurements down to sub-part per billion levels and integrates with Mass Hunter software for powerful data collection, processing, and reporting to help you keep up with newly emerging pollutants. Reshape expectations, reinvent capabilities, and redefine what's possible when small meets powerful. The Agilent Altivo Triple Quadrupole Mass Spectrometer.
There is no promise we cannot keep. There is no commitment we cannot honor. There is no delivery we cannot guarantee. There is only one Bethel Logistics Company Limited, a licensed international freight forwarding and clearing agent, a trusted global brand with an extensive network for the movement of air freight and ocean freight worldwide. We are the trusted name for road freight into Western and Northern Africa. We are fully accredited by Ghana Customs and Excise. We cater for all your freight forwarding, customs clearing and forwarding, cross trades, inland transportation, internal cargo tracking systems, customer freight costing and consultancy, cold chain logistics management, logistics and warehousing, quality inspection, and much more. In Accra, Tema, and Takrady, we are always available to ensure easy movement of cargo countrywide. Call us on 0204 528 900 or 0244 707 859. Email info@bethellogistics.com. Locate us in Accra at North Ridge, adjacent Accra Girls High School, or in Tema at Community 4, or in Takrady on the second floor of the GCB building. Website bethellogistics.com. Bethlog, experts in shipping logistics.
Welcome to Biotech, Africa's genomics company. Established in 2002 in Pretoria, South Africa, Biotech is a rare and unique company, as suggested by the name, and we thrive to give reliable and efficient services to all our customers. How it all began. With the concepts developed in the year 2000 to speed up the arrival of primers in molecular labs in the country, Dr. Oliver Pricer, co-founder and executive director, together with a venture team of 5, secured a loan from Swiss government and got rights to be the very first company to synthesize primers locally, and thus it all began. With capacity to synthesize over 1,000 primers a day, Inqaba remains the only company that does this in Africa. This is made possible by the Mermaid 192E. Using an ABI 3500 exome, we offer DNA sequencing from various templates, and we offer primer design, PCR amplification, cloning of PCR products, fungal and bacterial identification, and much more. In 2007, we launched Next Generation Sequencing Services, which provides NGS projects design inputs, quality control library preparation, and sequencing workflow using either the MySeq, NextSeq, or the PacBio systems. We have more services that include SNP genotyping and methylation analysis using the Agena Biosciences mass array systems, bioinformatics solutions that complement our NGS services, and qPCR analysis. We also are a distributor of strong international brands, which includes New England Biolabs, Zymo Research, Eppendorf, Glentham, CG, Nolato Treff, Charlab, just to name a few. Inqaba boasts a responsive technical team with expert knowledge in the services we provide. We also have a well-trained engineering staff making sure that the installation and servicing of instruments purchased at Inqaba is well taken care of. A fleet of vehicles commissioned for local trips and deliveries assures we arrive at your lab on time. In the past 18 years, we have started subsidiaries in East and West Africa, and we have offices in Kenya, Nigeria, Ghana, Tanzania, Senegal, and still maintain a strong presence in our neighboring countries. All this is achieved with a highly trained and professional team that is always ready to assist. Welcome to Inqaba Biotech.
If you need a reliable procurement and supply chain partner, look no further.
Global procurement company Excelsior Global Limited is here for you.
We work across multiple industries and sectors with one goal in mind, ensuring that our clients focus on core businesses while outsourcing procurement.
Excelsior Global takes care of purchasing process while reducing all procurement and supply chain costs.
We offer a wide variety of procurement solutions connected mainly with medical, COVID-19 antigen test kits, COVID-19 antibody test kits, COVID-19 antigen test kits, virus transport media, specimen collection swab, sample preservation tubes, HIV test strips, hepatitis C test strips, hepatitis B test kits, syphilis test strips, glaucometer test strips, pregnancy test kits, hepatitis C, HCV, examination gloves, syringe, surgical gloves.
Excelsior Global makes procurement easy, simple, on time, and most important, the job is in safe hands.
We do what others don't.
Visit our office at 35 Patrice Lumumba Road, Airport Residential Area, Accra.
For more information, call us at 0559 122 092.
Mail us at info@excelsiorgh.com
or visit www.excelsiorgh.com.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, was established in 2014 as one of the World Bank's Africa Centers of Excellence. WACCBIP is located on the campus of the University of Ghana, Legon.
WACCBIP was established as one of the World Bank African Centers of Excellence. And the inspiration behind it is basically that a number of us who were at the Department of Biochemistry and the Noguchi Memorial Institute for Medical Research, we had trained outside and we did PhDs in various science disciplines outside the country. When we got back and realized that there was not much by way of facilities to train scientists, we saw this as an opportunity to create an environment which would enable many young scientists to get the opportunity to get the quality of training that we got overseas.
This world-class research center started off as an infectious disease center, but along the line, it's added non-communicable diseases to its focus areas.
WACCBIP is providing something to local and regional students that we probably don't have in many places within the country or within the region. Facilities, financial resources, as well as an environment that encourages student learning and research. And that has significantly increased students' output in the Department of Biochemistry, Cell and Molecular Biology.
WACCBIP has a whole range of collaborators at the institutional, national, regional, and international levels. These collaborations include the Noguchi Memorial Institute for Medical Research, Ghana, the Department of Biomedical Sciences at the University of Ghana, among other departments, the Ghana Health Service, and the Ministry of Health. The center also has collaborations with institutions within and outside Africa.
Currently, there are about 6 labs in the center, and these labs do research based on the faculty who is in charge of these labs. We have a malaria group that has 3 different units. There's an immunology unit, there's a vaccine development unit, and also a transmission unit in there. We also have a molecular biology lab that has 2 faculty in charge there. One does work with African animal trypanosomes, and the other one, who is myself, works in Buruli ulcer and antimicrobial resistance. We have a huge virology group. They do research, anything that has to do with viruses. So they do HIV research. They even look at some bat viruses and gastroenteric viruses. More recently, too, we've added a COVID lab, and this lab does routine testing for COVID positivity and negativity. And we also look for seroprevalence across the population. So people who have been prior exposed to COVID, but maybe have been asymptomatic, so they don't know whether they had COVID or not. And we've actually just recently published a paper on our findings.
The virology laboratory of WACCBIP was set up right from the beginning with one main lab, and they were doing things like rotaviral, researching into rotaviral infections in Ghana. The virology lab at WACCBIP was also involved in the first identification of a dengue case in Ghana, and also doing HIV research, as well as research on viruses which are in bats, but which might be have a risk of causing disease in humans. So these are filoviruses, the same type of viruses that cause Ebola. So that's work that was being done.
The center currently has 6 research laboratories, namely the Cell Biology and Immunology Lab, commonly known as the Malaria Lab, the Molecular Biology Lab, the Virology Lab, the Medicinal and Natural Products Lab, the Next Generation Sequencing Lab, and the Forensic Biochemistry Lab. At WACCBIP, the scientific tools and equipment are always and readily available to students, regardless of the courses being studied at the center.
Really, the driving force was to try to create a world-class research and training environment where young African scientists from across the continent can come here and develop their talent and do high-quality research that can be comparable to research that's done in any part of the world. So basically, that was the vision that we were pursuing.
Throughout my academic journey, I'd always looked for opportunities to come back home. My plan has always been to establish myself, to spend my most productive, my most useful years in Africa and hopefully in Ghana. So even though I studied abroad altogether for 13 years, followed by 6 years as a postdoc also abroad. So altogether, I was away from Ghana for about 18 years. I was always looking for opportunities to return. So during my second postdoc, I actually met Professor Wandare, the director of WACCBIP, in London when he came to actually receive an award. And that was the first time I heard about WACCBIP. And so hearing about WACCBIP, hearing what Professor Wandare was trying to do in Ghana, I felt that it was a place that would welcome someone like me, a place that someone like me potentially could thrive. So I began to look for opportunities to return, and I was able to get funding to support my return to Ghana, and so applied for a position and was appointed as a research fellow. It was basically a fulfillment of something I'd always wanted to do, and WACCBIP stood out as a place that, even though it was a young center, was led by somebody who himself was very dynamic, who identified a lot with the ideals that I myself identify with. And so was somebody that I thought I could work for, somebody who could mentor me.
WACCBIP has evolved to become a world-class research center, thriving on initial funding from the World Bank and subsequent support from the Wellcome Trust and the Africa Academy of Science.
We were very fortunate to get the funding through World Bank to establish the Center of Excellence here. And part of that funding gave us a huge chunk of money to buy core equipment that could be used and shared by all faculty in the department and opened up to faculty within the university and within the sub-region. We have a core facility for flow cytometry. We also have a core facility for sequencing of DNA. Just recently, we've even upgraded it to include a NextSeq Illumina sequencer, which is probably one of two in the entire Africa. And that has the capacity to sequence both DNA and RNA, which is world-class. We have a confocal microscope core facility, which can be used to take images of cells and for us to understand actual cell biology as it happens in real time. We also have the protein expression lab that I talked about, and we have state-of-the-art equipment in these laboratories that help us, again, make proteins, express proteins, and take it to the next level of trying to identify what these proteins actually do within cells.
Since I've been in the virology department, I've been doing mostly HIV research, that's my specialty, looking at how the different genotypes we have in Ghana affect the ability of treatment and the progression of the disease. And of course, since the pandemic, we have been right at the forefront of doing research of SARS-CoV-2, that's the virus that causes COVID-19 in Ghana. The HIV research that we've done here at Virology, prior to my coming, there's been some findings that in the Ghanaian population, some of the genes that may be involved in progression are deleted in some of the population. Actually, a few of my colleagues did that work. And since I've been here, we've been looking at the new drug which has been introduced in Ghana, dolutegravir, and our data, which is yet to be published, is showing that it is one of the best drugs to be used, and we are also selecting some mutations for this drug.
My experience in WACCBIP here has been very great. I came here in 2014. So after my undergraduate, I joined the lab in 2015 and I served as a research assistant. When I entered, I didn't know anything about research, but I've got opportunity through mentorship and training. I've had great experience here. Sure, I think there's fairness in the enrollment process. For what I know, I was fortunate after my first year master's, I was enrolled into the PhD program. Then what I have seen so far, I realized before you get admission to the program, you write in exams and they try to balance, look at gender balance and also the international and the local students. So there's fairness in the selection process. So my PhD thesis or project seeks to develop novel diagnoses for detecting Plasmodium species. So Plasmodium species are parasite that causes malaria, and currently we have RDTs and microscopy at the point of care for detecting these species. But unfortunately, these tools are not sensitive or specific enough for detecting these species. So there are more reliable tools, which are the PCR or molecular-based diagnostic tools. And if I can give you an example, that's what we are using to detect COVID now. But the unfortunate thing is these tools are not readily available in terms of the reagents. So my project seeks to develop these reagents here in Ghana so that we can make them readily available and also cost-effective for detecting infectious diseases.
The center runs 3 programs at the master's level, which includes Master of Philosophy in Molecular Cell Biology of Cell and Infectious Diseases, Master of Philosophy in Biochemistry, and Master of Philosophy in Molecular Biology. The PhD level includes PhD in Biochemistry and PhD in Molecular and Cell Biology of Infectious Diseases. The center also engages the community in its activities from time to time.
What was of common interest to all of us was that we wanted to create a world-class research and training environment to do basic science. Now, this was led by me, but with support from, several colleagues here at the Department of Biochemistry and also at the Noguchi Memorial Institute for Medical Research. In terms of the resources we need sustainable funding, predictable funding. If you look at all the countries that have developed, they have predictable funding for research and for science. If you look at the USA, they have something called the NIH, which they pump billions and billions of dollars in every year as part of their national budget. It's locked in. Ghana, we still don't have that. We've been talking about a research and innovation bill for a long time. Still not been enacted. We need to have such things so that we as scientists, there's a fund that we can apply to access funds to do research and to innovate on a sustainable basis. As we say, we compete for funds from overseas, and that is not predictable nor sustainable, because a lot of the funders, they keep asking us, how much is your government contributing? Okay, we've given you 8 million, we've given you 6 million, we've given you 5 million. How much is your government contributing?
Everybody knows that Africa has problems. Our problems are all over the news. We get offended when people point out only the problem. But it's also very clear that the only people who will solve Africa's problems are Africans. We are waiting for malaria vaccines to be developed in Europe and North America for us to use. We are waiting for people, for President Jimmy Carter to come and eradicate guinea worm in Ghana. If Africa and in Ghana is going to achieve what I believe and we all believe we can achieve, we have to take responsibility for our problems and come up with locally relevant solutions to solve those problems. For me, WACCBIP represents a place that is trying to do its bit to contribute towards this. We are trying to train the next generation of scientists, not to come and work at WACCBIP. We are training the next generation of scientists to think of ways to solve problems they see in their own community.
International students are helped through WACCBIP to obtain accommodation, and basically what we do is that we liaise with the hostels and the halls on campus and arrange some of the rooms for them before they actually come in. At WACCBIP, we provide at least two forms of scholarship. One from the ACE World Bank grant, and the other from the Delta's program.
Because we are in the tropics, a lot of patients report to the hospitals with high temperature. This is usually attributed to malaria, but we found out that malaria answers, let's say, 40% of those cases. For the other 60%, they are presumptively treated as malaria. And in most cases, the patients keep going back because it's not malaria. So what else is causing it? It's not clearly known. And the diagnostic systems in the hospitals are not advanced enough to take care of a variety of disease conditions. So this project was undertaken by one Nicholas, who looked at a whole panel of pathogens. So this was a discovery project. Okay, so once a child reports with fever and it's not attributed to anything else but infection, and they check for malaria and malaria is not there, they then take those samples and look for all manner of pathogens. Okay, and they were able to discover a whole variety of viruses and bacterial pathogens that explain those other febrile illness. So going forward, that would instruct a new way of attending to patients in that category, so that we can. What we haven't done is to develop diagnostics that can be used in the hospitals, but that is the next phase that we can look at. The second is diagnostic technologies that have been developed by two PhD students, Francis Krampa and Felix Ansa. So here they are looking for electronic ways of detecting malaria. Okay, so what we have in the system right now is glucometer. You put a drop of blood and it tells you the level of glucose in your blood. We are trying to do the same for malaria so that people can even test at home, right, and not have to go to the hospital when this becomes available on the market. The third one is a project that concerns a new way of developing antibiotics, and what we are trying to do is to target what we call the genome stability of pathogens. This has not been done before, and we are pioneering that from this center for the first time. And we're hoping that over the years we will make progress that will lead to the next generation of antibiotics.
The first output that we had from our sequencing was a manuscript which was published, and that was the first 46 sequences of SARS-CoV-2 in Ghana. We published that last year, and since then we've done almost 1,000 sequences. So we are preparing that also to publish it. And why that is important is we are tracking basically the changes in the virus locally, because there's, if you look at literature or the news, you see how it's changing globally, but you have to know the local outbreak, you have to know the local epidemic. And what we see in Ghana is that sometimes the variants which are increasing are not necessarily what is increasing in the rest of the world. For now, for instance, we have Delta, which is also on the rise in Ghana. But there's another variant which is nobody's talking about, but it's rising almost faster than Delta in Ghana. So that's the sort of things that we do. And when we get the results, we write what we call policy briefs, advising the government that this is what we found. Earlier on in this year, when we did our sequencing of airport samples, and we first found the Alpha variant, that's the UK variant. And then we found it in local circulation, and we wrote we communicated to the government, and that was what led to the reinstatement of some of the restrictions, like restricting the funeral size, party size, and things like that. That was as a direct result of the work that we're doing.
The key message that we want to put out there is that Africa has a lot of talent. If you look across the world, there are African scientists doing great things all over the place. Some of these COVID vaccines that we are seeing, African scientists played key roles in developing them so quickly in different countries. If we create the right environment for them, a lot of these people will come back home. We have already brought 7 scientists, African scientists who were in the diaspora. We brought them here. And the reason why they were able to come back, or they were convinced to come back, is because they came and looked around, like you guys have looked around, and they saw that they can do good science here, and that this environment looks like where they've been working in the UK or in the US, and they are convinced that this is world-class enough. If we create that, we can start to bring all our talent back, and Africa can start to develop our own vaccines, can develop our own drugs, we can be more innovative and build new technologies that we can even export to other parts of the world, and not sit as we are now waiting for people to develop vaccines, use for their people. When they've had enough, then they share with us. That's where we are now. Okay? That situation has to change. That means we have to take matters into our own hands. That means the African governments have to start to put some money into science on a consistent basis. Let's have a fund, such as we have National Health Insurance Fund, we have GET Fund, we have all these funds that a percentage of the taxes goes directly into that fund every year. So that fund is available for scientists to access. Let's do that. Let's put some money aside for science, because this type of investment have to be done for us to develop. Otherwise, we keep marking time. We keep just treading water, because if you keep relying on people for your innovations, that means you are not making progress. I keep telling people that if you think about investment in science, think about it like the way we invested in petroleum exploration. Ever since I was a child, I was hearing about GMPC spending billions and billions of dollars doing oil exploration. Okay? They didn't find oil until just maybe 10 years ago or less, after spending billions and billions of dollars. Nobody complained that they were spending billions of dollars, because they knew that when they found oil, they'll get a lot of money from it. It's the same with science. Don't think about what you'll get tomorrow. You know, don't say that, oh, I've given you 200,000, so what have you brought after 2 weeks? No, it doesn't work that way. It has to be consistent investment over time. And then when you succeed, it will pay for itself. Okay. If we made a vaccine right now, can you imagine if we had a COVID vaccine and we're supplying to the whole of Africa? Can you imagine how much money Ghana will be making? But if you don't invest, you won't get. You won't get there. So you have to invest, and then eventually you will hit the jackpot.
If there is any biomedical research center that promotes innovation and research, or that provides its students with unmatched financial resources and an environment for learning and research, it is the West Africa Center for Cell Biology of Infectious Pathogens, WACCBIP, providing world-class postgraduate training and research into tropical diseases in sub-Saharan Africa.
In the lab, you face consistent pressure to do more with less while trying to keep up with a growing workload. Success means having instruments that you can rely on to overcome diverse challenges. The Agilent 6470B Triple Quadrupole LCMS, the latest version of our trusted workhorse, gives you the robustness and versatility you need to overcome these challenges. The 6470B has a proven reputation for robustness with non-stop operation, backed up by thousands of users, with numerous application notes and peer-reviewed articles to reduce downtime and increase efficiency. Enhance versatility with a wide array of ion sources to analyze any application. Ion source options include ESI, the standard choice to broadly ionize analytes of interest, AJS, which enhances the desolvation power of standard ESI for ultra-high sensitivity, APCI, for increased signal-to-noise for very small analytes, and MMI, to carry out ESI and APCI simultaneously for a complementary analysis. With the new WACCShield, you can reduce maintenance time by 92%. WACCShield lets you remove the capillary and run the cleaning cycle in approximately 30 minutes, compared to about 6 hours for maintenance by venting and disassembly. So you can gain back valuable lab time and keep up with high demands. From analyzing environmental contaminants and ensuring food safety to complex life science research, the 6470B meets your need for reliable, accurate results. These accurate results are provided by our Mass Hunter software, including Quant My Way, which enables you to personalize the software with tools that highlight only the capabilities of interest. With comprehensive end-to-end workflow solutions and application services, you can ensure proper sample preparation, expertly developed acquisition and data analysis protocols, and fully customizable reports to get the answers you need as soon as possible. Our solutions are designed to work together to provide efficiency gains that help you get more done and reduce operation costs. The Agilent 6470B Triple Quadrupole LC-MS is unbelievably robust to increase your lab efficiency and remarkably versatile to keep up with your evolving demands. Visit agilent.com/chem/6470B for more information. Decades of innovation have brought us into a new era of mass spectrometry with the design of Altivo, Agilent's revolutionary triple quadrupole LC-MS. At just 1/3 the size of our previous triple quad LC-MS instruments, Altivo integrates seamlessly into the stack of the Agilent Infinity Lab LC System Series, saving bench space while offering the same power and accuracy of our larger systems. The footprint of Altivo is 70% smaller than that of similar LC triple quadrupole mass spectrometers, so you can triple your lab's capacity in the same space. The efficiency-optimized Agilent Infinity Lab LC Series system can sit directly on top of Altivo, making it the perfect partner for high-throughput mass spectrometry. The innovations that enabled Altivo were specifically designed to maximize quantitative performance, enhance instrument reliability and robustness, and promote greater uptime. The more ions you get into the instrument, the better your MS results. With Agilent's Jetstream ion source, a superheated nitrogen sheath gas surrounds the electrospray to quickly and efficiently desolvate the spray plume and tightly cluster the resulting ion cloud, maximizing the number of ions that are created and transported into the LC-MS. WACCBIP features fast ion injector exchange without the need for system venting and improved diagnostics that guide the operator to appropriate corrective action, leading to less downtime for maintenance and higher throughput for your laboratory. Once the ions enter Altivo, they pass through our innovative precision-manufactured Cyclone Ion Guide. With its twisted and tapered dual hexapole configuration, the Cyclone Ion Guide provides maximum ion capture, compression, and transmission across a large pressure drop region, giving you better, more reproducible results. From the Cyclone Ion Guide, the ions continue to the first quadrupole. Better mass transmission improves analytical results. Each quadrupole assembly includes DC-only, wafer-thin pre- and post-filters, which cancel fringe field regions at the quadrupole entrance and exit to improve ion transmission. This allows for faster switching, enabling more MRMs to be performed. With the analyte selected in Q1, the ions pass through the vortex collision cell. Its innovative twisted and tapered dodecapole hexapole geometry maximizes ion collection, fragmentation, and transmission. The high-efficiency detector offers a wide dynamic range to ensure linear responses for your most challenging analyses. The detector was designed to be easily replaced by anyone in the lab. Working together, the compression and focusing of the Cyclone Ion Guide, the improved ion transmission through Q1 with its pre- and post-filters, the optimized fragmentation in the vortex collision cell, the improved transmission through Q2, and precise data capture of our innovative detector technology give you better MS results. Altivo uses an entirely redesigned, making it the smartest LC-MS on the market. New intelligent diagnostics tools enable Altivo to monitor several parameters in real time to ensure that it's operating at peak performance. If it isn't, it lets you know so that you can take appropriate action. For food safety labs, Altivo offers the selectivity and sensitivity needed to confidently monitor for and identify contaminants, even in difficult matrices, helping keep the food supply safe. For environmental analysis, Altivo provides robust and accurate measurements down to sub-part per billion levels and integrates with Mass Hunter software for powerful data collection, processing, and reporting to help you keep up with newly emerging pollutants. Reshape expectations, reinvent capabilities, and redefine what's possible when small meets powerful. The Agilent Ultivo Triple Quadrupole Mass Spectrometer.
There is no promise we cannot keep. There is no commitment we cannot honor. There is no delivery we cannot guarantee. There is only one Bethel Logistics Company Limited, a licensed international freight forwarding and clearing agent, a trusted global brand with an extensive network for the movement of air freight and ocean freight worldwide. We are the trusted name for road freight into Western and Northern Africa. We are fully accredited by Ghana Customs and Excise. We cater for all your freight forwarding, customs clearing and forwarding, cross trades, inland transportation, internal cargo tracking systems, customer freight costing and consultancy, cold chain logistics management, logistics and warehousing, quality inspection, and much more. In Accra, Tema, and Takrady, we are always available to ensure easy movement of cargo countrywide. Call us on 0204 528 900 or 0244 707 859. Email info@bethellogistics.com. Locate us in Accra at North Ridge, adjacent Accra Girls High School, or in Tema at Community 4, or in Takrady on the second floor of the GCB building. Website bethellogistics.com. Bethelog, experts in shipping logistics.
Hello, how are you doing today? Oh no, I need a little sample of your blood for the lab. It's not painful, just a little sample of your blood. It's not painful, it's not painful. Blood.
Is everything okay?
No, please, there's a problem.
What is it?
I don't know how to communicate to the patients that we need a sample of her blood for the lab.
Oh, no. You see, that is why we need sign language interpreters in all health facilities. Don't worry, I'll handle it.
Okay.
Bring her to the lab.
Okay, please, I'll do that. Doctor, we are here.
Oh, good. I hope you are well. Don't worry, we are here to help you, all of us. To do that, we need a sample of your blood. A sample, Don't be afraid. Your blood, small sample, okay? For our diagnosis, you see, please, it's okay, take it easy. Don't be afraid. Oh my God. How am I going to explain this to her? It's okay. You take her back to the ward. You'll be fine, okay? You need a sign language interpreter.
Hey, stop looking at them. Who told you I'm interested? I can see it in your eyes. See. Doris, they are deaf and dumb people. It's true. I hear these people are cares though. How do you even introduce them as your friends?
Every physical condition has its medical cause. It has nothing to do with curses.
Oh, hello, sir. Your money.
Oh, I see. You're not deaf?
No, I'm not deaf.
That is interesting.
Yes.
Aha. Oh, okay. I will need your assistance to communicate with a deaf patient.
Oh, okay. Which hospital?
Legon Hospital.
Wow. Actually, I've just been transferred from the Alpha Clinic to the X-ray department.
Really?
Yes.
Wonderful. That makes my work easier.
Yes. I think I can help.
Yes.
Okay.
Please.
Okay.
This is what then we guess this for this.
Let me see how far.
You!
Hey! What is this?
Masha!
What is it?
Warn your son! Warn your son! Or else you will see what you don't want to see and hear what you don't want to hear. How many times have I not warned him not to play with my son or come to my house?
Oh, Eja Pia. What's wrong? If Kofi plays with your son, are we not from the same clan?
Hey, don't you know there is a case on you and your husband?
Me and my husband?
Yes.
Me and my husband?
Yes.
Very good. My husband is in.
Did you say case? All right. We'll see who that case is on.
Deal with him. Case him. Deal with him. I'm shipping. Come on.
Doctor, we can't find Amma.
Where is she?
I don't know. We've looked everywhere.
What?
Yes.
In that critical condition? Oh God, this is unbelievable. We have to find her. We have to find her at all costs. I'll handle it.
Thank you.
My friend, thank you very much for coming. I'll call you when she turns up.
I'll shout. What more tests do you need? We know the people who did all the antenatal tests. How can you tell us our child is deaf? We will solve our own problem. Let's go.
Excuse me, sir. Is there any problem?
What I say is final. We are not going to any pastor. We are going straight to the village.
I am not ready to take my daughter to the shrine.
No one in my family is deaf. And your family too, there is none. So how can you tell me my daughter is deaf? We must look into it. This is purely spiritual.
What about going to do the test? So the doctor can determine how to help her.
It is my daughter who is deaf. What do they need my blood for?
Our blood. I think that is what will help her.
Blood is sacred. I am not giving my blood to any doctor. I will call Papa at the village to arrange a meeting with the diviner.
Thank you.
Oh, great diviner, eye of the gods, the great owl whose gaze pierces through the thickest shadow and the darkest night. There is a deity that must be appeased in your family. She has been abandoned and has come with a vengeance of deafness. You must pay the due and purge your child. 3 rams and 3,000 Ghana cedis.
Your Highness, can we pay in cash?
Oh, the gods can take cash, even mobile money. Pala wenzu.
Welcome to Biotech, Africa's genomics company. Established in 2002 in Pretoria, South Africa, Biotech is a rare and unique company, as suggested by the name, and we thrive to give reliable and efficient services to all our customers. How it all began. With the concept developed in the year 2000 to speed up the arrival of primers in molecular labs in the country, Dr. Oliver Pricer, co-founder and executive director, together with a venture team of 5, secured a loan from Swiss government and got rights to be the very first company to synthesize primers locally, and thus it all began. With capacity to synthesize over 1,000 primers a day, Inqaba remains the only company that does this in Africa. This is made possible by the Mermaid 192E. Using an ABI 3500 exome, we offer DNA sequencing from various templates, and we offer primer design, PCR amplification, cloning of PCR products, fungal and bacterial identification, and much more. In 2007, we launched Next Generation Sequencing Services, which provide NGS projects design inputs, quality control library preparation, and sequencing workflow using either MySeq, NextSec, or the PacBio systems. We have more services that include SNP genotyping and methylation analysis using the Agena Biosciences mass array systems, bioinformatics solutions that complement our NGS services, and qPCR analysis. We also are a distributor of strong international brands, which includes New England Biolabs, Zymo Research, Eppendorf, Glentham, CG, Nolato Treff, Charlab, just to name a few. Inqaba boasts a responsive technical team with expert knowledge in the services we provide. We also have a well-trained engineering staff making sure that the installation and servicing of instruments purchased at Inqaba is well taken care of. A fleet of vehicles commissioned for local trips and deliveries assures we arrive at your lab on time. In the past 18 years, we have started subsidiaries in East and West Africa, and we have offices in Kenya, Nigeria, Ghana, Tanzania, Senegal, and still maintain a strong presence in our neighboring countries. All this is achieved with a highly trained and professional team that is always ready to assist. Welcome to Inqaba Biotech.
If you need a reliable procurement and supply chain partner, look no further.
Global procurement company Excelsior Global Limited is here for you.
We work across multiple industries and sectors with one goal in mind, ensuring that our clients focus on core businesses while outsourcing procurement.
Excelsior Global takes care of purchasing process while reducing all procurement and supply chain costs.
We offer a wide variety of procurement solutions connected mainly with medical, COVID-19 antigen test kits, COVID-19 antibody test kits, COVID-19 antigen test kits, virus transport media, specimen collection swab, sample preservation tubes, HIV test strips, hepatitis C test strips, hepatitis B test kits, syphilis test strips, glaucometer test strips, pregnancy test kits, hepatitis C, HCV, examination gloves, syringe, surgical gloves.
Excelsior Global makes procurement easy, simple, on time, and most important, the job is in safe hands.
We do what others don't.
Visit our office at 35 Patrice Lumumba Road, Airport Residential Area, Accra.
For more information, call us at 0559 122 092.
Mail us at info@excelsiorgh.com
or visit www.excelsiorgh.com.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, was established in 2014 as one of the World Bank's Africa Centers of Excellence. WACCBIP is located on the campus of the University of Ghana, Legon.
WACCBIP was established as one of the World Bank African Centers of Excellence. And the inspiration behind it is basically that a number of us who were at the Department of Biochemistry and the Noguchi Memorial Institute for Medical Research, we had trained outside, and we did PhDs in various science disciplines outside the country. When we got back and realized that there was not much by way of facilities to train scientists, we saw this as an opportunity to create an environment which would enable many young scientists to get the opportunity to get the quality of training that we got overseas.
This world-class research center started off as an infectious disease center, but along the line, it's added non-communicable diseases to its focus areas.
WACCBIP is providing something to local and regional students that we probably don't have in many places within the country or within the region. Facilities, financial resources, as well as an environment that encourages student learning and research. And that has significantly increased students' output in the Department of Biochemistry, Cell and Molecular Biology.
WACCBIP has a whole range of collaborators at the institutional, national, regional, and international levels. These collaborations include the Noguchi Memorial Institute for Medical Research Ghana, the Department of Biomedical Sciences at the University of Ghana, among other departments, the Ghana Health Service, and the Ministry of Health. The center also has collaborations with institutions within and outside Africa.
Currently, there are about 6 labs in the center, and these labs do research based on the faculty who is in charge of these labs. We have a malaria group that has 3 different units. There's an immunology unit, there's a vaccine development unit, and also a transmission unit in there. We also have a molecular biology lab that has 2 faculty in charge there. One does work with African animal trypanosomes, and the other one, who is myself, works in Brûlé ulcer and antimicrobial resistance. We have a huge virology group. They do research, anything that has to do with viruses. So they do HIV research. They even look at some bat viruses and gastroenteric viruses. More recently, too, we've added a COVID lab, and this lab does routine testing for COVID positivity and negativity. And we also look for seroprevalence across the population. So people who have been prior exposed to COVID, but maybe have been asymptomatic, so they don't know whether they had COVID or not. And we've actually just recently published a paper on our findings.
The virology laboratory of WACCBIP was set up right from the beginning with one main lab, and they were doing things like rotaviral, researching into rotaviral infections in Ghana. The virology lab at WACCBIP was also involved in the first identification of a dengue case in Ghana, and also doing HIV research, as well as research on viruses which are in bats, but which might be have a risk of causing disease in humans. So these are filoviruses. They are the same type of viruses that cause Ebola. So that's work that was being done.
The center currently has 6 research laboratories, namely the Cell Biology and Immunology Lab, commonly known as the Malaria Lab, the Molecular Biology Lab, the Virology Lab, the Medicinal and Natural Products Lab, the Next Generation Sequencing Lab, and the Forensic Biochemistry Lab. At WACCBIP, the scientific tools and equipment are always and readily available to students, regardless of the courses being studied at the center.
Really, the driving force was to try to create a world-class research and training environment where young African scientists from across the continent can come here and develop their talent and do high-quality research that can be comparable to research that's done in any part of the world. So basically, that was the vision that we were pursuing.
Throughout my academic journey, I'd always looked for opportunities to come back home. My plan has always been to establish myself, to spend my most productive, my most useful years in Africa and hopefully in Ghana. So even though I studied abroad altogether for 13 years, followed by 6 years as a postdoc also abroad. So altogether, I was away from Ghana for about 18 years. I was always looking for opportunities to return. So during my second postdoc, I actually met Professor Wandare, the director of WACCBIP, in London when he came to actually receive an award. And that was the first time I heard about WACCBIP. And so hearing about WACCBIP, hearing what Professor Wandare was trying to do in Ghana, I felt that it was a place that would welcome someone like me, a place that someone like me potentially could thrive. So I began to look for opportunities to return, and I was able to get funding to support my return to Ghana, and so applied for a position and was appointed as a research fellow. It was basically a fulfillment of something I'd always wanted to do, and WACCBIP stood out as a place that, even though it was a young center, was led by somebody who himself was very dynamic, who identified a lot with the ideals that I myself identify with. And so was somebody that I thought I could work for, somebody who could mentor me.
WACCBIP has evolved to become a world-class research center, thriving on initial funding from the World Bank and subsequent support from the Wellcome Trust and the Africa Academy of Science.
We were very fortunate to get the funding through World Bank to establish the Center of Excellence here. And part of that funding gave us a huge chunk of money to buy core equipment that could be used and shared by all faculty in the department and opened up to faculty within the university and within the sub-region. We have a core facility for flow cytometry. We also have a core facility for sequencing of DNA. Just recently, we've even upgraded it to include a NextSeq Illumina sequencer, which is probably one of two in the entire Africa. And that has the capacity to sequence both DNA and RNA, which is world-class. We have a confocal microscope core facility, which can be used to take images of cells and for us to understand actual cell biology as it happens in real time. We also have the protein expression lab that I talked about, and we have state-of-the-art equipment in these laboratories that help us, again, make proteins, express proteins, and take it to the next level of trying to identify what these proteins actually do within cells.
Since I've been in the virology department, I've been doing mostly HIV research, that's my specialty, looking at how the different genotypes we have in Ghana affect the ability of treatment and the progression of the disease. And of course, since the pandemic, we have been right at the forefront of doing research of SARS-CoV-2, that's the virus that causes COVID-19 in Ghana. The HIV research that we've done here at Virology, prior to my coming, there's been some findings that in the Ghanaian population, some of the genes that may be involved in progression are deleted in some of the population. Actually, a few of my colleagues did that work. And since I've been here, we've been looking at the new drug which has been introduced in Ghana, dolutegravir, and our data, which is yet to be published, is showing that it is one of the best drugs to be used, and we are also selecting some mutations for this drug.
My experience in WACCBIP here has been very great. I came here in 2014. So after my undergraduate, I joined the lab in 2015 and I served as a research assistant. When I entered, I didn't know anything about research, but I've got opportunity through mentorship and training. I've had great experience here. Sure, I think there's fairness in the enrollment process. For what I know, I was fortunate after my first year master's, I was enrolled into the PhD program. Then what I have seen so far, I realized before you get admission to the program, you write in exams and they try to look at gender balance and also the international and the local students. So there's fairness in the selection process. So my PhD thesis or project seeks to develop novel diagnoses for detecting Plasmodium species. So Plasmodium species are parasite that causes malaria, and currently we have RDTs and microscopy at the point of care for detecting the species. But unfortunately, these tools are not sensitive or specific enough for detecting the species. So there are more reliable tools, which are the PCR or molecular-based diagnostic tools. And if I can give you an example, that's what we are using to detect COVID now. But the unfortunate thing is these tools are not readily available in terms of the reagent. So my project seeks to develop these reagents here in Ghana so that we can make them readily available and also cost-effective for detecting infectious diseases.
The center runs 3 programs at the master's level, which includes Master of Philosophy in Molecular Cell Biology of Cell and Infectious Diseases, Master of Philosophy in Biochemistry, and Master of Philosophy in Molecular Biology. The PhD level includes PhD in Biochemistry and PhD in Molecular and Cell Biology of Infectious Diseases. The center also engages the community in its activities from time to time.
What was of common interest to all of us was that we wanted to create a world-class research and training environment to do basic science. Now, this was led by me, but with support from, several colleagues here at the Department of Biochemistry and also at the Noguchi Memorial Institute for Medical Research. In terms of the resources we need sustainable funding, predictable funding. If you look at all the countries that have developed, they have predictable funding for research and for science. If you look at the USA, they have something called the NIH, which they pump billions and billions of dollars in every year as part of their national budget. It's locked in. Ghana, we still don't have that. We've been talking about a research and innovation bill for a long time. Still not been enacted. We need to have such things so that we as scientists, there's a fund that we can apply to access funds to do research and to innovate on a sustainable basis. As we say it, we compete for funds from overseas. And that is not predictable nor sustainable because a lot of the funders, they keep asking us, how much is your government contributing? Okay, we've given you 8 million, we've given you 6 million, we've given you 5 million. How much is your government contributing?
Everybody knows that Africa has problems. Our problems are all over the news. We get offended when people point out only the problem. But it's also very clear that the only people who will solve Africa's problems are Africans themselves. We are waiting for malaria vaccines to be developed in Europe and North America for us to use. We are waiting for people, for President Jimmy Carter to come and eradicate guinea worm in Ghana. If Africa and in Ghana is going to achieve what I believe and we all believe we can achieve, we have to take responsibility for our problems and come up with locally relevant solutions to solve those problems. For me, WACCBIP represents a place that is trying to do its bit to contribute towards this. We are trying to train the next generation of scientists, not to come and work at WACCBIP. We are training the next generation of scientists to think of ways to solve problems they see in their own community.
International students are helped through WACCBIP to obtain accommodation, and basically what we do is that we liaise with the hostels and the halls on campus and arrange some of the rooms for them before they actually come in. At WACCBIP, we provide at least two forms of scholarship. One from the ACE World Bank grant, and the other from the Delta's program.
Because we are in the tropics, a lot of patients report to the hospitals with high temperature. This is usually attributed to malaria, but we found out that malaria answers, let's say, 40% of those cases. For the other 60%, they are presumptively treated as malaria. And in most cases, the patients keep going back because it's not malaria. So what else is causing it? It's not clearly known. And the diagnostic systems in the hospitals are not advanced enough to take care of a variety of disease conditions. So this project was undertaken by one Nicholas, who looked at a whole panel of pathogens. So this was a discovery project. Okay, so once a child reports with fever and it's not attributed to anything else but infection, and they check for malaria and malaria is not there, they then take those samples and look for all manner of pathogens. Okay, and they were able to discover a whole variety of viruses and bacterial pathogens that explain those other febrile illness. So going forward, that would instruct a new way of attending to patients in that category, so that we can. What we haven't done is to develop diagnostics that can be used in the hospitals, but that is the next phase that we can look at. The second is diagnostic technologies that have been developed by two PhD students, Francis Krampa and Felix Ansa. So here they are looking for electronic ways of detecting malaria. Okay, so what we have in the system right now is glucometer. You put a drop of blood and it tells you the level of glucose in your blood. We are trying to do the same for malaria so that people can even test at home, right? And not have to go to the hospital when this becomes available on the market. The third one is a project that concerns a new way of developing antibiotics, And what we are trying to do is to target what we call the genome stability of pathogens. This has not been done before, and we are pioneering that from this center for the first time. And we're hoping that over the years, we will make progress that will lead to the next generation of antibiotics.
The first output that we had from our sequencing was a manuscript, which was published, and that was the first 46 sequences of SARS-CoV-2 in Ghana. We published that last year, and since then we've done almost 1,000 sequences. So we are preparing that also to publish it. And why that is important is we are tracking basically the changes in the virus locally, because there's if you look at literature or the news, you see how it's changing globally, but you have to know the local outbreak, you have to know the local epidemic. And what we see in Ghana is that sometimes the variants which are increasing are not necessarily what is increasing in the rest of the world. For now, for instance, we have Delta, which is also on the rise in Ghana. But there's another variant which is nobody's talking about, but it's rising almost faster than Delta in Ghana. So that's the sort of things that we do. And when we get the results, we write what we call policy briefs, advising the government that this is what we found. Earlier on in this year, when we did our sequencing of airport samples, and we first found the Alpha variant, that's the UK variant. And then we found it in local circulation, and we wrote it, we communicated to the government, and that was what led to the reinstatement of some of the restrictions, like restricting the funeral size, party size, and things like that. That was as a direct result of the work that we're doing.
The key message that we want to put out there is that Africa has a lot of talent. If you look across the world, there are African scientists doing great things all over the place. Some of these COVID vaccines that we are seeing, African scientists played key roles in developing them so quickly in different countries. If we create the right environment for them, a lot of these people will come back home. We have already brought 7 scientists, African scientists who were in the diaspora. We brought them here. And the reason why they were able to come back, or they were convinced to come back, is because they came and looked around, like you guys have looked around, and they saw that they can do good science here, and that this environment looks like where they've been working in the UK or in the US, and they are convinced that this is world-class enough. If we create that, we can start to bring all our talent back, and Africa can start to develop our own vaccines, can develop our own drugs, we can be more innovative and build new technologies that we can even export to other parts of the world, and not sit as we are now waiting for people to develop vaccines, use for their people. When they have had enough, then they share with us. That's where we are now. Okay? That situation has to change. That means we have to take matters into our own hands. That means the African governments have to start to put some money into science on a consistent basis. Let's have a fund, such as we have National Health Insurance Fund, we have GET Fund, we have all these funds that a percentage of the taxes goes directly into that fund every year. So that fund is available for scientists to access. Let's do that. Let's put some money aside for science, because this type of investment have to be done for us to develop. Otherwise, we keep marking time. We keep just treading water. Because if you keep relying on people for your innovations, that means you are not making progress. I keep telling people that if you think about investment in science, think about it like the way we invested in petroleum exploration. You know, ever since I was a child, I was hearing about GMPC spending billions and billions of dollars doing oil exploration. Okay? They didn't find oil until just maybe 10 years ago or less. After spending billions and billions of dollars. Nobody complained that they were spending billions of dollars because they knew that when they found oil, they'll get a lot of money from it. It's the same with science. Don't think about what you'll get tomorrow. You know, don't say that, oh, I've given you 200,000, so what have you brought after 2 weeks? No, it doesn't work that way. It has to be consistent investment over time. And then when you succeed, it will pay for itself. Okay. If we made a vaccine right now, can you imagine if we had a COVID vaccine and we're supplying to the whole of Africa? Can you imagine how much money Ghana will be making? But if you don't invest, you won't get. You won't get there. So you have to invest, and then eventually you will hit the jackpot.
If there is any biomedical research center that promotes innovation and research, or that provides its students with unmatched financial resources and an environment for learning and research, it is the West Africa Center for Cell Biology of Infectious Pathogens, WACCBIP, providing world-class postgraduate training and research into tropical diseases in sub-Saharan.
In the lab, you face consistent pressure to do more with less while trying to keep up with a growing workload. Success means having instruments that you can rely on to overcome diverse challenges. The Agilent 6470B Triple Quadrupole LCMS, the latest version of our trusted workhorse, gives you the robustness and versatility you need to overcome these challenges. The 6470B has a proven reputation for robustness with non-stop operation, backed up by thousands of users, with numerous application notes and peer-reviewed articles to reduce downtime and increase efficiency. Enhance versatility with a wide array of ion sources to analyze any application. Ion source options include ESI, the standard choice to broadly ionize analytes of interest, AJS, which enhances the desolvation power of standard ESI for ultra-high sensitivity, APCI, for increased signal-to-noise for very small analytes, and MMI, to carry out ESI and APCI simultaneously for a complementary analysis. With the new VacShield, you can reduce maintenance time by 92%. VacShield lets you remove the capillary and run the cleaning cycle in approximately 30 minutes, compared to about 6 hours for maintenance by venting and disassembly. So you can gain back valuable lab time and keep up with high demands. From analyzing environmental contaminants and ensuring food safety, to complex life science research, the 6470B meets your need for reliable, accurate results. These accurate results are provided by our Mass Hunter software, including Quant My Way, which enables you to personalize the software with tools that highlight only the capabilities of interest. With comprehensive end-to-end workflow solutions and application services, you can ensure proper sample preparation, expertly developed acquisition and data analysis protocols, and fully customizable reports to get the answers you need as soon as possible. Our solutions are designed to work together to provide efficiency gains that help you get more done and reduce operation costs. The Agilent 6470B Triple Quadrupole LC-MS is unbelievably robust to increase your lab efficiency and remarkably versatile to keep up with your evolving demands. Visit agilent.com/chem/6470B for more information. Decades of innovation have brought us into a new era of mass spectrometry with the design of Altivo, Agilent's revolutionary triple quadrupole LC-MS. At just 1/3 the size of our previous triple quad LC-MS instruments, Altivo integrates seamlessly into the stack of the Agilent Infinity Lab LC System series, saving bench space while offering the same power and accuracy of our larger systems. The footprint of Altivo is 70% smaller than that of similar LC triple quadrupole mass spectrometers, so you can triple your lab's capacity in the same space. The efficiency-optimized Agilent Infinity Lab LC Series system can sit directly on top of Altivo, making it the perfect partner for high-throughput mass spectrometry. The innovations that enabled Altivo were specifically designed to maximize quantitative performance, enhance instrument reliability and robustness, and promote greater uptime. The more ions you get into the instrument, the better your MS results. With Agilent's Jetstream ion source, a superheated nitrogen sheath gas surrounds the electrospray to quickly and efficiently desolvate the spray plume and tightly cluster the resulting ion cloud, maximizing the number of ions that are created and transported into the LC-MS. VacShield features fast ion injector exchange without the need for system venting and improved diagnostics that guide the operator to appropriate corrective action, leading to less downtime for maintenance and higher throughput for your laboratory. Once the ions enter Altivo, they pass through our innovative precision-manufactured Cyclone Ion Guide. With its twisted and tapered dual hexapole configuration, the Cyclone Ion Guide provides maximum ion capture, compression, and transmission across a large pressure drop region, giving you better, more reproducible results. From the Cyclone Ion Guide, the ions continue to the first quadrupole. Better mass transmission improves analytical results. Each quadrupole assembly includes DC-only, wafer-thin pre- and post-filters, which cancel fringe field regions at the quadrupole entrance and exit to improve ion transmission. This allows for faster switching, enabling more MRMs to be performed. With the analyte selected in Q1, the ions pass through the vortex collision cell. Its innovative twisted and tapered dodecapole hexapole geometry maximizes ion collection, fragmentation, and transmission. The high-efficiency detector offers a wide dynamic range to ensure linear responses for your most challenging analyses. The detector was designed to be easily replaced by anyone in the lab. Working together, the compression and focusing of the Cyclone Ion Guide, the improved ion transmission through Q1 with its pre- and post-filters, the optimized fragmentation in the vortex collision cell, the improved transmission through Q2, and precise data capture of our innovative detector technology give you better MS results. Altivo uses an entirely redesigned electronics architecture, making it the smartest LC-MS on the market. New intelligent diagnostics tools enable Altivo to monitor several parameters in real time to ensure that it's operating at peak performance. If it isn't, it lets you know so that you can take appropriate action. For food safety labs, Ultivo offers the selectivity and sensitivity needed to confidently monitor for and identify contaminants, even in difficult matrices, helping keep the food supply safe. For environmental analysis, Ultivo provides robust and accurate measurements down to sub-part per billion levels and integrates with Mass Hunter software for powerful data collection, processing, and reporting to help you keep up with newly emerging pollutants. Reshape expectations, reinvent capabilities, and redefine what's possible when small meets powerful. The Agilent Ultivo Triple Quadrupole Mass Spectrometer.
There is no promise we cannot keep. There is no commitment we cannot honor. There is no delivery we cannot guarantee. There is only one Bethel Logistics Company Limited, a licensed international freight forwarding and clearing agent, a trusted global brand with an extensive network for the movement of air freight and ocean freight worldwide. We are the trusted name for road freight into Western and Northern Africa. We are fully accredited by Ghana Customs and Excise. We cater for all your freight forwarding, customs clearing and forwarding, cross trades, inland transportation, internal cargo tracking systems, customer freight costing and consultancy, cold chain logistics management, logistics and warehousing, quality inspection, and much more. In Accra, Tema, and Takrady, we are always available to ensure easy movement of cargo countrywide. Call us on 0204 528 900 or 0244 707 859. Email info@bethellogistics.com. Locate us in Accra at North Ridge, adjacent Accra Girls High School, or in Tema at Community 4, or in Takrady on the second floor of the GCB building. Website bethellogistics.com. Bethelog, experts in shipping logistics.
Welcome to Biotech, Africa's genomics company. Established in 2002 in Pretoria, South Africa, Biotech is a rare and unique company, as suggested by the name, and we thrive to give reliable and efficient services to all our customers. How it all began. With the concept developed in the year 2000 to speed up the arrival of primers in molecular labs in the country, Dr. Oliver Pricer, co-founder and executive director, together with a venture team of 5, secured a loan from Swiss government and got rights to be the very first company to synthesize primers locally, and thus it all began. With capacity to synthesize over 1,000 primers a day, Ingaba remains the only company that does this in Africa. This is made possible by the Mermaid 192E. Using an ABI 3500 exome, we offer DNA sequencing from various templates, and we offer primer design, PCR amplification, cloning of PCR products, fungal and bacterial identification, and much more. In 2007, we launched Next Generation Sequencing Services, which provide NGS projects design inputs, quality control library preparation, and sequencing workflow using either the MySeq, NextSeq, or the PacBio systems. We have more services that include SNP genotyping and methylation analysis using the Agena Biosciences mass array systems, bioinformatics solutions that complement our NGS services, and qPCR analysis. We also are a distributor of strong international brands, which includes New England Biolabs, Zymo Research, Eppendorf, Glentham, CG, Nolato Treff, Charlab, just to name a few. Inqaba boasts a responsive technical team with expert knowledge in the services we provide. We also have a well-trained engineering staff making sure that the installation and servicing of instruments purchased at Inqaba is well taken care of. A fleet of vehicles commissioned for local trips and deliveries assures we arrive at your lab on time. In the past 18 years, we have started subsidiaries in East and West Africa, and we have offices in Kenya, Nigeria, Ghana, Tanzania, Senegal, and still maintain a strong presence in our neighboring countries. All this is achieved with a highly trained and professional team that is always ready to assist. Welcome to Inqaba Biotech.
If you need a reliable procurement and supply chain partner, look no further.
Global procurement company Excelsior Global Limited is here for you.
We work across multiple industries and sectors with one goal in mind, ensuring that our clients focus on core businesses while outsourcing procurement.
Excelsior Global takes care of purchasing process while reducing all procurement and supply chain costs.
We offer a wide variety of procurement solutions connected mainly with medical, COVID-19 antigen test kits, COVID-19 antibody test kits, COVID-19 antigen test kits, virus transport media, specimen collection swab, sample preservation tubes, HIV test strips, hepatitis C test strips, hepatitis B test kits, syphilis test strips, glaucometer test strips, pregnancy test kits, hepatitis C, HCV, examination gloves, syringe, surgical gloves.
Excelsior Global makes procurement easy, simple, on time, and most important, the job is in safe hands.
We do what others don't.
Visit our office at 35 Patrice Lumumba Road, Airport Residential Area, Accra.
For more information, call us at 0559 122 092.
Mail us at info@excelsiorgh.com
or visit www.excelsiorgh.com.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, was established in 2014 as one of the World Bank's Africa Centers of Excellence. WACCBIP is located on the campus of the University of Ghana, Legon.
WACCBIP was established as one of the World Bank African Centers of Excellence. And the inspiration behind it is basically that a number of us who were at the Department of Biochemistry and the Noguchi Memorial Institute for Medical Research, we had trained outside, and we did PhDs in various science disciplines outside the country. When we got back and realized that there was not much by way of facilities to train scientists, we saw this as an opportunity to create an environment which would enable many young scientists to get the opportunity to get the quality of training that we got overseas.
This world-class research center started off as an infectious disease center, but along the line, it's added non-communicable diseases to its focus areas.
WACCBIP is providing something to local and regional students that we probably don't have in many places within the country or within the region. Facilities, financial resources, as well as an environment that encourages student learning and research. And that has significantly increased students' output in the Department of Biochemistry, Cell and Molecular Biology.
WACCBIP has a whole range of collaborators at the institutional, national, regional, and international levels. These collaborations include the Noguchi Memorial Institute for Medical Research, Ghana, the Department of Biomedical Sciences at the University of Ghana, among other departments, the Ghana Health Service, and the Ministry of Health. The center also has collaborations with institutions within and outside Africa.
Currently, there are about 6 labs in the center, and these labs do research based on the faculty who is in charge of these labs. We have a malaria group that has 3 different units. There's an immunology unit, there's a vaccine development unit, and also a transmission unit in there. We also have a molecular biology lab that has 2 faculty in charge there. One does work with African animal trypanosomes, and the other one, who is myself, works in Buruli ulcer and antimicrobial resistance. We have a huge virology group. They do research, anything that has to do with viruses. So they do HIV research. They even look at some bat viruses and gastroenteric viruses. More recently, too, we've added a COVID lab, and this lab does routine testing for COVID positivity and negativity. And we also look for seroprevalence across the population. So people who have been prior exposed to COVID, but maybe have been asymptomatic, so they don't know whether they had COVID or not. And we've actually just recently published a paper on our findings.
The virology laboratory of WACCBIP was set up right from the beginning with one main lab, and they were doing things like rotaviral, researching into rotaviral infections in Ghana. The virology lab at WACCBIP was also involved in the first identification of a dengue case in Ghana, and also doing HIV research, as well as research on viruses which are in bats, but which might be have a risk of causing disease in humans. So these are filoviruses. They are the same type of viruses that cause Ebola. So that's work that was being done.
The center currently has 6 research laboratories, namely the Cell Biology and Immunology Lab, commonly known as the Malaria Lab, the Molecular Biology Lab, the Virology Lab, the Medicinal and Natural Products Lab, the Next Generation Sequencing Lab, and the Forensic Biochemistry Lab. At WACCBIP, the scientific tools and equipment are always and readily available to students, regardless of the courses being studied at the center.
Really, the driving force was to try to create a world-class research and training environment where young African scientists from across the continent can come here and develop their talent and do high-quality research that can be comparable to research that's done in any part of the world.
Good afternoon.
So basically that was the vision.
2 minutes past 3. So please, those outside, please, let's give ourselves a minute to settle, and we can start.
All right. Good afternoon. So this is still a continuation of session 1, the SARS-CoV-2 pathogen genomics and host responses, part 2. We had to break it up for the panel discussion because of our panelist schedules. And the moderator for this session, the 2 session chairs for this session are Dr. Emmanuel Tego and Dr. Daniel Odro-Mensah. So I'll hand it over to them for them to call the next speaker.
For our first talk, we have Kesiho Tapela. She will talk about the identification of cellular population associated with either symptomatic or asymptomatic COVID-19 disease in Ghana. Ms. Tapela, you have 12 minutes.
Okay, good afternoon. The topic of my talk today is about identification of cellular population associated with either symptomatic or asymptomatic disease in Ghanaian COVID-19 patients. My name is Kesiho Tapela. So, as other speakers already said, we still have cases of COVID-19 worldwide, including Ghana. And as of yesterday, we have had around 567 million cases worldwide. And here in Ghana, where the study was based on, we have around 167,000 cases. And of those, we have lost about 1.45 thousand people. So as of yesterday, we had around 669 cases, which marks our 5th wave. So with that, we still have new cases, and the research on COVID-19 still continues. So COVID-19 is caused by a virus known as SARS-CoV-2. So the virus has 4 most important structural proteins: the spike, the nucleocapsid, the membrane, and the envelope. So those are the 4 important structural proteins that have helped this virus on its pathogenesis. So like any other pathogen, when this virus enters a host, the body elicits an immune response. We have the innate immune response, which is the first immune response. Then after that, we have the adaptive immune response, where we generate T and B cells. Okay. Okay, so just a little bit on the immune response. So upon infection with this virus, the virus enters either through oral and respiratory cells. The dendritic cells, which are the antigen-presenting cells, will then engulf the virus, and then with that, it stimulates the adaptive system. So it stimulates the CD4 cells, which then differentiate into the cytotic cells in order to help neutralize the virus or kill the virus. This also helps stimulation of the B cells, which further produce some antibodies to help and neutralize the virus. All in all, the body is trying all its best in order to neutralize or kill the virus. Now, so the immune response depends for an individual. So looking at the cases here in Ghana, we have a lot of asymptomatic cases in Ghana, and we have had a lot of symptomatic cases elsewhere. Now the question is the immune response different or similar between those 2 populations? Hence this study. So this study was trying to identify those cellular population associated with either symptomatic or symptomatic, since we have most of asymptomatic cases in Africa, especially here in Ghana. So the study sites, samples were collected in big hospitals here in Ghana. We have the Cape Coast University Hospital. We also have some archive sample here in WACCBIP that were collected during the first outbreak. Some samples were also collected here in WACCBIP. We also collected some samples at Ridge Hospital. So the total sample size was around 104, of which 61 were symptomatic and 43 were asymptomatic. So from those, so these are like two types of study. It's a cross-sectional and longitudinal. So from those 61 in the symptomatic, we followed up 35 of those, and we're sampling them every week for 4 weeks, like for a month. Same applies to asymptomatic. We had 29 patients that we were following up for 4 weeks. For sample processing, we're using PBMC. So we'll collect blood, then we isolate PBMC, then we acquire through flow cytometry. This is just a brief about flow cytometry. So we are storing our samples in either liquid nitrogen or -80. Then we did some surface staining, which involves some washing, some incubation. Then we acquire it through flow cytometry. So these are some of the markers that we're targeting. We're generally targeting the T cells. We're also targeting the B cells, some monocytes, and activation and exhaustion markers to try to find if our cells are being activated or exhaustion during the disease progression. So this is my cell gating. Upon acquiring my samples in the flow cytometries, I was using FlowJo to analyze my sample. So this is my gating where I get my T cells. Then I check for activation and exhaustion markers. And so the B cells, the monocytes where I had markers for dendritic and other monocytes. So this the general first result that we got. So I just wanted to compare how the immune has reduced, looking at a healthy person. This the frequency or the range of someone who is considered to be healthy. So from a COVID-19 patient, it has reduced, for example, for monocytes, it reduced to 1.2%, and we expected like 10 to 30 for a healthy individual. Same applies to lymphocytes. So this was just to show you how the reduction has occurred. And also we tried to see the difference between CD4 and CD8. Remember, the CD4 has to be twice higher as CD8. So we wanted to see if there's much difference or the CD4 were really higher than CD8. And with that, we wanted to see the ratio between CD4 and CD8. So a normal ratio of CD4, CD8 has to be between 1 and 3. So below 1, it shows like a severe infection, and it's between 1 and 3, then it shows like that is normal, even though the person is infected. So we found that there was no much significant difference between asymptomatic and symptomatic. However, the CD4, CD8 ratio was normal for both symptomatic and asymptomatic, which might be explaining why we don't have such severe cases here. But however, in other places, there were some cases of low CD4, CD8 ratio, which was then associated with the critical condition of COVID-19 patients. Just going deep into the CD4. So we tried to compare the CD4 levels between symptomatic and asymptomatic. We couldn't find any significant difference between the two. But however, when we tried to look at the activation and exhaustion markers, we found that the CD4 are being activated and exhausted more in symptomatic as compared to asymptomatic. This shows that someone who's symptomatic, his cells are being overworked or they're working more than asymptomatic. But for CD8, it was higher in asymptomatic as compared to symptomatic. And however, the activation was also still higher in symptomatic as compared to asymptomatic. But however, we can explain this in different ways. Remember, this study was done in peripheral blood. So when the body releases those cells, sometimes the cell will move across the body into tissues in the lymph nodes, try to fight this virus. So since our study was basically in peripheral blood, it's possible that some of the cells have moved to other parts of the body. Then we did try to see the B cells, how different between symptomatic and asymptomatic. So for here, we found that the B cells were higher in asymptomatic as compared to symptomatic. Since was this contradicting in some study, we tried to look into literature and see if there was any similar study that had the same information. Then we found that this study by, I think it was in China, they also found this the same, that there were higher B cells in asymptomatic as compared to symptomatic. This can also be explained that maybe some of the B cells have moved to other parts of the body, since we expect antibodies to be higher in symptomatic as compared to asymptomatics, as other studies has found. So we also have some...
you have 2 minutes, guys.
Okay, so we also have some classical monocytes. So classical and non-classical monocytes, so we're trying to see if they're higher in symptomatic as compared to symptomatic. So basically, the classical monocytes are the ones that are mostly involved in pro-inflammatory cytokines. So they were higher in symptomatic as compared to asymptomatic, which may be obvious because most of the symptomatic patients have higher cytokine levels. So we also tried to compare this, and it was still similar to other studies. And also we tried to look at the dendritic cells. So the dendritic cells are more like the antigen-presenting cells. So we are also trying to see if there was any difference between asymptomatic and symptomatic. So the myeloid dendritic cells are the ones that are important for the antigen-presenting cells, and they form like 80% of all the dendritic cells. So we found them to be higher in asymptomatic and symptomatic. This was also similar to some other studies. So, and also just remember my study was longitudinal. We're trying to see the trends of how the T cells, the B cells were changing over time. So there was really no clear trend of how it's going, but we can still see a change or a reduction in cell frequencies as the disease progresses. Same for both B cells and T cells. And lastly, I'd like to conclude that our COVID-19 patients have increased cell activation exhaustion in symptomatic as compared to asymptomatic. And so there were higher B cells frequencies and activation in asymptomatic compared to symptomatic. And also the classical monocytes were higher in symptomatic and asymptomatic. So we can further suggest that the immunity in symptomatic is being activated than asymptomatic, and the immune response in asymptomatic is more like it's not really as active as in symptomatic. And also we can also say that since our study was in the peripheral cells, peripheral blood, some of our cells have moved to other parts of the body. And finally, I would like to thank my supervisors, Dr. Kwashi, Dr. Tego, Prof. Ki, Dr. Bediako, and my participants, Ms. Diana for helping with this flow cytometry, Kwashi Lab, my funders, WACCBIP, Juanita, Ken, and Bill and Gates. Thank you.
Thank you, Kes. We'll take the next talk. So please keep your questions for at the very end. So the next talk is from Seth Asiedu. He's online. Okay. All right. So Seth will be talking about in silico identification of potential anti-inflammatory compounds targeting the COVID-19 induced cytokine storm. Seth.
Please, am I audible? Please, am I audible?
Yes, Seth, we can hear you.
Okay, sure. Thank you very much. So my name is Seth. So my name is Seth Osei-Siedu, and I will be presenting on the in silico identification of potential anti-inflammatory compounds targeting COVID-19 induced cytokine storm. So COVID-19 is caused by the acute respiratory syndrome coronavirus, that's the SARS-CoV-2, and it is a positive sense single-stranded RNA virus. As of today, there have been 6.38 million people have died from this disease. In critical stages of the disease, acute respiratory distress syndrome, pneumonia, septic shock, arrhythmia. Arrhythmia is actually abnormality of the heartbeat rhythm and disseminated intravascular coagulation. That's a condition which actually affects blood clotting, has been observed in complicated cases. And studies have reported aggressive inflammatory response with the release of very large amounts of pro-inflammatory cytokines in this critically ill COVID-19 patients. And this condition is being termed as cytokine storm. So the P38 mitogen activated protein kinase has actually been implicated in the disease in cancers and other viral infections. And its mechanism is quite simple. I'll try and simplify it. So upon cell entry, SARS-CoV-2 binds and downregulates the ACE2. So the loss of ACE2 activity upon viral entry allows the uncontrolled inflammation by a mechanism, notably the P38 MAP kinase pathway. So hence, this kind of propagates the SARS-CoV-2 life cycle. Moreover, SARS-CoV, a very close neighbor of the SARS-CoV-2 virus, expresses a protein that directly upregulates P38 MAP kinase in vitro. And this process seems to be a pathogenic step in the life cycle of many RNA respiratory viruses. So a study on the effect of P38 MAP kinase inhibitors on SARS-CoV showed an 80% increase in survival after treatment. So that's why the P38 MAP kinase has been suggested as a plausible therapeutic target for COVID-19. So on our objectives, we seek to use computational techniques to screen a library of natural products from Africa and China against the P38 MAP kinase structure. And why computational techniques? Because it's faster and it's relatively cheaper. And natural products because they are structurally and chemically diverse. And we also sought to predict the pharmacological profiles of the identified compound. That's their human intestinal absorption, permeability, glycoprotein binding, cytochrome P40 inhibition, and most importantly, toxicity, which leads to gas adhesion. And we also use machine learning technology to predict the biological activities of the compound. And finally, we also aim at identifying compounds which can inhibit the activities of the P38 MAP kinase. So with our methodology, first we retrieve the structure of the protein from the protein database and we clean it up. And we also use a large database of compounds from the African Natural Product Database, the traditional Chinese medicinal database, and the North African Natural Product Database. So this was a compendium of about 38,000 compounds. And we also circulated and had around 11 inhibitors which were used to and we validated our docking protocol. Okay, sure. And we validated our docking protocol. Is am I audible?
Sir, can you hear me?
I can hear. Please, am I audible?
You can hear me. Please put it on slideshow.
Okay, sure. Sorry.
Right.
Okay, sure. Thank you very much. And we validated our docking protocol. Then afterwards, we screened our compounds. That's over 38,000 compounds against the receptor that we've identified. We did our pharmacological profiling, which I've explained earlier on. We looked at the protein-ligand interactions, and we did a machine learning-based prediction of activity. and we did molecular dynamics and MMPDSA computations. So with the results and discussion, and the figure as shown is the surface representation of our protein structure, and it was taken from the PD, the protein database. It has an ID of 3ZS5, and it was solved using X-ray diffraction at a resolution of 1.6 angstrom. Now this structure has 362 amino acid residues, and the binding pocket, which is colored green, is actually located in the catalytic HB binding pocket between the C and N terminal. And we computed the volume and the surface area of our structure. So we further went on to look at the molecular docking, which is a row screening. So before we did that, we had to validate our docking protocol. And what I mean by validating is our tool that we are using, would it be able to discriminate between active compounds and decoy compounds? And decoy compounds being compounds with similar physicochemical properties as the active. Now, after validating it, we realized that orthodox brainer has an area under the curve of the ROT test of 0.701. So this is suggesting that it can reasonably distinguish between inhibitors and non-inhibitors of the p38 MAP kinase. And moreover, a previous study has also used orthodox brainer to successfully screen compounds against the p38. And the compounds which were found from the docking were the experimental studies corroborated the potential inhibitory activities of the compound. So after we validated our docking results, then we came to the real screening. So we realized that we screened over, as I stated earlier, on over 30,000 compounds. And after screening the compounds, we had a total of 42 compounds which had a binding activity of binding energy of lesser than minus 10.0 kilocalories per mole. And this threshold has already been defined in literature as a threshold for determining potential inhibitors of the p38 MAP kinase. So the ones zinc are all annotations of the zinc database. So zinc database was where we actually got the compounds from. So these are the annotations. And the SP202 we use 11 known inhibitors. So these are one and two of them which were further used as controls in our study. So we looked at the protein ligand interaction. And hydrogen and hydrophobic bonding are critical in the stabilization of ligand within the pocket of a receptor. So all our 42 compounds were observed to bind deep inside the binding pocket. So for visualization sake and for presentation sake, I'll be presenting on one. That's zinc 1691180. And this one forms 10 hydrophobic contacts and one hydrogen bond. This is a hydrogen bond with tyrosine 35. And we looked at our residues and also tried to understand the roles of the various residues. So we looked at them and realized that in literature, actually, lysine 53 and phenylalanine 169 has been identified as critical for the stabilization of a ligand inside the p38 MAP kinase from crystallographic studies. Then after the protein ligand interaction, we looked at predicting the activity of the compound using computational and machine learning technology. So, for instance, some of the compounds were predicted as platelet aggregation inhibitors, some were predicted as antiviral, some were predicted as anti-inflammatory compounds, with high anti-inflammatory activity. And one thing we found from our studies was that randomly, out of the 38 compounds, which were shown to have very good affinity and all this stuff, one of our compounds, that is zinc 5519433, is already a known kinase inhibitor. So this actually gives a bit of weight to whatever we were doing with our study. Then we further went on to look at the molecular dynamic simulation, and we evaluated a lot of things. So we looked at the RMSD, which is used to assess the stability of the protein structure. And when the RMSD curve tends to converge, it is indicative that the structure is stable and the system is well equilibrated.
Seth,
and we also
you have 2 minutes if you can hear me.
Yes, I can hear you. And we also looked at the hydrogen bonding. Then we took snapshots of our simulations over the simulations and realized that it was fairly stable and it didn't shift that much. Then we did MMPDAT analysis which is used to address some of the limitations of the current storage functions. We looked at the contribution, the energy contribution of the various residues. So overall, we came up with 4 compounds, which could be further experimental studies could be done on it. So to conclude, the study sought to identify C38 MAP kinase inhibitors, and we use a lot of computational techniques to come up with it. And the identified compounds could be used to attenuate the effect of COVID-19 induced cytokine storm. So this study has been published in Biomolecules NBTI, and I thank you very much for your time and attention. Thank you very much.
Don't go away, stay on. You finish this session, then we have the questions. All right, the next presenter is Francis Jabin. Francis Jabin. All right. He's presenting on classification, predictive modeling of SARS-CoV-2 seropositivity based on self-reported clinical symptoms. Thank you.
Yeah. Thank you. Are you able to? Great. So thank you. I'm Francis Jabin. So I work with WACCBIP. I'm the data manager, and I've been working on the seroprevalence projects based on COVID-19 for the past 2 years. I've been in the team, a dynamic team that's very diverse background, and they've been at it for this while. Okay, so the key question we are seeking to answer, or the key questions we are seeking to answer, are what is already known about these subjects. It's already known about the subject, what's done already, and what was found in our study, in this specific study, and what's the future direction for COVID-19 with regards to our study. Okay, so the main challenge has been that clinicians have difficulty trying to diagnose patients at the heat of pandemic, where we have COVID-19 and other related diseases prevailing. And so identification and isolating patients with COVID-19 were based solely on symptoms, and the recommendation at the time was, or even as at now, is that once you experience clinical symptoms such as headache, sore throat, and others, you got to isolate yourself. But unfortunately for us, we are confronted with a number of viral infections as well, plus malaria, which have similar infections just as COVID. And so that's the challenge we have to battle with. So we sought to evaluate the symptoms decision-making approach in isolating and trying to see how these would impact the classification of people being either positive or testing positive or negative to COVID. And our approach is basically using machine learning algorithms, which we would use the data and then subject it to the algorithms and see what it can do for us. Okay, so our approach, again, we've been doing population-based study with 5 partners in 3 countries, Nigeria, Ghana, and Burkina Faso. In Ghana, the average seroprevalence has been between 20 to 60 thereabouts, and the positivity rate in Burkina was around 32, and that of Nigeria about 56. Yeah, so just a bit on ML so we can appreciate the subsequent results. Okay, good. So basically, we sought to define this challenge or problem as a machine learning algorithm problem. And now COVID is either you test positive to COVID or you test negative. And so it's in this paradigm, it's referred to as supervised learning. And because it's supervised learning, and we sought to classify these groups, we use an approach under supervised learning called classification. And there are clearly algorithms that are clearly available for us to utilize. So we would basically rely on these classification algorithms. Okay, so these particular ones. And then proceed further to do ensemble learning. And essentially, we're saying that we want to put all algorithms together and see to what extent can these classifiers predict or classify COVID positivity. So this our basic workflow. This study that we're doing, we're utilizing data from June 2021 to May 2022. And we've excluded antigen components of it for obvious reason that we do not have enough data to train our models. So we relied heavily on antibody components of the study. And all the data from the 3 countries were utilized in our study. We proceed to subject that to further cleaning, extract features, and features here, basically the variables of interest. That's the symptoms in this case, and run it through the algorithms, and then assess the performance of these algorithms. And the metrics basically we used were accuracy, sensitivity and specificity, and ROC, AUC. And AUC basically because if you randomly choose a positive and a negative case, then your classifier should be able to assign a higher probability to a positive case. And it's very useful even when you have an imbalanced class in your learning process. Okay, so our basic analysis we sought to do, all we did earlier was to look at the symptoms, its association with antibody. We found that cough, headache, and loss of smell and taste were significantly associated with COVID positivity. And so we saw that of sore throats and running nose. Well, the others may be clinically associated with the disease or COVID, but these were found to be statistically significant. Same when we attempted to do multivariate analysis, we still saw the results statistically significant, even after we controlled for other explanatory variables like age and others, and even for comorbid variables in the database. Okay, so this is the slide that tells us how our algorithms performed. And various algorithms were assessed. Logistic regression, which we saw earlier, but in this case was also part of the machine learning algorithm. K-nearest neighbors, classification, which is a regression tree, naive, Bayes, and SVM were assessed. And we noticed that logistic regression still could classify the positivity for us at just above 58. We found key nearest neighbors below 56% of classifying the positivity. And so when we proceeded to do further analysis, having used an assembled approach, we noticed that AdaBoost algorithm performs much lower than gradient boosting approach, random forest and extra trees. And when we assessed the ROC curve, we noticed that our classifier could tell us about 60%, or assign a higher probability of about 60.60 to a positive case. And on average, we found our precision to be 56%, and that's, 58. And then our recall, which is the same as specificity. Yeah. So, and an average of these two can give you 61%. And that's what we call the F1 score. Okay, in summary, our data shows that approximately 60% of COVID positivity could be classified using machine learning algorithms. And our work indeed indicated some future in informing decision making in trying to isolate COVID cases. And this further reinforces the message of still testing before treatment. So in the near future, our team is looking at trying to improve these algorithms or performance of these algorithms using IgM, which tells us the current infection, and if possible, PCR, and also add malaria positivity and other viral infection data. And so we can also see to what extent these can be classified and proceed further to develop an AI system, if possible, such that with these symptoms and other key variables, we can automate the process of isolating these cases or clinical cases in our hospitals. Again, of interest to us is also to model household level transmissibility. And the reason is that these are symptoms, and so once you cough, for instance, you could infect your next neighbor in the household. And that's we failed to do in this study. We'll look at it in the near future. We're grateful to our partners, our funders, and then our study participants, staff, and that's WACCBIP staff, COVID team in particular. Thank you.
Thank you, Francis, Seth, and Kesego. We have 9 minutes to interact with our presenters. All right, so the floor is open for your question. Right.
Hey, thanks so much for the presentation, all very wonderful. I have a question to the first presenter. Okay. Thanks so much for the interesting talk. Now, I was wondering two questions actually. The first thing is, have you looked at any more specific T-cell response to SARS-2? Have you found any T-cells that would particularly attack SARS-2? There was like indication of this UK healthcare workers that some previous coronavirus infections would actually cause kind of specific T-cell response. That is question A. And question B is, with the T-cell and B-cell going down over time, where you saw you didn't see a lot of differences. Have you really tried and do the patient-by-patient plot of how it goes down? So, or let me rephrase, have you tried really to do a two-way ANOVA to test for that?
Okay, so the first question is antigen specificity, whether we try that.
T-cell.
T-cell, yeah. Okay, yeah. No, yes. So that's what we are currently working on right now. So the data is not ready yet to share. The second question.
When you saw this time series with the T-cell levels going down and the B-cell levels going down, you saw there was not much difference between the consecutive days. But I was wondering, have you looked at it really patient by patient? And have you really done a two-way ANOVA to check for that?
Okay, so we didn't do patient by patient. So we just took the median of all the patients. So between days, it wasn't really significant. That's why I was saying the trend was not really clear enough to define it.
Okay, but you have it is you have the measurements per patient, right?
Yeah, I don't have. Maybe I don't hear you. Please repeat your question.
Yeah, if you have the measurements per patient, so you have all the different days per patient, yeah, then you got to do a two-way ANOVA to check for differences.
Yes, we did two-way ANOVA to check for the differences.
Okay.
Yeah. No.
Hello. Yeah, my question is to the second presenter and the third presenter. So I think I'll go with the second first. Seth, you mentioned machine learning techniques you used in your work. Would you mind sharing the types you used, or as in what particular techniques, machine learning techniques you use?
Hello. So did you get a question?
So we use PASS, and PASS is a tool built on Bayesian-based machine learning technology. So that's what we use. So it was PASS that we used to do the prediction that is built based on Bayesian.
Okay, thank you. And to Francis, I want to find out if you had any issue of data imbalance with respect to your machine learning training process, and as to whether the performance was due to that or maybe some data challenges, as to what could be done to improve the performance of the models presented. Thank you.
Yeah, thank you. Good question. So, yes, indeed, we did assess imbalance of the data. We know there are methods available, for instance, the smooth method. But in our case, we did not see much imbalance because you could see the positivity was around 59 versus the opposites. And so we did not see any much of that having any consequent effects on our results. What again did you ask? Yeah. We suspected that there may be, it may be possible that our model was not complex enough. We tried to assess the model complexity, so using AIC, Archaic Information Criteria, and all that. But it looks good, but I think that there could be other, say, latent variables available, which we could not really explore. And so we should be able to do that in the near future. Thank you.
Okay. I think, Kristen, to your question, we can do the patient-by-patient tracking over time. And we haven't done it yet, but we have the data, so it can be analyzed that way just to make sure.
Our colleagues online, you can also post your question. It will be read here. Right. We've been given additional 7 minutes to interact. So let's keep on firing.
Hi. I have a question for Seth.
Seth, okay.
For your docking, what when you prepared your receptor, was it is it like a rigid docking, or is it like flexible? How flexible was your receptor? Because I was wondering with your, I know you said that the tyrosine 35 is critical, but I was wondering whether there's no other maybe positive basic residue there that could have given a much stronger interaction.
Okay, it was a rigid docking and was eventually compensated when we did the molecular dynamic simulation, compensated for the dynamic bit of it. And for the residues, the one I presented on was for 1. So actually, that compound formed hydrogen bond with tyrosine 35. So in my slide, the latter part of my slide, I looked at residues that contributed. I couldn't talk much more on there because of time constraints. But I looked at residues that contributed quite significant energies to binding. And aside tyrosine 35, there was phenylalanine 169. I think there were about 7 of them or so. So other residues also contributed. I don't know if I've answered your question.
It's okay. And I guess as a follow-up, are you looking to maybe test some of these compounds in vitro, or do you have a collaborator who would want to do that?
Yes, please. So actually, these compounds have been purchased, and I'm not a lab person, but to the best of my knowledge, I think they are testing, or I think so, but they have been purchased, of which I'm aware of.
All right, since there's a bit more time, Francis, in the answer you provided, you talked about complexity. There's complexity of the algorithms as a possible, factor. Is it possible to consider neural networks for this application, or like are there any is there that possibility to consider that if we are looking at complexity, since we can expand the architecture to be more complex, is there that possibility?
Is that a question or a suggestion?
It's, I don't mean it to be a question, but
okay,
probably maybe you are considering it, so whether it's a possibility, it could be part, so that we compare, because of the complexity issue you mentioned.
So yes, neural network, yes. We haven't explored that. It's possible.
Thank you.
Thank you very much.
I think we have a good time. Let's unite, big applause. Good. Thanks.
Okay, so moderators, thank you so much for chairing this session ahead of time. Thank you so much. Much appreciated. Coffee is ready, or sort of almost ready. So please, as usual, make your way upstairs, first and second floor. It's only 15 minutes, so please be back here at 4:15 so that we can push through the last session for today. Okay, so did I miss something? Okay, so 15 minutes, please make your way up, and then we'll be back here at 4:15 to push through the very last session. Thank you so much.
There is no promise we cannot keep. There is no commitment we cannot honor. There is no delivery we cannot guarantee. There is only one Bethel Logistics Company Limited, a licensed international freight forwarding and clearing agent, a trusted global brand with an extensive network for the movement of air freight and ocean freight worldwide. We are the trusted name for road freight into Western and Northern Africa. We are fully accredited by Ghana Customs and Excise. We cater for all your freight forwarding, customs clearing and forwarding, cross trades, inland transportation, internal cargo tracking systems, customer freight costing and consultancy, cold chain logistics management, logistics and warehousing, quality inspection, and much more. In Accra, Tema, and Takrady, we are always available to ensure easy movement of cargo countrywide. Call us on 0204 528 900 or 0244 707 859. Email info@bethellogistics.com. Locate us in Accra at North Ridge, adjacent Accra Girls High School, or in Tema at Community 4, or in Takrady on the second floor of the GCB building. Website bethellogistics.com. Bethelog, experts in shipping logistics.
Welcome to Biotech, Africa's genomics company. Established in 2002 in Pretoria, South Africa, Biotech is a rare and unique company, as suggested by the name, and we thrive to give reliable and efficient services to all our customers. How it all began. With the concepts developed in the year 2000 to speed up the arrival of primers in molecular labs in the country, Dr. Oliver Pricer, co-founder and executive director, together with a venture team of 5 secured a loan from Swiss government and got rights to be the very first company to synthesize primers locally, and thus it all began. With capacity to synthesize over 1,000 primers a day, Inqaba remains the only company that does this in Africa. This is made possible by the Mermaid 192E. Using an ABI 3500 Excel, we offer DNA sequencing from various templates, and we offer primer design, PCR amplification, cloning of PCR products, fungal and bacterial identification, and much more. In 2007, we launched Next Generation Sequencing Services, which provide NGS projects design inputs, quality control library preparation, and sequencing workflow using either the MySeq, NextSeq, or the PacBio systems. We have more services that include SNP genotyping and methylation analysis using the Agena Biosciences mass array systems, bioinformatics solutions that complement our NGS services, and qPCR analysis. We also are a distributor of strong international brands, which includes New England Biolabs, Zymo Research, Eppendorf, Glentham, CG, Nolato Treff, Charlab, just to name a few. Inqaba boasts a responsive technical team with expert knowledge in the services we provide. We also have a well-trained engineering staff making sure that the installation and servicing of instruments purchased at Inqaba is well taken care of. A fleet of vehicles commissioned for local trips and deliveries assures we arrive at your lab on time. In the past 18 years, we have started subsidiaries in East and West Africa, and we have offices in Kenya, Nigeria, Ghana, Tanzania, Senegal, and still maintain a strong presence in our neighboring countries. All this is achieved with a highly trained and professional team that is always ready to assist. Welcome to Inqaba Biotech.
If you need a reliable procurement and supply chain partner, look no further.
Global procurement company Excelsior Global Limited is here for you.
We work across multiple industries and sectors with one goal in mind, ensuring that our clients focus on core businesses while outsourcing procurement.
Excelsior Global takes care of purchasing process while reducing all procurement and supply chain costs.
We offer a wide variety of procurement solutions connected mainly with medical, COVID-19 antigen test kits, COVID-19 antibody test kits, COVID-19 antigen test kits, virus transport media, specimen collection swab, sample preservation tubes, HIV test strips, hepatitis C test strips, hepatitis B test kits, syphilis test strips, glaucometer test strips, pregnancy test kits, hepatitis C, HCV, examination gloves, syringe, surgical gloves.
Excelsior Global makes procurement easy, simple, on time, and most important, the job is in safe hands.
We do what others don't.
Visit our office at 35 Patrice Lumumba Road, Airport Residential Area, Accra.
For more information, call us at 0559-122-092.
Mail us at info@excelsiorgh.com
or visit www.excelsiorgh.com.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, was established in 2014 as one of the World Bank's Africa Centers of Excellence. WACCBIP is located on the campus of the University of Ghana, Legon.
WACCBIP was established as one of the World Bank African Centers of Excellence. And the inspiration behind it is basically that a number of us who were at the Department of Biochemistry and the Noguchi Memorial Institute for Medical Research, we had trained outside, and we did PhDs in various science disciplines outside the country. When we got back and realized that there was not much by way of facilities to train scientists, we saw this as an opportunity to create an environment which would enable many young scientists to get the opportunity to get the quality of training that we got overseas.
This world-class research center started off as an infectious disease center, but along the line, it's added non-communicable diseases to its focus areas.
WACCBIP is providing something to local and regional students that we probably don't have in many places within the country or within the region. Facilities, financial resources, as well as an environment that encourages student learning and research. And that has significantly increased students' output in the Department of Biochemistry, Cell and Molecular Biology.
WACCBIP has a whole range of collaborators at the
In the lab, you face consistent pressure to do more with less while trying to keep up with a growing workload. Success means having instruments that you can rely on to overcome diverse challenges. The Agilent 6470B triple quadrupole LCMS, the latest version of our trusted workhorse, gives you the robustness and versatility you need to overcome these challenges. The 6470B has a proven reputation for robustness with non-stop operation, backed up by thousands of users, with numerous application notes and peer-reviewed articles to reduce downtime and increase efficiency. Enhance versatility with a wide array of ion sources to analyze any application. Ion source options include ESI, the standard choice to broadly ionize analytes of interest, AJS, which enhances the desolvation power of standard ESI for ultra-high sensitivity, APCI, for increased signal-to-noise for very small analytes, and MMI, to carry out ESI and APCI simultaneously for a complementary analysis. With the new WACCShield, you can reduce maintenance time by 92%. WACCShield lets you remove the capillary and run the cleaning cycle in approximately 30 minutes, compared to about 6 hours for maintenance by venting and disassembly. So you can gain back valuable lab time and keep up with high demands. From analyzing environmental contaminants and ensuring food safety to complex life science research, the 6470B meets your need for reliable, accurate results. These accurate results are provided by our Mass Hunter software, including Quant My Way, which enables you to personalize the software with tools that highlight only the capabilities of interest. With comprehensive end-to-end workflow solutions and application services, you can ensure proper sample preparation, expertly developed acquisition and data analysis protocols, and fully customizable reports to get the answers you need as soon as possible. Our solutions are designed to work together to provide efficiency gains that help you get more done and reduce operation costs. The Agilent 6470B triple quadrupole LCMS is unbelievably robust to increase your lab efficiency and remarkably versatile to keep up with your evolving demands. Visit agilent.com/chem/6470B for more information. Decades of innovation have brought us into a new era of mass spectrometry, with the design of Altivo, Agilent's revolutionary triple quadrupole LC-MS. At just 1/3 the size of our previous triple quad LC-MS instruments, Altivo integrates seamlessly into the stack of the Agilent Infinity Lab LC System series, saving bench space while offering the same power and accuracy of our larger systems. The footprint of Altivo is 70% smaller than that of similar LC triple quadrupole mass spectrometers, so you can triple your lab's capacity in the same space. The efficiency-optimized Agilent Infinity Lab LC series system can sit directly on top of Altivo, making it the perfect partner for high-throughput mass spectrometry. The innovations that enabled Altivo were specifically designed to maximize quantitative performance, enhance instrument reliability and robustness, and promote greater uptime. The more ions you get into the instrument, the better your MS results. With Agilent's Jetstream ion source, a superheated nitrogen sheath gas surrounds the electrospray to quickly and efficiently desolvate the spray plume and tightly cluster the resulting ion cloud, maximizing the number of ions that are created and transported into the LC-MS. VacShield features fast ion injector exchange without the need for system venting and improved diagnostics that guide the operator to appropriate corrective action, leading to less downtime for maintenance and higher throughput for your laboratory. Once the ions enter Altivo, they pass through our innovative precision-manufactured Cyclone Ion Guide. With its twisted and tapered dual hexapole configuration, the Cyclone Ion Guide provides maximum ion capture, compression, and transmission across a large pressure drop region, giving you better, more reproducible results. From the Cyclone Ion Guide, the ions continue to the first quadrupole. Better mass transmission improves analytical results. Each quadrupole assembly includes DC-only, wafer-thin pre- and post-filters, which cancel fringe field regions at the quadrupole entrance and exit to improve ion transmission. This allows for faster switching, enabling more MRMs to be performed. With the analyte selected in Q1, the ions pass through the vortex collision cell. Its innovative twisted and tapered dodecapole-hexapole geometry maximizes ion collection, fragmentation, and transmission. The high-efficiency detector offers a wide dynamic range to ensure linear responses for your most challenging analyses. The detector was designed to be easily replaced by anyone in the lab. Working together, the compression and focusing of the Cyclone Ion Guide, the improved ion transmission through Q1 with its pre- and post-filters, the optimized fragmentation in the vortex collision cell, the improved transmission through Q2, confidently monitor for and identify. Altivo provides robust and accurate reporting to expectations. Repole mass spectrometer.
There is no cannot guarantee clearing agent.
For road freight into West Trades, inland transportation, internal cargo tracking, inspection, and much more. 900 or 0244 school or instix.com. Bethlog.
Welcome to Inqaba is a rare and unique company. As how it all began, Pricer, co-founder locally, and thus it all. Inqaba remains the only using an ABI 3500 exome cloning of PCR products, fungals, which provide MySec, NexSec, bioinformatics solution national brands, AutoTref, Charlotte, we also well taken care of. It is in East and West Africa, and we have up in our neighboring countries.
If you need a reliable procurement work across multiple industries,
Excelsior Global takes
patients connected mainly with medical kits, virus transport media, hepatitis B tactical gloves.
Excelsior Global makes procurement easy at our office at 35, but mail us at info@excelsiorgh.com
or visit www.excelsiorgh.com.
14 as one of the WACCBIP
WACCBIP was inspiration behind and the Noguchi in various science facilities with an environment overseas.
It's added non-communicable.
Local and regional students, financial resources as significant.
Audience would come, and in particular this one. Annie. All right. Introduce one of the chairs for this evening's session, Dr. Anastasia Akins, to the podium. Applause. So please take it away for the last session of the day. I hope we are not too tired or overfed. All right.
So without much ado, I'd like to invite Dr. Stasis in T-cell immunity.
Sorry, the first slide. Okay, so what I'm going to do is, first of all, I know that a lot of them are here on this. Do you recognize your immunodeficiency? So he couldn't against any pathogens. So, you know, that's the what we have is full. Well, that's not the case. So, you defend the bodies from all those invasion by just bring up the most recent outbreak. You know what this is. So you have the much severe case. There you're going to see is the virus load is not controlled. What you see is, in milder case, upon exposure, adaptive immune response, this immune response is not active immune response is just uncontrollable. It's causing it. So obviously having two is so important to study. Now I'm going to introduce the iron of them. Now, obviously, I'm going to be focusing on immune. The other one is the relevance of iron and malaria parasite, because the iron malaria infection, which is interesting, and that's in your cells. And then I will not talk about malaria. This is all different types of immune cells. But obviously, the cells and getting into the cells and coming out of, and then they will bring up to the endosome. And about iron, and then some of the iron going to be stored, the ferroprotein. So this intake maintenance. So with that, studies have shown what's not known is, you know, the first experiments we did among those many different heavy metal ions. So they to look at what happened with this ion when T cells get active. We stimulate for 3 days in vitro. It's quite interesting pattern because we see higher released into the culture media. The iron we want to do was that, you know, really, and the other one is we heal later. So by using the genetically modified cells. And this is measured DFO. You see cellular iron level, the cells iron levels are drops, right? I mean that. So the ions do not really want to use. So we stopped here mice that do not have the ability. So, you know, the knockout cells has, and then we compare their behavior data. So, so the 100% from the more proliferation, you see more, their cell death is increased. Cases, knockout cell has a more cell death, much less, and the knockout cells after. Because of the IL-2 signal, then you phosphorylate to step 5. Step 5 is phosphorylated, but not that different between the wild type and the knockout. So coming is induced by this ion. So we take your membrane potential in the system, you know, rate, then you're going to see ion. Although mass is not, so either the ion is parameter, we looked at what? Andrea. So you that, this shows the something like this is a cytoplasmic ion. When this is going up, pattern suggesting that ion is ion, and then if you compare them, barely any induction. So in other words, I'm using the microscopy. Then this turquoise, we see huge morphology. And we see cleared by having generate a lot of new glycolysis. This is biochemistry 101. From the glutamine, they also generates by another pathway. It's called the knockout system pathway. So clearly, cell for the mitochondrial integrity, where the T cells are really regulating mitochondria function, as opposed to cell division work. Here? Yeah. So,
ones that gets the iron out, transports it out of, exports it out of the cell.
Import into the mitochondria. Export system is even less understood. So now we are working on mitoferrin expression in the water. You mean normally?
Yeah.
So I guess normally when cells need either go to the ferritin protein to get stored or getting out to the ferroportin released into the circulation.
Mitochondria, looking at the fact that the mitochondria has a threshold of iron that it can carry at any particular point in time.
So we think that there are two mitoferrins, mitoferrin 1 and 2. Know about those yet. So by the way, I feel you're going to be working on this project.
Loss from the cells when the cells rupture for iron overload into T cells?
So the genetic system we are using is a heme and by the HO1 enzyme, and then iron is using other machine. With the malaria, I have no idea. Yeah. But again, when red blood cell it has only 30 days phase, iron homeostasis and regulation is much better studied than in T cells.
Around, and I'm just wondering whether that does feed into your system at all.
So there are a lot to do here. I mean, we did look at a lot of other elements.
Statistics and any iron in the
It's totally fine. So there's a clear.
Thank you.
Very much.
And we're starting with Tomitope Wilson. Wilson, you take it over.
Me. I know because so my name is Temito PhD, and what I'm going to do today is to share with you some parts of my PhD work, history of organisms that require both immune response to adipose tissue during malaria. So, organisms which are immune mediated and they are different from immune tolerance, but essentially what targeted at the pathogen itself. But what they do is that they limit, let's put it that way, that the infected host survives the infection. So as a way of demonstrating, let's say humans or mice that get infected, the pathogen load on the mice are able to recover, or the humans recover, and then they're fine, right? While some would survive. So the mechanism that actually operates here is what we call the disease tolerance mechanism. So is that when a host gets infected with either parasites or viruses or fungi or bacteria, it's sensed by the innate immune system, and when that sensing happens of molecules like TNF-alpha and IL-1 beta and IL-6, and it includes things like lethargy. Actually, what sickness behavior leads to is what we call infected host gets infected with a bug. It develops sickness behavior. Hosts do not want to eat any that it needs to use in a manner that is a sick human, and this will be a sick mouse, another animal in the world that get infection. So what I've told you is that this infection leads to we need to mobilize from its storage organs. The primary storage organ, and it is because it takes one molecule of fat, which is called triacylglycerol. So what we did to be able to delve into the question is that we took advantage of infected red blood cells and we inject it into a non-infected mouse. And this way we can bypass that. What you can see here is that as the infection progresses, around the peak of infection, which is day 7 of these infected animals, one would see that sites have reduced dramatically, and the mass of that is into very tiny adipocytes. And this is true for another adipocyte. So it seems as if infected mice with very, very little adipose tissue. So of whether or not you're infected, this loss of adipose tissue could just be because the mice are starving. The cohort of mice, which is what is called perfed mice here, we made them as anorectic in a manner we are controlling for the effect of anorexia. And if one does that, and again check the adipose tissue, that they do not lose the adipose tissue. What we know from the literature is that adipose tissue lipolysis requires a 3-step process where the moves one free fatty acid from the triacylglycerol, and now you have diacylglycerol, and another enzyme which is called hormone-sensitive lipase takes it. In the end, you would have free fatty acids and glycerol. One would see that the proteins that are involved in breaking down the fat, they are all upregulated. So this rather gets phosphorylated in a manner that, in the fatty acids that one measures in circulation. And that this is caused by the first enzyme under the premise that if it's a, what we see is the following, that mice that are unable to that here, seeing that they have bigger adipose tissue, you can look at the adipose tissue and see that that is released from the adipose tissue at the peak of infection, which is fatty acids. So suggesting that this infection, so this will be a wild type mice, which is the white circles, and being able to break down the adipose. And this is important because in the beginning of the presentation, I talked about resistance mechanism, which takes mobilize the adipose tissue depots, is actually a disease tolerance mechanism. Infected mice that cannot mobilize the adipose tissue, they have a. What I've told you so far is that breaking down the fats is essential to survive malaria. But then the next question is, how do you know from the literature is that under stressed states, including starvation or being exposed to cold, there is the sympathetic arm of the autonomic nervous system. And when these guys get activated, the action potential leads to the release. So we wanted to see whether this is what is happening. So what we did was the following. So we generated a mouse to run through it. So all neurons that are sympathetic, that are Th positive, we were able to kill them in a map. So, right, they will have Parkinson's disease because sympathetic neurons are also in the brain. But what we did was that we make the killed. And if you do that, you see that the mice are now unable to induce the enzyme involved in depots, and they now also die when they are sick. So that leads to adipose tissue lipolysis. And this is, again, a disease tolerance mechanism because it doesn't negatively impact by this gene, which is called adipose tissue triglyceride lipase. So I'm not going to go into the conclusions because I'm out of time, but now I'll be interested in taking that.
So we'll take two burning questions.
Yes, I would say for the sake of privacy, the answer is yes. Does that answer your question? So there are some medicine, some hold medicine actually, whereby before coming for the treatment, and then they give them some parasites that stays in them for 7 days. Whether or not we can just make people sick in a manner that we want them to lose weight, but the principle is the same, right?
Okay, there's a hand.
Responses, but I want to know if there are some immune responses that could lead to this lipolysis in the.
I'm going to start from the second one. What brings about anorexia in groups like TNF alpha, IL-1 beta, and IL-6? These are potent. Showed that we just published a paper recently that during the process of mutations today. So if you take heme by itself and you inject it into a that is used to induce anorexia, to induce lipolysis during infection, the answer is yes. But I'm not presenting that data, but they are rather increased. So, which means that these two pathways, neurons and immune sensing of the pathogen, answer your question.
Yeah. Thank you very much, Wilson.
Thank you.
We have.
Okay, so I'm in Anabiri, and I will towards placenta malaria is an issue of grave malaria infections. But what it should from West Africa, and then prevent malaria in pregnancy. The folic acid supplementations, preventive approaches, critical of which gene called VAR2-CSA. Now, VAR2-CSA, villus space of the placenta, usually leading to intrauterine. So, if you look at the antigen, what has been shown is that vaccine induced there's a clear issue of limited cross-reactivity. To help explain placenta, what happens is that a woman who is exposed, but however, if they become pregnant for the first antibodies that protect them from the deleterious effects of placenta malaria. Across pregnant women in Africa shows that VAR2-CSA specific to natural infections, various alleles of VAR2-CSA, they would lead into protection. And we describe this as applicable to all the various allelic variants, and this we describe as true cross-reactivity. But so to be able to address this, what we have done is that is capable of analyzing single-cell cross-reactivity. Critically in this assay, bodies that are released from antibody secrete to different or double cross-reactive or triple cross-reactivity to do the multiplex analysis of several analyses. We get the tagged and that express VAR2 CSA, and that is the IT fact, the individual domains with specific. So we needed two types of cells from in to the Abra Asebu Kwaman Kettle. In these numbers. This is using a gradient freezing system. Then they are using them in the flow response assay. Now, typically, the spots for each of the you merge, then you have cross-reactive, and this is that you would have antigen-specific SPAV8. So this is for each of the donor pregnant women. So to be able to normalize this data, we usually normalize the B cell responses across alleles for each of the BC, even though there was a little bit of some background. Look at if these memory B cells vary, dominated by variant-specific responses and less of cross-reactive antibodies. Then if you compare it across, what have we shown? I've shown that if you take pregnant women who are exposed to domain, there are more responses to the immunodominant domains, that's DBL3 and DBL5. Basically develop a handful of broadly cross-reactive team. I'll thank the team of researchers that helped me in my fieldwork, Ejayao, Reverend, and then the health personnel and work with.
I think the vaccine fails to generate cross-reactive antibodies, but a natural infection does.
Well, so basically what we have seen is just reactive antibodies, but what we are looking at is, do we have cross-reactive antibodies in natural population?
Oh, I was a bit, I was at least very interested to see what we are presenting. For having tested this vaccine.
Is that?
I'm saying that I was really interested to know what would be your conclusion for having tested the vaccine you were talking about.
What would, I don't get to it. Okay. My conclusion as to what?
Yeah, no, in terms of the cross-reaction.
Yes.
I'm a bit surprised you are very firm in terms of your conclusion saying that there is no cross-reaction with vaccines.
Yes. So
and now, let me just go ahead on that.
Okay.
As I was saying, I have been lucky to be the one who has tested that vaccines.
Yes.
And I know that we are still in evaluations to see if this vaccine have this cross reaction with naturally exposed malaria itself.
Okay.
But I'm seeing here that you are very firm in your conclusion.
Okay, so both clinical trials, they themselves admit that there's limited cross-reactivity for both of those vaccine candidates. And these are two individual domains. Now, what is the caveat here? Now, before these studies, what we knew is that the minimal binding domain is quite conserved. But recent studies using about 2,000 genomes have realized that there are actually some variations across the ID1, ID2 domain, across countries and across geographical areas. So how did these candidates represent variations in our populations that we don't know? So at least to the candidates we have tested, there's limited cross-reactivity. Does that reflect in real natural immunity? If you're looking at native proteins, we can't tell.
Okay, well, let's, I think I'm still not in line with you because what you are saying is very exactly the theoretical approach that people have. But on field, the reality may be different and seem to be different.
All right,
That's what I'm saying, because the evaluation is still there, and even now we are still following the women who has been vaccinated. And we haven't yet, I'm not going to give any result here as
all right, so I agree, because this, if you look at the PAMVAX, it was in Benin and Germany, and that the clinical trials have done. But critically, some of our researchers at the Center of Medical Parasitology, they thought that the model that was used for the clinical trial wasn't a good model. So they tried to establish a primate model, and the results is consistent with what we see. It produces IgG that only recognize the monoclonal. It does recognize the heterologous parasites, but the rate of recognition very, very limited. So the degree of single-cell allelic cross-reactivity is really very low. But so the idea now is which domains do we have epitopes that would elicit broadly cross-reactive antibodies? That I think we would have to look into. But well, let's see.
Okay, let's see. I'm saying let's see.
Yeah.
Yeah, so we'll see. There'll be more discussions after here. Thank you so much, Godwin. Right. So our next presenter is Nicholas Ewuku-Ofe.
Hi. My name is Nicholas Ukofe, and today I'll be presenting on the effect of metformin and epigallocatkin 3 gallate combination on migration and EMT-related stemness in prostate cancer. I think today we've had a lot of infectious diseases, but then still prostate cancer remains the second most frequent case of cancer recorded, which is second to lung cancer amongst males. And this was recorded by GlobalCan 2020. Aside its high frequency, it is also the fifth leading cause of death, and it's also second to stomach cancer. I know that there's been a lot of solutions or chemotherapeutics to it, but then most of these chemotherapeutics are limited in that there is often development of chemoresistance to most of these drugs. And these chemoresistances are often related to metastasis. And research has attributed chemoresistance to a subpopulation of cells within the tumor. And these cells are referred to as the cancer stem cells. One may ask, what are these cancer stem cells? These are cells that are present within the tumor, and they are capable of renewing themselves and differentiating into tumor of more aggressive behavior. So when they differentiate into tumors of more aggressive behavior, they employ mechanisms such as drug efflux and epithelial to mesenchymal transition. And this epithelial to mesenchymal transition is seen when these cancer cells, they downregulate their cell-to-cell adhesion molecules, as we can see over here. And after downregulating the adhesion molecules, they assume fibroblastic natures and gain a highly migratory potential. When they assume this highly migratory potential, they upregulate their mesenchymal genes. After upregulating their mesenchymal genes, they invade and intravasate into circulation and are carried to secondary sites, and this consequently results in metastasis. So from this, we can see that any target or any therapeutic that we seek to develop should at least be able to target metastasis and also the cancer stem cells that are present within a tumor. And I know the process of discovering a drug is expensive and it's also time consuming. So we can resort to improving the present therapeutics that we have and also improving the ones that we have. So repurposing drugs such as metformin. Most of us know metformin to be a first-line anti-diabetic drugs. And if it is used, it's used in treating type 2 diabetes. It acts in that it stimulates the cells to pick glucose from the system and reduces the level of expression of glucose levels in the blood. It has also been found to potentiate anti-cancer activity in that it induces cell death and also inhibits proliferation. It is isolated from the leaves of the goat rue plant, and recent research has shown that metformin enhances the anti-cancer activity of polyphenols. So what can we do with this? We can look for a potent or a recent polyphenol, and this polyphenol has been found in a widely consumed green tea. Green tea contains 4 catechins, but then the most potent one, which mediates most of its health benefits, is epigallocatkin 3 gallate, which for this presentation we call EGCG. EGCG mediates anti-diabetic, antimicrobial, antioxidant, but for this research we are looking at its anti-cancer effect. So looking at its anti-cancer effects from literature, it says that EGCG targets the transforming growth factor signaling pathway, which is also important to epithelial to mesenchymal transition. So if you combine EGCG and then metformin, there could be a way of targeting epithelial to mesenchymal transition, as well as migration and the stemness of D145 prostate cancer cell lines. For this work, we chose D145 prostate cancer cell lines because they are androgen receptor negative and also have a high migratory potential. And our aim was to identify the effect of this combination on migration, EMT, and stem gene expression in the D145 cell lines. To begin our work, we had to culture cells. So we cultured cells, and we cultured cells, we stimulated them, or we treated them with our various treatments, that is the metformin, the EGCG, and then the cisplatin, which for this work was our positive control. It's a conventional anti-cancer drug. And then we subjected it to the MTT cytotoxicity assay. And we read the OD, and the result we obtained from this showed that the metformin, the EGCG, and the cisplatin showed a time and then a dose-dependent cytotoxicity. So this represents the graph for the metformin, the EGCG, and then the cisplatin, which we can see that for the periods of 24 hours, 48 hours, and 72. So let's look at the last one. The last one, you can see that for a period of 72 hours, the metformin concentration of 10 millimolar was very cytotoxic, and the same was seen for EGCG and then cisplatin. For the main focus of our work, we sought to investigate the effect of the combination on migration. So for any assay that we choose, we should be able to see the migration of the cells. So upon seeding the cells, we created a wound. You created a wound and then monitored the closure of the wound following treatment of the metformin, EGCG, the combination, and then cisplatin. And we recorded the closure rate 48 hours post-treatment. And the results we obtained. So from this, you can see that if the cancer is bad, we know the cancer is bad. So if it's highly migrating, we can see that it will be, the wound area will close faster over here. And if it's not highly migrating, after a period of, say, 48 hours, the closure rate will be quite high. And this was seen in that without treating, the wound closed faster, and with treating, there were different degrees of closures. But then for the metformin and then the EGCG combination that we saw for the metformin and the EGCG, we saw that the closure rate was very slow, and after a period of 48 hours, the closure rate was, say, 50%. The individual treatment of the epigallocatechin-3 gallate, which for this work is EGCG, and the metformin showed some degree of inhibition on the cell lines, and the cisplatin also showed its activity. So from this, we can say that the stimulation of metformin and then EGCG had some role in inhibiting the migration of the cells. So if they could inhibit the migration, then we resort to looking at the gene expression levels, what genes are implicated. But then we know that epithelial to mesenchymal transition helps them to move. So we resorted to investigate the mRNA expression levels of these EMT genes. The genes of interest were E-cadherin, N-cadherin, SLAG, and TWIST for the epithelial to mesenchymal transition. E-cadherin is an epithelial marker. So if a cell down-regulates its epithelial marker and then assumes a mesenchymal marker, it means they want to migrate and be more aggressive and cause metastasis. And then the N-cadherin represents the mesenchymal marker, which is the bad thing which we don't want to see. And the SLAG and then the TWIST are regulators of the EMT. The SOX2 and the OX4, we will see it in further slides. So from what I've said thus far, we can see that there's SLAG, there's TWIST1, there's E-cadherin, and then there's N-cadherin. When SLAG expression goes high, there is suppression of E-cadherin, which is the epithelial marker. And when TWIST goes high, there is expression of N-cadherin, which is a mesenchymal marker, which causes the cells to move faster. So all of these three are what we seek to down-regulate with our treatment. And then this is what we seek to maintain or sort of up-regulate it in order to prevent the epithelial to mesenchymal transition. Of interest was the one in red. So we can see that the stimulation of the metformin and the EGCG could suppress the expression of the SLAG gene, and it also had little to no effect. It had little to no effect on the N-cadherin. And then for the E-cadherin, we saw that it was appreciably expressed. So if SLAG was down-regulated and E-cadherin was up-regulated, then that is what is the good thing. That's the good thing that we want to see. And we saw that for TWIST, which was down-regulated, N-cadherin was not downregulated, which was quite not expected, because if slag and then twist are regulators of epithelial to mesenchymal transition, they should be able to upregulate N-cadherin when they are expressed and downregulate N-cadherin when they are not expressed. So from this, you can see that in our prostate cancer cell line D145, suppression of the slag and the twist did not necessarily downregulate the expression of the N-cadherin. In investigating the effect of the D145 on the stemness of the cells, the ability to regenerate when treated, we had to ascertain that, yes, indeed, we had some degree of stem cells within our treatment and then within, for the cultures. So we saw that most of the D145 cells highly express CD44 cancer stem cell marker. And so we went ahead to find the effect of the combination on stem cell markers such as SOX2 and then OCT4. The results we saw here showed that the combination of metformin and EGCG downregulated the stem cell marker SOX4, implying that as a combination of EGCG and metformin has the ability to downregulate the expression of stem cell markers, so they can't regenerate themselves after treatment. The same was not seen for OCT4, but then if it was able to downregulate SOX2, which is a stem cell marker, it shows some benefits which we seek to delve deeper, So for this work, we can see that we saw that the combination of metformin and EGCG had a potential to downregulate the migration of our cancer cells. We saw that it downregulated EMT transcription regulators such as SLAG and then TWIST1, and then it also downregulated the cancer stem cell marker, which is SOX2. Ongoing works seems to investigate the protein expression levels of these EMT and then the stemness markers. And we will further investigate other phenotypes, such as the ability to invade into matrices with a D145, and then try our experiment also on other cell lines, such as prostate cancer cells, other cell lines, such as LNCAP, PC3, and 22RV1 prostate cancer cell lines, in order to ascertain the effects we are seeing. And I would like to thank my supervisors for their supervision, Dr. Anastasia Rosebud Akins, Professor Osborne Quay, the Akins and Quay lab members, and then faculty members, and finally WACCBIP. Thank you.
Can take two questions.
Hi. Can we pull back the slides?
Okay.
I just want to ask a question. Where you were showing the combination of the two.
Okay.
Go on. Keep going. Yes. No, it's not this one. Next one. Next. Yes, here. So I'm wondering, you said the combination of the metformin and the EGCG were able to downregulate the mRNA expression, right?
Yes.
But I don't see either an additive effect or a synergistic effect there. So what do you think is happening there?
So for the combination?
Yes.
Right, so thank you for your question. So for this combination, for this work, our main focus is on the combination. We are yet, can you put it? Okay.
Mm-hmm.
We are yet to ascertain other markers, but then if the combination has an effect on the expression, I believe you are looking at the individual effect of the EGCG and then the metformin, right?
Yes.
You are looking
yes.
at the individual expression effects of metformin and EGCG. If the metformin and EGCG had the ability to downregulate this particular stem cell marker, I think it is worth mentioning and it is worth investigating further than to neglect it and say maybe possibly it wasn't any synergistic effect. But then this is quite appreciable.
I mean, okay.
I mean, yes.
Okay, Prof. Chang. There's a question from Prof. Chang.
So I have two comments. Metformin is inhibitor of AMPK. EGCG downregulates glutamine metabolism. It's inhibited, another inhibitor. So you are using two inhibitors. They will downregulate the cell metabolism. And then now you're studying all these effects. So you show the cells are dying if you had a high concentration of those inhibitors, which is not surprising, right? So that's one thing is I'm not sure how you're going to make a conclusion if cells are dying or cell metabolism itself is changed. I mean, that will have a lot of other effects and impacts. Second comment is you had a CD44 staining. Without the control, negative, positive control, how can you say CD44 is high?
Okay. All right. Thank you for your question. So can you put it to my slide? All right. Thank you very much. Yeah. Right, so for the treatment with metformin, we all know that metformin is an anti-diabetic drug. So we... Okay. Okay.
Because it's targeting glucose metabolism, that's why.
Not clear now. All right. All right, thank you.
Thank you very much, Nicholas. Thank you all. That brings us to the end of our session and the day's business. Dr. Mosi, over to you.
Thank you. Thank you. Thank you, moderators. Okay, so, hello. So we've come to the end of day 2. I'm sure you've been as impressed as I have been by the plethora of talks that we've had today. I'm actually quite pleased because majority of the people who presented today were our students, past students, and actually the last presenter is just an intern. And I mean, yeah, I'm proud. Just so that you know, all the talks and all the posters that are upstairs are being scored. So we would have a best presenter, also a best poster. Thank you so much for your engagements today during the poster sessions. It was very nice to see all of you interacting. In fact, it was difficult to pull you back to the room after the poster session. I would like the same sort of engagement tomorrow, which is the last day of the conference. Also, the other housekeeping thing that I want to tell you is tomorrow we will have a live evaluation of the conference, and I'll be announcing it as and when the link is put up. We want to hear your feedback about the entire experience. We want to hear your feedback about things you think we can do better, and so on and so forth. So do have a very wonderful evening, and we will see you bright and early at 9:30 a.m. tomorrow morning. Thank you.
There is no commitment we cannot honor. There is no delivery we cannot guarantee. There is only one Bethel Logistics Company Limited, a licensed international freight forwarding and clearing agent, a trusted global brand with an extensive network for the movement of air freight and ocean freight worldwide. We are the trusted name for road freight into Western and Northern Africa. We are fully accredited by Ghana Customs and Excise. We cater for all your freight forwarding, customs clearing and forwarding, cross trades, inland transportation, internal cargo tracking systems, customer freight costing and consultancy, cold chain logistics management,